PGR research, Kerala. Madam has developed combined resistance for lead blight disease and cyst nematode in potato, which lead to the release of TNU, Nilgiri, Kufri, and Nilima varieties. She has conducted 15 explorations to various parts of Kerala, Karnataka, and Tamil Nadu, and collected more than 1,000 accessions, which comprises major collections in Vigna species, Chinese potato, Alpinia species, and small millet and crop wild relatives. Madam has described a new species, Vigna conconensis from Konkan region. Over the years, she has characterized more than 1,500 accessions of 9 crop varieties and species. She has multiplied and deposited more than 1,300 accessions of crop plants and wild relatives under long-term storage at National Gene Bank. And she has published more than 60 articles and presented papers in a seminar, a symposium, and she has written a 12 book chapter. Thank you, ma'am. And I am handing over to you and share your PPT and present, ma'am.
Thank you, Dr. Subhash. I would like to share my screen. The screen is visible?
Ma'am, it's showing screen is sharing. Yes, ma'am. Now it's visible, ma'am.
Is it okay now?
Yes, yes, ma'am.
Okay. Okay. Good morning, friends. And I'm very happy to see you all because many of them are from agriculture universities, and I think now by this time you would have been well familiarized with all the PGR activities that are being taken at NTPGR. And my center is at Trichur. It is in the southernmost region, and we are the custodians for PGR activities at Kerala, Tamil Nadu, Karnataka, and coastal regions like Andaman, Nicobar Islands, Lakshadweep, and Goa. With this introduction with our station, I would like to start my today's presentation of conservation and utilization of horticulture crops of coastal region. Uh, my slide is not moving.
Ma'am?
Yeah, it's not moving, I think.
Yeah, ma'am, it's not moving.
Yeah, just
Again, ma'am, just unshare and then share.
just a minute. It is okay now?
It's okay, madam.
Okay, yeah.
Yeah, madam, but showing another, it's not a presentation, ma'am.
Okay.
You have selected some internet, your email.
Acha. Wait, I'll join once again.
No, ma'am. Still,
Is it okay now? No?
no, ma'am. Your present first you select presentation, ma'am. You have selected,
Yeah, I'm selected, but
ma'am. Is your presentation is on open? Yes, ma'am. Right now, now it's okay, ma'am. Yes, madam. It's now it's moving.
okay. Sorry for the introduction, friend. And okay. It's not moving. I don't know why. I don't know why it's not moving.
Earlier it was moving, ma'am.
I'll
Yes,
do.
ma'am. It's moving.
Okay, likewise, diversity in the land, I mean the plain land, you can see the diversity in the coastal regions also. So you will be seeing plain terrain land and hilly region, marshy lands, everything. This is just a photograph to show you the diversity habitat present in the Andaman Island, diversity habitats in the coastal Tamil Nadu region, etc. So my presentation flow will be like what are the collections we have made from coastal region, what are the diversity we have collected and maintained at our region station Trichur, and how it can be utilized or how it can be used by you, or how the crops can be familiarized to you, like that. So my presentation will be full of photograph and some new crops and species that you are not very familiar, so that you can utilize in your career for your as a as a species to be utilized, or to be used in the breeding program, or to just conserve it, etc. Okay. So coming to the collections we have made about some totally 2,480 accessions from coastal regions of Tamil Nadu, Lakshadweep, Kerala, Karnataka, Goa, and Andaman Island. Not only me, all my senior colleagues, they have thoroughly explored Andaman Island for 2 months or so, and they have collected a very beautiful collections in different species that are available in Andaman Island, and also some collection that we have made in Lakshadweep also. So we have made some 17 to 18 trips specifically to coastal region to collect these accessions of species since 2000. Okay. When you see what are the major collections we have made from the coastal region, mainly we have different variability in amaranth collection and yardlong bean collection, and we have a good variability in kokum, which is Garcinia indica, and also in Malabar Tambi. So very diverse accessions we have in our center, and we are maintaining that. Okay, this is one of I think many of you may not be familiar with this species Garcinia indica. This is commonly called as kokum, and it is a species used as a spices in the Konkan regions of Karnataka. They use this rind as a just we used as a tamarind of in the land region. They use this kokum rind as a spices in their curries and all. And this kokum seed, it is a oil seed crop also, and from the seed you can extract butter. That butter is very much useful in all this cosmetic region, which is very famous, or which is very extensively used in the cosmetic industry. And this kokum also, you can see the sarbat. The kokum sarbat is very famous. It is a rejuvenating fresh drink. So these are some of the variability that we are maintaining at our center at Trichu. And another important species is Malabar tamarind, and we have also maintained some 152 accessions of Malabar tamarind in collected from this coastal Karnataka region and Kerala. And this is the one of the major collections. Of course, we are not maintaining this species in our station. All these accessions are being maintained. The seeds are being maintained in the long-term conservation at our NGB. These are the collections made in Noni from Andaman and Nicobar Islands. I mean, sorry, Lakshadweep Island. And some of the variability you can see in yard-long bean, which are being collected from the Karnataka and coastal Kerala region. Some of the variability in amaranth, leafy amaranth species, that is Chinese spinach or the amaranthus tricolor, or some of the amaranthus dubious is there, amaranthus hypochondriacus is there. So these are the collections we are maintaining at our Trichu Center in leafy as a leafy vegetable. And this is, I want to introduce one other landrace of Chinese spinach called as Latangara landrace. This is the landrace, this has some 6 month duration, and it flowers only after 6 months, and it is not susceptible to leaf spot or rust disease, and it remains fresh, and you can have a multi-cut. This is a local landrace that is being maintained in the Kerala coastal region, and is very popular here. And we have collected some Garcinia from different species of Garcinia from Andaman Islands, like Kidia, Madrona, Kova, Gardneria, Saxifolia, Lancifolia, etc. So these wild species of Garcinia, they are also being maintained at our station. And you can see some of the beautiful collections of piper, different species of piper in our field gene bank. And we have also different collections of gingiber and alphinias and cinnamons in our gene bank. One of the major species is the Atrocarpus species, which is very popular, which is very endemic to this region. And we have collected at least 7-8 species of Atrocarpus species from Andaman Island, and it is being maintained here at this station. Okay, so these are some of the diversity photographs I have shown, and just to see what are the utilization of these species as a future crop. See, one is the Alpinia concicera. This can be a replacement for your ginger species, the gingiber officinarum. This yield is very high, and you can have a year-round harvest of this rhizome, and it is used as a masala in the tea powder also, and it has a high gingerol content, and it can be a very good replacement for the ginger. This species, I think many from Orissa, Northeast, they must be familiar. This sweet gourd. I'm sorry, teasel gourd. This is the teasel gourd. It's a very vegetable. It is a very high nutraceutical vegetable. This species is having male and female plants separately, and one you can pollinate it, and you can have vegetable similar to the momodica, but it is bitterless, and a single fruit can have a weight of about 200 grams. So one fruit may be useful for a family members of 4. So such a highly popularized vegetable here in Kerala, we are popularizing it, and it is acceptable by all the consumers here. Yeah, this is one of the sweet kokum. This having a sweetness of more than 20%, so this can be directly used as a fruit also. Same way, this Garcinia kidia is another species. This can be also directly utilized as a fruit crop. This is one other species I want to introduce as a coonful. This is called as Hematocarpus validus. It is a collection from Andaman Island, and this is an edible fruit. And you see the color of this fruit. It's just like a coon only. That is a blood. So it is a very refreshing drink. Just you can put the fruit in that glass, and you can add water. You can drink as such. So such a nutraceutical fruit has been collected from Andaman Island, Poonful. Okay? And this is another good species of Garcinia speciosa. It is a very small fruit. It can be a fruit on par with this Garcinia mangustana, that is your mangustin. So the fruit rind is small, but more number of fruits you can get. and that these accession some 7 allied accession are being maintained in our station, and this also TSH content is more than 20, and we have good variability in this Garcinia species also. And when you see what are the crops that can be used as a wild ornamental, see this Caryota mitis. This is a palm, and hope many of you may be knowing Caryota uranus. It is a very tall growing palm tree, having a duration of about more, 100 years or more. So this is a species where you can grow up to only 15 years. So it starts.
हाँ 8:30 बजे बोले थे तो तुम्हारा जाना था 9:30 तक पहुंच जाए अब वो सब अपना मस्ती आने में लगे रहते थे उसमें रमा मारते सबसे
So this species can be a good ornamental as an avenue. It can be planted as an avenue plant, and these spikes will be coming from the top. That is, the bunch of palm will be coming from the top, and after it comes up to the bottom of the tree, and then the tree dies. It is the only 15 years duration it has. It can be a good avenue plant. And another one species is Curcuma rosaceana. This is having a very high shelf life. You can keep this ornamental flower for 2 months or so. Okay. This is a photograph just to show the Caryota mitis. And this Caryota mitis is having another good utilization as a Nira. It can be used for a Nira tapping also.
टेबल इतनी क्लीन है भी
So
आके
this
देखो
is
चमचमा रहा है टेबल ऐसी गचड़ी रहती है
I think somebody is speaking there.
कल बोला मैंने कहा तुम्हारे मैंने तो घर नहीं फैला
Here, participant, please mute yourself.
हमारा तो चट्टू 3 दिन रहा ना तो फैल गया अब जरा मम्मी आ जाए फिर कभी समेटूंगी फिर वो फिर कर देगा
Okay. This is the Piper clipiatum species. This is a species, it's a shade-loving Piper species. This is these tribals of Andaman. They consider this as a very as a holy species. They tie it around their head during the holy occasion like that. And this is also a species that can be that it's having a beautiful foliage and can be used for ornamental purpose. So another species I want to introduce, Macaragga nicorbarica. This is a species which is having a leaf length of about 70 to 65 centimeter on an average. And this species is it can be an alternative for your plastic plates, like how we are using as areca. This can be also can be utilized as a replacement for plastic plate, and where the leaf can be processed and used. So we have a good diversity in the Musa species. So you see Musa balbisiana, Musa cartuvalana, Musa indandamanis, Musa acumata. These are all seed-setting bananas, where you can have a seed about 5,000 to 6,000 seeds per ban, and these seeds are all they have 100% germinability. So it can be a crop where the elephants are more in the region. Elephant invasions or elephant damages are more in the region, so we can just sow these seeds so that the elephants are protect. I mean, elephants are protected from. I mean, our lands are protected from the elephants, so that the elephant feed the plants that have been sown in on their borders. Okay, on the home borders or forest borders, whatever it may be. Okay, so this is Musa andamanensis. I just want to show the leaf and fruit, how many seeds we are having in each fruit. So this is the a view to show the naturally occurring in Musa andamanensis in Andaman Island. This is another species, Pandanus liarum variety Andamanica. This is a special food of Andaman people, where they process this Pandanus liarum and they prepare as a cake and use as a food. So this is another species, Momodica cuisine variety Andamanica. This is called a sweet gourd. So this fruit is used as a vegetable when it is very young. So each fruit will be about more than 500 grams. And the bitter is, you cannot say it is not at all bitter, but bitter is very, very, very less. But the other important use of this crop is you see the color of that fruit. You can extract a natural dye from this fruit. We have tried using this pulp of this fruit, I mean this vegetable as a natural dye. We have prepared this prepared some rice using this natural dye, and see the color of the rice, it has become red without any artificial reason. You can get a good color of red using this pulp of this I mean, Momodica, Cochin Chinese. This is a view to just to show the species and seed of Momodica Cochin Chinese. So we have also got some leafy vegetable trees, Neetum nemon. This is also a vegetable. The leaf is used as a vegetable, and it is a perennial tree. And it's at a slow initial growth. And these are the this is the species used used as a vegetable. I mean leafy vegetable by the Andaman people, tribal people. So this is a fern, Diplasum esculentum. This is also a popularly used vegetable in the Andaman. They use this vegetable for making bhajis and as a just cooked as a vegetable, etc. Then coming to the ferment foods, we have some Dioscorea collection, Dioscorea piscatorium, Dioscorea numillaria, where these species can be used as a tuber crop. Dioscorea serpenticola, Dioscorea bulbifera, glabra. Of course, Dioscorea bulbifera is also a cultivated crop where you can have a aerial bulbils. That is used as a vegetable here called as adattapu. So that is the potato and what I was telling, bulbifera variety sativa. These are the aerial bulbil that can be directly used as a, you can just use this as a, like a potato only. You can just peel off the skin and you can use a vegetable, and it's very tasty also. We have got good collections of sweet potato, where you can have a purple color skin, I mean purple color inner flesh in the sweet potato. Another species, Hibiscus subdarica, which is, I think many of you must have been familiarized with this species. Solanum laciocarpum. I saw in yesterday also in Delhi, this species is being sold as a fruit there. Solanum laciocarpum. It is a somewhat species with a sweet and sour taste, and it's it has a very rich content of vitamin C also. Okay. Then another important is Piper valicae. It is a species which it can be a replacement for your what you call pepper. So this is the wood pepper, where the stem are used as a I used like a pepper. It's called as Piper valicae. This is also, it's a robust plant, and you can have a robust growth, and the leaf and the wood, everything can be used as a spices here. This is commonly used, commonly called as Piper sojwal, and it's also it has also got a medicinal value also. So this species can be highly popularized among the and another one is Piper shag. This is Piper sarmentosum. This is also, it is also another spices where you can use this leaf as a spice. It can be used in a rasam, and it has a peculiar flavor, because we have all tasted it. It has got a peculiar flavor in it, and it has a very good what you call medicinal purpose. This is commonly used by the tribal people of the Andaman Island. This we are making, and it doesn't it is not susceptible to any disease, and it has a robust growth, and you can have a more number of leaves. It can be also used as a ornamental, everything like that. Okay. And this is a Deep Vetla we call. It is collected from Lakshadweep Island. It is very highly pungent. This beetle leaf is very highly pungent, but it is very small, and it is also not susceptible to any disease. It is being maintained at the West End. Then some of the life support species. Licula nicobarica. You see the tent they have made, I mean, thatching, the species used for a thatching purpose. See how beautiful it is. So this is one of the species. And some of the diversity in cultivated crop. We have extensive collections of taro, Colocasia exculenta. We're having 23 collections collected from Andaman Nicobar Island, where the species are used directly as because without cooking it, we can eat directly. It has not got any acidity, so it can be used as a vegetable without cooking also. So like that type of diversity in taro we have collected. And some taro species, they are used as they are stolen. It's so stolen and leaf, they are used as a vegetable. And see, this is a wild edible leafy taro. Leaves are used as a vegetable here in taro species. Then we have some species of Alocasia macroaceae. This is a giant taro collection. And we have got green and purple form. So this is another diversity in the Dioscorea alata, commonly called as Gratria. So we have these collections from Genome Savior Award winner of the Nicobaran. Nicobar, we have collected from them. This is diversity in Dioscorea. You can see the anthocyanin rich flesh of Dioscorea alata. And we have got some collections of Xanthosoma sagittifolium also, both black and white. And taco leontopentaloides, it is a how do you call potato, yeah, small potato like form. This is also used as a replacement of potato there. This is some collection of Leicerium collection. See, Leicerium, here we don't have Leicerium flowering in this region, but this Andaman collection, they are prone to be flowering, and this can be used for the breeding purpose also. So another is the vegetable banana. See, this is called beach kela. This is called Musa indandamanica. This is a high yielding, and these fruits are used as a vegetable there in Andaman. So we have a very good diversity in citrus. Citrus, we have some 7-8 species, and Citrus aurantiaca medica, Citrus limon, everything, and it's a high yielding, and it is the fruit size is very large, and with one fruit you can I think you can make 2 liters of juice in from one species. That much quantity of juice is there with in Citrus limon. So this is another one species called as Manilkara littoralis. This is used as a coastal bioshield tree. So this species we are popularizing in Kerala as a coastal bioshield tree, where this forest and coastal guard people, they have collected some many seedlings from our station, and it has been planted in the coastal regions. So some of the species can be adoptable as a rootstock. You can use as a rootstock for salinity tolerance and flood tolerance, etc. One is that Manilkala littoralis and Atrocarpus pedangularis and Atrocarpus sama. See, this is another good collection of Atrocarpus subclassa. This is it's a wild underutilized fruit species, subclassa. Mystica andamanica, and some of the species has a direct potential as has a direct use potential for domestication, direct domestication. This Atrocarpus sama, Garcinia cova, Danakarinja, all are minor fruit crops. Okay. See, this is Gomesianus, another one species in Atrocarpus where you can directly use this species as a fruit. And these are some of the species which are used as a vegetables: Atrocarpus altilis, Camanchi, and Chaplasa. So we have got also some diversity in cultivated medicinal plant. We have good collections of collections in Alpinia, curcuma, sweet basil. Okay, this is some species of curcuma, Eriginosa, Reuben, Caesia, and Manga. Caesia, this is black turmeric. It is extensively used as a medicinal purpose. Kurkuma manga, it is used as a vegetable for making pickles, etc. And we have a very good diversity in Mangifera species. The pickling type, some of the allied genera of mango, Bohea opposite, it just tastes like mango, and it's a wild mango species, and the yield is more, and it is a small fruit, and it's very juicy and pulpy. This Mangifera sylvatica is another close relative of Mangifera indica, and this is another wild species, Mangifera camptospoma. Mangifera andamanica, endemic to Andaman region, and this is Mangifera nicobarica, and we have good diversity in Agil marmalus also. This is a species collected from Andaman Island, and we have some crop wild relatives. We have Benicasa hespida variety prurian. It is a wild ash goat, and Lufa egyptiaca, Momodica carentia variety muricata, Cucumis mellow variety agrestis. Okay, this is the wild species of ash goat. This is the only wild species of Benincasa hispida subplurian. And as I said, some of the species can be tried for this tried incorporation as a for a bacterial screening. We have identified Musa indandamanica resistant to stem borer, wilt, etc. So this can be used as a triad-specific incorporation of triad in the breeding program. We have also collected some primitive brinjal, Solana melangina. It's a very small fruit, but it is highly resistant to wilt and your stem borer, fruit and stem borer. It can be used in the breeding program. This is another species, Solanum insana, and Bulmoscus mostatus subspecies Biacainensis. Can be so actually these are the few species that can be used as a new fruit species for cultivation. See, as usual, Bovia oppositifolia, sweet rose apple, Griffithii, Hematocarpus, Agil marmalus, etc. Okay. Then this is some of the diversity in the Gigipus, wild species of Gigipus, and Pantanus amarifolia is another species where this is called as biryani pata, where the smell of this leaf is like biryani. So if you just add some of the leaves in the rice, normal plain rice, this becomes as the flavor of biryani comes. Okay? So these are some of the photographs showing them. Another wild species we have collected, Etlingera pendulae. This is called a torch plant, where the flower is like a torch, and it is a beautiful ornamental plant, and we have different colors, white, red, yellow, etc. And this is Vanilla andamanica. It another wild species related to Vanilla planifolia, and this is a wild species of Cinnamonum. And so what we can, how we can, these can be utilized. This can you can try these species for salinity tolerant, so that the triad can be introduced in our breeding program to incorporate salinity resistance, and some species can be tried for rootstock, where there is a problem in the problem of soil-borne pathogens like in Solanum, where we have in Solanum, we have Phytophthora root wilt. The species of Solanum can be tried as a rootstock for incorporating the resistance, and some wild CWS can be directly used as a introduced as a fruit crop, and some accession we can study for reproductive authenticity, whether it is bisexual, whether it is what do you call monoecious, dioecious, etc. We can use in. Okay, so thank you. So at this juncture, I remember Dr. Swaminathan's word: if conservation of natural resources goes wrong, nothing else will go right. Thank you.
Thank you, madam. Dear participant, if you have any question, You can ask from ma'am. This is open for discussion. Either you can also share your question in chat box. Any question, please? Anyone? I think there are no question, ma'am.
Okay, okay,
They have
okay,
understood everything.
okay, okay.
Then, ma'am,
Somebody,
so I will propose a vote of thanks, ma'am. I, on my behalf and behalf organizers, I thank you very much for delivering such a informative lecture on conservation and utilization of horticulture crops of coastal region. Madam, you have shown diversity of different horticulture crops of coastal region, such as Garcinia, Musa species, Piper species, Curcuma, Dioscorea, Colocasia, and etc. And several others, with very beautiful photographs, and you have also given insight over their utilization. Certainly, our participant has benefited from your presentation, and I urge all the participants, if you are working on these crops and planning for your work, you can collaborate for the further research, and you can ask from madam for these germ plants, and ma'am will be very happy to share these germ plants.
yes, definitely. Somebody has asked, can we get the seeds of Caryota mitis? Definitely, you can get, no problem at all. There is a large bunch is there in Caryota mitis, and the fruits are very small and red color, and it has some dormancy, I think. So it takes about 3 months to germinate. Definitely, you can collect Caryota mitis from us, no problem at all. I think Dr. Saha from DFR, I think. No problem. Dr. Saga, you can anytime you can request.
Okay, ma'am.
Yeah.
Then here, let's see. Okay, then no question. Then, ma'am, thank you very
Yeah.
much.
Thank you,
Thank you
for
for
the
such
organization. I
info.
thank all the organizers for giving me this opportunity. Thank you, Ernest. All the best, friend. Bye.
Okay, thank
Thank
you.
you.
And friends, we have another lecture at 10:45. Then we will meet.
डॉक्टर साहब,
Yes.
सर कुछ लोग अपना माइक खोल देते हैं जिससे कि
Yes, yes.
प्रेजेंटेशन को जो कर रहे हैं उनको
Yes,
दिक्कत
yes.
आती है, जिनमें कुछ लोग हैं शेफाली है, कुछ और भी हैं।
Okay.
उस सबसे रिक्वेस्ट है कि
Please,
कृपया अपना माइक बंद रखें।
please put off your mic all the time while presenter is presenting. While speaker is presenting his or her presentation. It is a request to all the participant. At now we'll meet at 10:45, and do not put off your this or log out your Zoom link. They are from different SEUs and KVK, Central University, ICR Institutes, Central Silk Board, and 2-3 participants from international organization like ICARDA and TFRI, Tropical Forest Research Institute, Jabalpur. We have a wide group of participants, sir.
Good, very good.
And there are from different subjects also, sir.
Very good. So you are working in which division?
I am working in, sir, tissue culture and cryopreservation.
Right, good. Dr. Rakesh Singh is there.
Yes, sir. Dr. Rakesh Singh is there, but he is in a division of genomics.
Genomics.
Yes.
Good.
Earlier, Anuradha ma'am was head here in TCCU, and now Dr. Sandhya Gupta is head.
That's good. So this is being organized by crop evaluation division or
No, sir, it's actually a NVPGR training.
and
Yeah, it's a overall training of PGR management, utilization and management of PGR, plant genetic resources.
that's good.
It's not like that. It's specifically in evaluation or some specific subject. It's overall from PGR.
So people should know everything.
Yes, sir.
If not master of all, at least they should know everything.
Yes, sir. At least when anybody do the training at NVPGR, they should know, because here are 5 division are there. So they should know each and everything,
True.
at least.
This previous lecture was so informative. I was attending.
Okay.
Maybe towards the end I attended, but that was so informative. Very good. She's basically a taxonomist.
Sir,
Madam?
she's from plant breeding, sir.
Plant breeding.
Yeah. But now she's working on this crop
PGR.
group. Yeah, PGR only.
They have lot of collections from island ecosystem.
Yes, yes, yes. Sir.
Lakshadweep, Andaman, all this. Those
Because
are
this-
very, very rare.
Yes, sir. Actually, this our regional station,
Sure.
they are working on coastal region.
Yeah, that is located
UST
there also.
coastal region.
Location is also there.
Yeah, location is also there. So
That's good. We are also located in coastal area, but then we have all ecologies for a single
yes,
crop.
yes, 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 sir.
All ecology. We are working for all ecologies for a
Cuttack
single
also
crop.
we have this regional station.
Yeah. Dr. Misra is there. Yeah.
Yeah, Dr. Mishra is
Dr. Panya.
there. And how is everything, sir, at Cuttack?
It was very nice. Yesterday, 3 days back, we had an international symposium, Indian Rights Congress.
Yes, yes, yes.
Honorable
Madam
President,
President.
Honorable President was here
Okay.
for inauguration of this program.
Yes, yes, yes.
President of India,
President of India. Yes, yes. Madam.
Draupadi Murmu. Yeah.
Yeah. Dr. Draupadi.
Came here to inaugurate.
Yes, yes.
And yesterday evening only the program was over.
3 days, sir. It was 3-day program.
3 days.
Okay.
Second Indian Rice Congress.
Second Indian Rice Congress. Yeah.
I've never met you, that's why I asked.
Okay.
I used to go there, say I know many people there.
Okay.
All my friends have retired almost, but still 1 or 2 are there. Dr. Gautam is there, Dr. Ashok Kumar is there.
Yes, yes, yes. They're evaluation division, sir. Dr. Gautam is head now.
Yeah, no, I know.
Evaluation.
He called me actually. He while fixing the program, he called me.
Okay.
Told yes, you can give me.
Namaskar, sir.
Namaste, madam.
morning,
Namaste,
sir.
Jyoti. Very good morning, Jyoti ma'am. How are you?
Fine, sir. Fine, sir.
So you are back at NBPG. Earlier you were in wheat, isn't it?
I am at NUPGR only, sir.
Earlier you were in IRI or Karnataka?
Earlier, sir. Earlier, long back, there I was working on pulses.
Pulses, such. I
But
thought wheat.
here in NBPGR, now more than 12 year, but 12 year, not more than 11 onwards, sir. I am here. Okay.
So here you are looking after wheat or pulses?
Wheat, sir. Wheat mainly. But
Because I have
associated
heard you
in
several times.
maize and some minor work in pulses also. But the major responsibility of wheat, sir.
I know
wheat-
because dust testing program also you make presentations there.
CRP, NICRA, and
And,
other
yeah, I've heard you several
DVT,
times.
those things. So 80% work on wheat, 20% other crops and all.
You are wheat eating man person, so you love wheat. We are rice eating people, so we love rice.
Yes. So how are things are there?
Fine. I'll be retiring very soon. So before that, I got an opportunity to make presentation.
Yeah, it is good, sir, to hear him, because our participant also requested in between to have lectures on rice. So,
Is it? But
yeah,
are there anybody from rice group crop working in
so
rice?
rice, sir, in this group,
Yeah.
yes, sir. Some
Some are
persons
there.
are there. I don't remember the name, but some persons are there, sir.
Otherwise, it will not be very interesting for other people because a single crop and then
No, they will learn other crops also, because the group is diverse, sir. They are working on several crops like wheat, rice, pulses, oilseed, tree crops, ornamental crops. So
even
all
Dr.
kind of
Subhash,
expertise are there.
yeah, Dr. Subhash was telling me forest species people are also there.
Yeah, forest also there's. So it is totally in diverse groups.
Yeah,
sir.
right.
So no problem, means they are learning the things, and wherever their specific interests are there, so they are means more like querying and all.
Yeah, right.
So like that.
Very nice.
Mhm. So that conference was very good, no? That
Yeah, international conference and Honorable President of India, she inaugurated our program.
yeah, I saw
That
some.
itself,
Yes.
that itself added all the flavor to this congress, you can say.
Really.
Good participation was there. More than 300, 400 people were there.
Acha, more than 300.
Mhm.
Acha. Okay, good.
Nowadays, the students, they are very interested to attend such symposia.
Yeah.
For their career building up, all these things are required
The
nowadays.
students from government as well as private organization also.
Yeah, even companies also, they were there.
Companies also they are joining.
They were also there, R&D,
This
there
kind of
R&D
also.
people.
Yes, sir. Yes, sir. Yes, sir.
Madam, now it is 10:45. If you can see the people are there, then we can start.
Just Suhas?
Yes, madam.
50 participant, no?
Yeah, 50 participants. I think we can start, or we will
Yeah,
make
you
it for 2 minutes more.
Okay, no
Yeah,
problem. But
yeah.
meanwhile,
Yeah,
can I share?
2-3 minutes you can wait.
Meanwhile, can I share my slide?
No,
Ma'am,
before that.
11:45, sir. Before 11:45, so that we can have some questions, sir, from audience.
Subhas,
participants.
you can start, Subhas.
Okay.
Uh-huh.
Okay, then.
Yeah, because if
I'll
someone
start.
have question also, no? So means they can ask. Okay.
Okay. Okay, then. Sir, first of all, I welcome you, sir, for your for this training program. I on my behalf and on behalf of organizer, I welcome you, sir, and friends. We have with Dr. Bhaskar Chandra Patra, and Dr. Bhaskar is working as a principal scientist in economic botany at ICR National Rice Research Institute, Cuttack. He served as an acting director and also head of crop improvement division of the institute. Dr. Patra has been involved in the exploration and collection, characterization, documentation, and conservation of rice genetic resources for more than 33 years. So far, he has conducted more than 48 explorations program from different region of the country for different trades, and more than 5,400 accessions of rice genetic resources have been added in the national active germplasm sites of NRI Rice Gene Bank as well as National Gene Bank of NVPGR. He has also characterized the germplasm as per thus descriptors and evaluated for biotic and abiotic stresses as well for trait discovery. He has registered 41 unique rice germplasm lines with plant germplasm registration committee of ICR and VPGR. He had received letter of appreciation from ICR and VPGR for compiling the annual manual of passport data for the rice germplants collected by CRI Cuttack since its inception. Besides, he has been instrumental for facilitating the registration process of new or extant varieties at PPVFRA New Delhi, and so far 33 new varieties of the institute has been registered. He is also involved in the process of commercialization of institute's technologies patent through licensing. He has published more than 90 research papers in different national and international peer-reviewed journals, 19 technical bulletins, 18 book chapters. He has been working as a reviewer for many national and international journals. He had received award for PhD work on plant taxonomy for District Flora Scheme of Botanical Survey of India. He is a recipient of Dr. M. Brahman Memorial Award for outstanding contribution on plant diversity conservation by Odisha Botanical Society in 2015. He has conferred a distinguished fellow of Indian Society of PGR in 2018. Thank you very much, sir, for joining with us. And friends, we have a lecture by Dr. Bhaskar on management and utilization of rice genetic resources. And I'm sure our participant will be benefited from your lecture, sir. Now, sir, to you, sir, and share your slides and present your work, sir.
Is it visible?
Yes, sir. Very much visible, sir.
Please presentation mode. Yes, working.
Yeah. Thank you. Welcome, Dr. Subhas. All the participants, good morning to each one of you. So I am Dr. V.C. Patre, as mentioned by Dr. Subhas, that I am working at National Rice Research Institute, Cuttack, for last, you can see 33 years. This is my last year of service. So for retirement, I got an opportunity to interact with all the participants. So I'll be sharing my experience and whatever I have gained here in this institute for since last 33 years on single crop, that's rice. Since beginning, I am here. I joined this institute in 1990, and this is the last year of my service. So, the topic was given to me is management and utilization of rice genetic resources. So, what is PGR? This PGR is nothing but consists of seed or vegetative propagule of any plant, also include pollen, cell, DNA, or any other component which usually contain the functional units of heredity. If that particular thing is gets the units of heredity, functional unit of heredity, then it can be called as PGR. They are generally referred to as germplasm or genetic material. In fact, Sir Otto Frankel, he did coin the word genetic resources. Now, if you see the PGR, the world personality, the first personality of the PGR will come as Nikolai Ivanovich Vavilov. You know that Vavilov theory. He was the foremost plant geographer of the erstwhile USSR. He had organized more than 100 collecting missions, and his major expeditions were to Iran, USA, Central and South America, Mediterranean region, Ethiopia, Africa, and then probably with his vast experience, he gave the Pavilion theory of origin of cultivated plants. So if you see the Indian perspective, Sardar Harbhajan Singh is the first the distinguished plant experts of India, and he is referred to as the Indian Vavilov. In 1917, he was conferred with Padma Shri for his outstanding contribution in making promising germplasm introductions from all over the world, and he is one person who started you can say the foundation stone for green revolution in India by introducing the germplasm, either maybe rice or wheat or other crop. If you see perspective of rice exclusively, then if you see this R.H. Richaria, the former director for CRRA, now NRRI, he is the man who actually collected more than 19,000 germplasm from only MP, that now it is MP and Chhattisgarh, and that is one of the best, I mean, the largest collections from Raipur. That is how people know the collection is Raipur collection. And he, when he was director of CRRI, he had introduced about this several Indian exotic germplasm. One of them was the Taichung native one, this TN1, which laid the foundation for green revolution in the country, in the rice crop itself. And if you see, this normal borlag is known as a father of green revolution in wheat. But if you see rice, Dr. Henry Bichel is recognized as the father of green revolution in rice. And then if you see this PGR, what are the activities we conduct in PGR? These PGR activities mostly involve the collection of germplasm either through exploration and collection program or through acquisition. Acquisition means if you want some material from other institute, other countries, then you, through correspondence, you can acquire from them on request. That is how we build up our collection material, and that is how we collect, conserve, and utilize all these PGR. And the other activity is regular routine work of PGR is a rejuvenation of the material, whatever is conserved in gene bank, characterize them, conserving them, evaluating them, utilizing them. And after finishing all these things, we can go for documentation. And after identifying the donors or whatever trait you identify, trait discovery you make, you can share the seed material to other researchers for their crop improvement program, rice and other things. So as I was telling, these are the global centers of biodiversity as proposed by Dr. Bhavilo. So India is, of course, known, this is one of the center of biodiversity for rice crop, and Southeast Asia, China, mostly China. And this is how rice originated from this part of the world, India and Southeast Asia, Southeast China. And then center this, how can you confirm this? This archaeological and historical evidence indicate that the northeast part of India and these mountainous regions of Southeast Asia and Southwest China is the primary center of origin of Oryza sativa, while the primary center of origin of Glaberrima is the delta of river Niger and Mali, Africa. I mean to say here that there are two cultivated species of rice. One is Oryza sativa, the other one is Oryza glaberrima. Oryza sativa originated from in Asian portion, where on the other side of the continent, Oryza glaberrima originated in Africa. That is exclusively for Western Africa, endemic to that region. And if you see this, our Odisha tract, the Jaipur tract of Odisha, once this had the highest number of diversity in rice, and that is how it was considered as the secondary center of origin of NRRI, origin of rice, and this was published by Dr. Ramya and Ghosh in 1951, when this institute was established and NRRI was established in 1946. First and foremost program was to collect this diversity, rice diversity from the entire country, and that is how they identified this center as the secondary center of origin for this house ecotype mostly. And you see the origin, how this rice crop came to this earth. If you see evolution process, this cultivated rice was nothing there, everything was there 10,000 years back, maybe you say, all over, everything was wild. And then people throw their domestication process, all these things, they domesticated these wild rices, and they developed these land races were there. And then maybe 10,000 some years, and then from land races, the farmers, the people who domesticated them, they themselves selected the best material, and that is how they become the farmers' variety. And only this green revolution started, maybe pure line selection came only 125 years plant breeding when it started. The breeders, the scientists, they developed, they made pure line selections from these farmers' varieties, land races, and that is how improved cultivar came. And last 30, 40, 50 years, in 1965, when this green revolution came to for this country, and several modern breeding materials were developed. And now about from this, more than 1,400 rice varieties have been developed in this country. So these are some of the, graphically, I wanted to mention how these things have developed through evolutionary process. And if you see this, this, as I was telling then, how this Oryza sativa, this cultivated rice came to this earth. There was nothing, as I was mentioning, everything was wild. So one hypothetical species was mentioned, that Oryza perennis was there, everything was wild, and perennis means perennial. One single in water bodies, the one Oryza perennis was there, that is not distinct species. But in the Asian context, if you see that the evolved as a perennial rice Oryza rufipogon, this is a distinct species. From Oryza rufipogon through natural selection on nature, this perennial species was evolved as an annual species that became an Oryza nivara. And then from annual nivara species, this all our cultivated species in Asian region, Oryza sativa started. Originated through human selection. And from human selection, this Oryza sativa, again, it became 3 ecotypes. One is Japonica type, one is Javanica type, and Indica type. Indica is a pantropic, it is a cultivator everywhere. But Japonica, if it is mostly in temperate regions, mostly in the northern part of this China, Japan, all these things, that is how it is Japonica. And Javanica is mostly Indonesia, Bulu type, you can say Indonesia, Jamata. And it is also called as tropical Japonica. So if you see the African context, this Oryza longistimata is a perennial wild species. From there, through natural selection, Oryza barthi, annual wild species, was evolved. And from barthi, through human selection, Oryza glabellum was evolved. This evolution is very recent, maybe it can be only 3,500 years old in West Africa. Whereas the Oryza sativa, around 6,000 years back, maybe it has originated. So this is what I wanted to mention here. So as I was saying, in genus Oryza, we have 25 species. Out of the 2 are cultivated Oryza sativa in Asian region, and Oryza glabellum is in Western Africa, you can say. And besides that, we have also several potentially useful traits to this adapted to Oryza genus. We have Oryza coarctata, we have Hygroryza, Zizania, and Zizania has also several other traits which are being now utilized for rice improvement program. If you see the rice journey, as I was mentioned here, this is the center of origin of cultivated rice of Oryza sativa. From here, from the northeast part and southern part of China, this is the primary center of origin of cultivated rice Oryza sativa. From here, it started its journey towards south, and it went to Indonesia and to Australia, and part of twins were also given, even taken to Sri Lanka, and then it also went to after Madagascar. From Madagascar to this new this Madagascar, this slavery system, when it was there in the America, these people were migrated from this place, and they had taken this rice Oryza sativa to this new world. That is only 350 years back, only rice Oryza sativa is being cultivated in eastern United States of America. At the same time, if you see the Oryza glabellum, the other cultivated species, this is very much endemic to this part of this continent. It was evolved about 3,500 years back, but it could not spread much because of its very stature, but poor characteristics. It's a drought tolerant, it's almost short duration variety. It cannot cope up except this upland situation. So even though it was taken to Latin America, it did not spread there also. It had its own established only in this part of the country. That is how it is endemic there. And based on the genome analysis and degree of sexual compatibility, the rice species are grouped under 11 distinct genomes. They are A, BB, CC, BBCC, CCDD. Like that, it has diploids, tetraploids, and this. And on the basis of crossability and ease of gene transfer, the status of gene pools like primary gene pool, secondary gene pool, tertiary gene pool are made. And then in genus Oryza, we have 5 gene complexes like sativa complex. This is mostly primary gene pool. Officinalis latifolia complex, this secondary gene pool. Mariana complex, tertiary gene pool. Ridley complex, tertiary gene pool. And unclassified complex Oryza coarctata is again the tertiary gene pool. And these are some of the species, all the species what I mentioned. I'm not going to read it. These are the species Oryza alta, Australiensis. Australiensis, as you know, this is only mostly, this alta is found in South Central America. It's a tetraploid. Australiensis is found in tropical Australia, Bartha in Sub-Saharan Africa, Brachyantha in Africa, Ichinjeri, Glumipetula, grandiculums, they are all in African and Central American species. And Oryza granulata, latifolia, longiglimis, longistaminata, meridionalis, mariana, minuta, nivara, officinalis, punctata, rhizomatis, ridley, rufipogon, sclecterian, coarctata. This is the coarctata is the perennial species, and then it is found only in tridal mangrove tridal forests. So these are some of the wild species. You can see this Oryza alta, officinalis, minuta, brachyantha, ridley, Rufipogon, longistamina, they have useful traits like instant disease resistance. They have the ability to tolerate abiotic stresses as well. And several cuties have been identified from all these species for yield. So that's this C4-ness, like as you know, the rice is a C3 plant, but some genes are available in wild rices we can give to C4 plant also. And they have also drought tolerance and heat tolerance, all biotic stresses. They look like grasses, but they are phenotypically inferior. But very valuable genetic resources are conserved, preserved, inherited from these wild species. And some species we have already started working on this. This some of the Rufipogon species we are maintaining in our field in net houses. These are some of these species like Brachyantha nivara. All this is, and these are some of the primary gene cries, as I mentioned already. We have identified them, we have characterized them, we have found several donors in a wild species like drought tolerance in this particular excision, BPH resistance in this particular excision, and then yield enhancing traits in Rufipogon, and similarly this particular variety where excision is also giving YSV and yield, yellow stem borer resistance and yield traits. And if you see in nature, this work, natural interrogation is also happening in the nature, like wild rices when they are found, they themselves grow up in the natural ditches and field bonds, and they naturally make process with the cultivated species, and through natural interrogation, this wild rices, the weedy rices evolve. That is how this process of evolution starts. And then there's weedy rice. These are very, nowadays they are very, you can say obnoxious weed, you can say very difficult to control them. You see their gigantic growth when they have strong dormancy. Even if you try to eradicate it, it will have a 3 month dormancy to 5 years dormancy. So even if you do it transplanting, all of a sudden some weedy rice will appear. People in Odisha, they call it as Balunga, and some other places they call it as Oryza spontanea. And this farmers field, you see how much weedy rices are there. They become a mixture like thing. And if you see in India, we find only 4 wild races. They are Oryza nivara and Oryza rufipogon and granulata and officinalis. The only 4 species you will find in India. But then in India, in world context, you will find 23 wild species and 2 cultivated species. And then the utility, as I was telling the wild races, what is the utility? Why should we conserve and preserve them? Wild races, in situ conservation, why it is required nowadays. If you see Oryza nivara, this particular accession from Basti region of Uttar Pradesh, this had given when in the southern part in sometime when International Rice Institute came and they started their work, that southern part of southern Asian continents, Indonesia, Malaysia, all these places, a particular disease, this Tunguro rice virus, that devastated millions of hectares of rice land. And then there was no donor available in cultivated rice. And then people started searching gene from wild rice. And incidentally, one collection of Oryza nivara, an annual species, which was collected in Basti region of Uttar Pradesh, they had given resistant source to grassiston virus, and none other than our J.S. Cruz. That he also identified this particular persistent virus. With this background, several eerie varieties have been developed, like several series of varieties have been developed. And if you see the Oryza rufipogon, again, it is a collection from China. It had given the cytoplasmic male sterility source of CMS line, and then that hybridized program started in China and international races through this source only. Similarly, Oryza rufipogon has also been a source for elongation ability in Erie's deep water rice breeding program. And if you, as I was telling, these are the 2 dominant wild rice species we find in India. Oryza rufipogon is perennial species, Oryza nivara is annual species. And these are some of these characteristics. I'm not going to read them in detail, but then the major differences I'll put in this in pictorial form. This is a phylogenetic analysis that shows that most of the A genome Oryza species are perennial and photoperiod sensitive, while except Oryza nivara and Oryza bharti, they are annual species. If you see, this is the rufipogon, is a perennial species. When we put them in the net houses, this grows, this doesn't die. This is annual species Oryza nivara. And then if you the panicle, if you see in rufipogon, this is mostly excited panicle and then very large. Whereas in nivara, it's an annual species, and panicle is very much compact and inserted panicle. And see the anthers in rufipogon, these are anthers are very large and indecent, and pendant. Whereas in Nivara, there is very small, dehiscent, and always upright. That is the thing. And in rice, you will always find 6 anthers. And if you see in the net house, when we keep it, it never dies. Even if some of the tillers are dying, but again simultaneously new tillers are appearing. At the same time, if Nivara is an annual species, it never survives. In completion of 1 year, once flowering, fruiting is over, the plant dies itself. Of course, in nature, through stubbles, they appear again in the nature. So some confusions are always there in the taxonomy in genus Oryza. There are people say several confusions. I am not going to detail because it may not be so much interesting to you. And few of you are working on rice, so I'm not going to tell. But one thing people say always, they were in Odisha, they say this Oryza Fatua, Oryza Balunga, all these things, they are nothing but our Nivara. Similarly, last few years back, somebody collected a material from Andaman, Nicobar Islands, and he gave a new name of Oryza Indandamanica. It is nothing but a synonym for granulata. It doesn't have taxonomically, it doesn't exist, or it is not a valid species. Similarly, when this Jaipur tract of Odisha was surveyed, they found some wild rices in Koraput region, Jaipur tract, and then they gave a new wild rice named Oryza jaipurensis. This is a misnomer, and it is nothing but a variant of Oryza sativa. Like that, several other things are there. In Oryza Malampuja, another wild species that was collected from that Malampuja river in Kerala, and they gave the name of Oryza malampujensis. Similarly, if you see these now these two particular this Oryza sativa species, one is cultivated in Oryza sativa, the Oryza species, all the Oryza species, and the only exception is Poteris, that is now Oryza quartata, that is found only in mangrove tidal regions. I'm not going in detail. So there, as I was mentioning, there are 2 cultivated species, Oryza sativa and Oryza glaberrima. These are some of the traits by which you can identify how distinguish sativa and glaberrima. Oryza sativa is, if you see the habit, it is essentially perennial. If you harvest the crop, some tillers are there, some ratoon will be appearing at the end of the season. Definitely, that is how they call it as a perennial, essentially perennial, not strictly perennial. But glaberrima is an annual species. Leaves are here mostly pubescent, here often glabrous. And then most of the panicle characters, if you see in sativa, it is panicle type, whereas glaberrima, there is no secondary branching. And several other things are also. Distribution sheep sativa is always a cosmopolitan, whereas glaberrima is endemic to West Africa. And if you see Oryza sativa, always on less. At some places in Basmati, other things you find on also. But here in glaberrima, always on less, like 2 other wild species of granulata and sclecteri. If you see it, ecotype different on cultivated Oryza, as I was mentioning, there are 3 ecotypes, or indica type, japonica type, and javanica type, and they are different. They have been distinguished taxonomically, and they have a habit. Everything is there. If you see the leaf characters in indica, they are broad, light green. In japonica, they are very narrow. In javanica, they are stiff and light green. And grains also similarly. This japonica I find very mostly round, is in indica slender, and javanica is very thick. And if you see the Saturnian habit also, in indica it is very easy. In japonica, it is very, very difficult. It is practically, it is not traceable. You have to almost pluck and pick each and every spikelet. And similarly, the javanica is also low. Now again, a new rice, that's Nerica rice, it has come very recently. As I was telling, this new rice of Africa, Nerica is nothing but a new rice for Africa. It could have been a bumper. This Nerica rice was developed by this WADA and IITA, and the West African Rice Development Authority in Africa. They developed this new rice for Africa because they found that Oryza glaberrima is slowly getting endangered and getting extinct. So what that made is that took this rice, Oryza sativa, was introduced in Africa in about 450 years back, and they started in late 90s. This particular still Monty Jones from in Sierra Leone, he made a process with Oryza sativa with glaberrima, and he developed this new rice for Africa. This new rice, descended from an endangered species, Nerica rice, is helping Africa to feed itself. Yet this opportunity would have been lost if glaberrima had gone extinct. Somehow this glaberrima is now being utilized, and new rice for Africa, several varieties have been developed. And now as Africa has become self-sufficient almost, you can say, in this rice production. Through this, again, another trade-based rice, you can say medicinal rice, Nava rice. This is again first indigenous rice of Kerala, which is now known as medicinal. World's first rice is known as medicinal rice. That is how it has got the GI also from IPR. And this is exclusively found in the Palakkad region of Kerala mostly. And this is estimated that about 300 tons of medicinal rice is used annually for Ayurveda treatment in Kerala itself. And studies found that Nava has increased level of protein and amino acid in the organically grown seeds. So this is how it is also considered as one of the very important medicinal rice. It should have been developed as a baby food and health product to save this wonder rice from extinction. And then unfortunately, this unique rice is now cultivated only in 30 acres of land in the state of Kerala, because the farmers are not taking up in spite of high demand, owing to the poor marketing network and the absence of remunerative price. So this germplasm should have been given much more priority and importance in the marketing level also. Now coming to this rice diversity in herbarium. If you see this herbarium, what is the importance of herbarium? If you see, the herbarium actually provides information respect to locality, habitat, flowering, fruiting time, and many more, which helps the researchers in confirming and determining the identity of taxon. Most of the must be visited. We have several herbaria in the country, but the major one, which is called Central National Herbarium in Kolkata, and the particular variety, this particular herbarium seat, but 5252, it was collected from 24 Parganas on 1908, but and people now said that this false spot disease of rice. This was recent disease evolving in the country in the rice crop. But when I studied the herbarium seat in the Calcutta National Herbarium and Central National Herbarium in Calcutta, this particular axon is having that false spot balls. That was long back collected, about 100 and 110 years back. So this is not a new disease. It is already there 100, 115 years back. So that is how you get an idea about this evolution of this diseases, pests, and other things through herbarium also. Similarly, another example of herbarium study, I can tell you one species was there, Oryza sclecteri. This was described by one scientist as sclecter in 1910 in an endemic area of Papua New Guinea. He collected in 110 years back. He had collected from Papua New Guinea. It was endemic to that place. However, after visiting this major world herbarium, studying the passport information, only 1 or 2 plants could be collected from the same site. After 82 years, Dr. Van, when he joined International Rice Institute, 1910 to 1994, almost 84 years after he collected another set of this material, that gives an idea about the importance of passport information, also the herbarium material. That is how that has not been properly utilized so far. This wild species might be having several other genes which can give you resistance, tolerance, or biotic, abiotic, whatever may be. Some genes may be available from that source also. So similarly, I had studied several. These are not much of your importance, but the scientific importance is there. How minute it was a misnomer. It was collected by some people, and it was given some other name. But on rice pioneer scientist, he studied it, and he found that this is not Oryza species, some other species. Similarly, some other species are there. And if you see the herbarium, as I was telling, this Oryza granulata, this is mostly, you can say, a forest species. This was the oldest collection in the mini herbarium so far, 1835, more than 150 years old collection. So if you find this major explorations in rice, we have only 3-4 collections. One is Jaipur Botanical Survey, JBS, that was collected in 1955 to 59, and only nearly 1895 accessions were collected. And similarly, Assam rice collection, it was made in 67 to 72. And here this was collected mostly organized by IARI, by Dr. Professor Swaminathan, S.B.S. Sastry, S.D. Sharma, and they had collected about 6,630 accessions. This is one of the best collections of the country which are being utilized for rice program. Unfortunately, the Jaipur Botanical Survey, even though it had given several traits, but it could not be utilized much or evaluated much, or probably that time they did not give much importance for their utilization. But however, all these materials are being conserved in National Gene Bank as well as National Gene Bank of NRRI. Similarly, in 1975, one NCS collection, national collections from states were organized by the seed by the NRRI. And very recently, 20 years back, NATP, there was ICAR had a program of a National Agriculture Technology Project from 1999 to 2004, where we were also involved, and we were given the responsibility to collect the wild rice species. And we collected about 500 accessions of all wild species, mostly the Nivara and Rupi program. Now, if you come to characteristics aspect, these are some of the unique traits. You can see unique descriptors per descriptor. These are some of the unique descriptors. The stigma, mostly you'll find pink, but here it's purple, white, and other something. This is an enclosed panicle. If you see, this is a traditional variety. You can say landrace. The center panicle is found within the leaf sheath. It doesn't get come to the limelight. That panicle spikelets, they mature within the leaf sheath itself. That is how they're called. They are known as enclosed panicle. And that matures, as I was telling, this is popularly found in Chotnagpur belt, that it's a mostly tribal belt, that the people say that Sati, because it matures in 60 days, it is grown in upland, mostly unbounded uplands. And there are a few different characteristics as per descriptor. As per, if you see rice, we have a DOS descriptor, about 62 characters there. And we take all the characters, morphological traits, about 30 traits. We characterize them as per the descriptor. These are some of the epiculus and other things, epiculus and sterile lemma, all these things are there. These are the various non-characteristics. These are completely on, less here short, partially on. 1 or 2 grain fine spikelet you find on. Here it is short and fully on, big tones. Here long, partially on, and it is on, long, completely on, fully on. Similarly, if you see another trait, unique trait here, this is a clustered panicle. The plant, you see the panicles are clustered. I mean, spikelets are found in clustered form. Similarly, in all colors, this is straw color, brown color, green color, golden brown. And similarly, this black color rice, green color rice, and then lemon, color purple, and then see very small grains also. And the seed coat color, when you dehusk them also, you'll find several unique seed coat color. And this is what the diversity says about this, our unique rice plasma, rice variability maximum. This in case of basmati also you find these are traditional indigenous aromatic rices, ASG rice, aromatic short grain rice, where these are the basmati rice. The unique characteristics of basmati is, see, when it is cooked, it expands, a linear expansion there, not the whole expansion, only linear expansion, about 2 mm, 2 centimeter, expansion goes up to 2 centimeter. This is again a characteristic green rice, Chotnagpur area, and Raipur area. This is one of the accessions we have also collected from there. And if you see, when we characterize all these materials, national genomic materials, in our own collections, we found that we collected one accession, we found that the 1,000 grain note of this particular accession, it is about 50 grams. Can you imagine one panicle to 1,000 grain weight? Test weight usually remains within 20-25 grams. But in this particular accession, we found 50.4 grams. At the contrary, if we collected one collection from Assam, that is known as Kunkuni Juha. It's a scented variety of aromatic short grain rice, and it is 1,000 grain, which is only 8 grams. So this is the variability from 8 grams to 50 grams. So much of variability in rice diversity. Now, some of the unique accessions here again you find in nature the bounty. You can see beautiful examples. This is within one spikelet you find 2 grains, double kernel. This is again a unique accession. Similarly, this is of course a particular variety. In India also we find this Malaysian accession. These are exoticals here. The panicle has not matured. The spikelets have not matured, but still that has started germinating. This is called as vivipary germination. This is undesired character. And when we collect this hill rices, mostly these are collected from Arunachal Pradesh. Mostly the hill rices, they are very small. And these are some of the collections we made from for our aromatic short grain rice in Odisha. These are the ASG rices, very fragrant, very small, very useful, and then very premium price we get from these varieties. And these are some of the variability in different colors, mostly the aromatic short grain rices. Some are on, some are not on, different colors. So much of variability in color, size, shape, on, everything. So much of largest variability you find. And now coming to the evaluation aspect of this rice germplasm, This we do it is for screening against major biotic and abiotic stresses, and that gives us this. We identify the donors for particular trait, and that is how we have been identifying several traits over the years, and for different varieties for different traits. So I'm not going in detail because this is not much of importance to you. As I was telling you, the wild rices also, I will explain to you how important this particular excision was for giving this resistant virus to resistance to persistent virus. And I was telling 23 EDD variety, higher varieties have been released after I-28, basing on this particular excision. And they have become so popular worldwide. And these are some of the trait-specific genotypes we have identified for different traits. And then I'm not giving details, so I will just give them. Similarly, wild rices also, you will find two species, whereas an Ivara rupicogon, quite two predominant species of in India, and several collections were made. We made in this institute in collaboration with sometimes with NBPGR also, and some we have evaluated them very thoroughly, and we find two excisions, particularly 100374 and 100476. Even though they are wild rices, but they have the trait of this drought tolerance. This particular two, they were collected from West Bengal, and they are identified as a drought tolerant line species, drought tolerant accessions, you can say. When they are grown in the field, most of the plants are withering, they are drying, but that particular accession is still alive and alive. Similarly, for submergence, we have the different facilities for screening the submergence tolerance in rice germplasm. Here these are the particular tanks we have developed for screening. We put the plants once it is there. We put the water completely submerge it, and after 15 days we desubmerge it, and we search which are the plants which have survived. One or two plants, after maybe several accessions, you will find one or two accessions will be there alive. That is how we make screening, and that gives us the tolerance or submergence tolerance level. Similarly, for some of this particular variety, you can see after screening about 10,000 germplasm accessions, under different control conditions, several donors have been identified which have given better performance than international FR13A. This particular Swarna Sawana variety was developed from the Swarna, giving this FR13A. This is the variety. This is the landrace which had given the Sawan gene, which was incorporated into Swarna variety, mega variety of India. And that is how Swarna Sawan is now given, is also widely accepted in low-lying areas, and where stagnation, it can stagnate, withstand, and that submergence tolerance is there for 14 days, even up to 21 days, 3 weeks tolerance is also there. That is how these people, the scientists, are working in different aspects. Similarly, this is when we took this Swarna variety and Swarna Sawan variety. Both are same, almost same, but this particular single gene from our land as FR13A, that had given this particular varieties. Everything survived after 25 days, after 21 days of submergence. This Swarna variety, even though it is a mega variety, it could not withstand the submergence. This particular only one interference of only one gene that has given so much of confidence to the farmers, to the scientists, and the yield loss is not at all there. Similar for salinity situation in India, we have intercoastal belt, peninsular India, and then we have Bengal, Arabian Sea. Everywhere the salinity is always a problem, and then we have found the salinity tolerance in particular variety, particular accession, and then we have screened, and now slowly saltol gene being incorporated in different varieties. And also we have, can I continue? And after identifying all the donors, we have a system in ICR and BPJ to register them as unique germplasm. And at this institute, we have already identified several from donors, and so far we have registered about 41 germplasm. Somebody is talking. Can I continue?
Yes, sir. Sir, continue.
Yeah.
I muted him.
Yeah, okay. Thank you. So as I was telling, 41 germplasm, we have already identified different traits, mostly some of the abiotic stresses, mostly. Some are just maybe salinity, maybe drought, maybe other things. At places also we have gone for biotic stresses like VPH. This is a particular insect, this VPH and other things. And also some gallmeas and other traits also are there. This even recently, very recently, we got one high protein content rice from Assam, ARC collection 10075. This is the collection we made in 70s, like 1970s, late 70s, 50 years back. Now that particular accession is giving this high protein rice. We identified this as having high protein content about 15%, Usually in rice, you get 7 to 8% of protein content. And then we transferred this particular content, made the several process, and we developed a variety like CR-Dhan 310, which is the first Indian rice variety which contains more than 10.2% protein content. And so in the process, we have developed several other biofortified rice varieties. Now coming to the utilization aspect, this, as I was telling already, given this aspect about how this TN Taichung Native 1 was introduced to country, and then Green Revolution was started through this institute for rice purpose, and then first two high-yielding variety of the country. Two high-yielding variety. Now there are 1400 high-yielding varieties of rice, but these two first high-yielding, Jaya and Padma, that in 1962-68 they were released first two high-yielding variety of rice. Similar, several miracle rice varieties have been developed. Now, if you see the wild rice, as these are the traits, or as a longistamina, this is again a story is there. This is particular, this old Raja longistamina, this is a perennial species. It was identified as it has the source of XA21 gene that gives the BLV resistance. That is what I am telling, this BLV resistance, this was identified, and then BLV resistance, in particular XA21 gene was identified from this source, this Horizon longistamina. That's what I mean to say, no horizon, no wild rice is unimportant. So the people, this conservation, the rice workers, everybody, biologists, they should conserve all the habitats of wild rice habitats. And then there were scientific comparison there. And then how this was, I already told the story, how this was identified in this institute from longistamina, how they identified this new source of resistance to BLV. As I was telling, this is again a variety we have developed from a male sterilite, this first kind of in India. The CR-Dhan 312, it was released and notified with CBRC. It has a widespread, but it is now been released for 9 states of the country. Similarly, as I was telling, fast high protein rice CR-Dhan 10, basing on this particular IIRC collection, this particular 50 years after collection, we got the utility of that particular germplasm. And then CR-Dhan 310 was released. Similarly, CR-Dhan 311, it is high protein content 10.1%. And here in addition, you find gene content is more than 20%, 20 ppm. And with mega variety Swarna background and Nobin background, we are transferring them. And then recently we have last, during 2 years back, I mean, our Honorable Prime Minister released this variety as one of the best biofortified variety, CR-Dhan 315. It is having the high gene content of more than 25 ppm. And it is a high yielder also, 5 tons, and also several things are also there. So I'm not going to into detail this nutritional rice or this high protein Swarna also. It is also released now as Swarnanjali. And these are some of the biofortified rice varieties here. Then Prime Minister was dedicating this rice to the nation on the occasion of FAO's Platinum Jubilee celebration in 2018. And then if you see, these are some of the VPS resistant varieties we have identified. And this is the color, see the BTH, brown for how devastated they make to the rice crop. The entire crop is devastated here, if you see. And that particular gene we identified from one of these donors, we can send our Salkati-1 landrace, and that is how permanent this was restricted, and the new varieties have been developed as CR-Dhan 317. Now coming to golden rice, I'm covering almost all aspects of the rice diversity. That's what I take mainly to take, but then I'll try to complete it in time. This is the golden rice. This was long back, it was developed, but due to biosafety reasons, it has not been released so far in the country. But recently, it's Philippines, it was released, and it was developed by the Syngenta, the private company in America. And then, you know, this is a biofortified, it has a beta carotene there. When beta carotene is consumed in rice, it gives a vitamin A correction, vitamin A additional addition. Now coming to the conservation aspect of the PGR, in the rice PGR, if you see in global scenario in International Rice Institute, they have about 132,290 accesses of an international gene bank. At the same time, in NBPGR, we have National Gene Bank. And then we are also nowhere less in now 100, almost reaching 110,000. So if you see, the majority of collections were from either from Raipur or from our institute. So about 110,000 accessions we have so far conserved for single crop rice. Similarly, in IIT also, International Institute of Tropical Agriculture in Nigeria, they have about 20,000 accessions. China also, they have 50,000 accessions in National Gene Bank of China. USDRS, they have also larger collections. So even that they don't cultivate rice, about 12,000 germplasm excess are also conserved. And very recently, if you see in Japan, they are also accumulating the germplasm for safety of this mostly tropical, temperate and tropical Japonicas. See, these are some of the collections from we are made and collected and deposited in National Gene Bank, about 36,000 from this institute. So I'm not going, this is our small gene bank, and we have a MTS, medium-term storage. We maintain a temperature of 4 degrees centigrade, humidity 33% more than 35,000 germplasm we have conserved here. And if you see the recent development, we have a SGSV comes global seed vault in the Arctic region of North Pole. It was established and installed in 2008, and it conserves about 0.8 million germplasm. It is being managed by the Norway's Department of Agriculture and Global Crop Diversity Trust, and it is being funded by Bill and Melinda Gates Foundation. It has a capacity of holding several germplasm excess as a safety deposit. It doesn't require any seed, manual seed, energy. This outside temperature is mostly 40 minus 40, and India recently joined this treaty, and they have started depositing about this 2014-25 excess. And very recently, last year also, some germplasm were conserved and deposited there. And we were signatory to this treaty, and as 59th nation, we submitted and deposited our germplasm, started depositing our metal there. Now, if you see in India, in our institute, we have developed a reference garden, a wild rice garden, what is a species reference garden, where we are maintaining all the complexes, Sativa complex, all these 5 complexes. And then, of course, this is, of course, recently we have developed, and it is being collaborated with NVPGR West Center, Cuttack. So this is the recently we have developed a reference all wild rice species. All these colors are different you can say complexes, whereas a sativa maybe sativa complex, meridional complex, meridionalis complex, classified. So all these complex, officinalis complex, all complexes are there. This is the only exception in rice is this is a tidal rice, mangrove rice. This is called a Oryza quartata. Mostly you find in Sundarbans and then one Bhitarkanika mangrove forest. But incidentally, 2 years back, I had visited Andaman Nicobar Island. I didn't find a single quartata occurrence of that species there. Maybe some other, maybe climate change. I mean, something is there, but I could not observe single plant there. So in situ conservation, we have also taken up in our Jaipur region, Koraput region. This is the Bunda tribe, one of the very primitive tribes of the country. They are still conserving and cultivating their own traditional varieties. That is how, in one way, they are conserving our land resources and our precious wealth, genetic wealth. And some people are still cultivating, they're collecting these wild rice seeds for their own consumption as an offering to the deity and other purposes. That is how they collect through the winnow. And because these wild rices are very shattering in nature, they take the winnow and collect through this winnow. And these particular wild rice seeds, they make it some type of sweet dish and offer to the deities, their local deities. This is again a quarter of this we are maintaining in ex situ conservation in our net houses. And see, this is the one of the almost it's time now, another 10 minutes there. So I'll take a few minutes here. This is species structure, you can see. This is steel structure. This weighs about 6 tons. This is what the awareness program was made by one of the scientists, one of the architect, you can sculpturist from Japan, named is Mitsuake Tanabe. In the year 2000, he donated this strong structure, a huge structure of wild rice. This is a wild rice seed. It's the longest one. Sorry. And this is the plumule. And this is the radicle. Radicle goes to the root, to the soil. This emerges as a shoot. And this is the one. This entire seed he has depicted in the steel form, iron and steel. And that is why he made that in Japan. These wild rice habitats are being degraded day by day. In India also we are facing by expansion of the roads, most of the ditches, roadside ditches, ponds, trees, everything is getting vanished nowadays. That is why he wanted to make awareness among the people, the students, the researchers, the farmers, all stakeholders. They should not destroy these habitats. Other day will come, we will not find anything in the nature, only in gene bank where we restrict the evolution. And that is how new land races or nothing will be evolved. So that is why he wanted, not being a scientist, he wanted to donate to this, since he would make the awareness program. made awareness by donating this. Not only to Indian NRI, he has similar thing he has given to International Rice Research Institute also. To whosoever visits there, they should make feel that this importance of this seed, because they have given so much of important traits to this wild development of wild this cultivated rice. Even but several exceptions have been utilized. Now coming to this aspect, germplasm sharing, this is another activity. Even though we in a national gene bank, we have several germplasm accessions, about more than 100,000 germplasm, rice germplasm, but their mandate is not to share to any germplasm for their crop improvement program by to any researchers. But at the same time, the mandate of the MTS, or we are the national what is that germplasm site that NAGS, something like that. So we are also sharing to TN. Recently, about 20 years back, we had given some wild rice to Potassic water to TNA, Tamil Nadu Agricultural Coimbatore. When 2 years back, I visited that institute. They are showing, sir, from you we had collected, and we are still maintaining. That is how this seed sharing nowadays has become very useful for the researchers across the country. Now coming to this sharing aspect, again, I am telling that about 3,000 accessions of rice and everywhere. We get indents from different rice researchers, we supply them. Of course, we maintain some MTA material transfer agreement. And from NBPZ headquarters Delhi also, we get indents from different scientists, rice researchers. We also supply to them. And then with this small, this is our rice germplasm field, you can see. So this is how we have been managing the rice germplasm in this institute. Thank you very much.
Thank you, sir. Thank you very much for such a nice presentation. Dear friends, you can ask a question or any query if you have. Dear participant, you can ask, or you can put your question in chat box.
If there is any question, please raise your hand. If there is no question, it's over, sir.
Sir, hello.
Yes, somebody is asking.
Please introduce yourself.
Sir, basically, myself is Dr. C.S. Pandey from Jawaharlal Nehru Krishi Vishwavidyalaya, Jabalpur. Sir, my specialization is in horticulture. I am scientist in horticulture. सर मेरा इंटरेस्ट ये है कि जैसे राइस में इतने जर्मप्लाज्म इकट्ठा होते हैं, कलेक्ट किए जाते हैं, तो जो यूजफुल होते हैं वो रख लिए जाते हैं, बाकी सब कैसे उसको फील्ड जीन बैंक में रखते हैं, कैसे करते हैं सर?
We have access to conservation. National Gene Bank is there. This is orthodox seed. No need to go to field gene bank. You can keep it -20 for 100 years. -20 in National Gene Bank, they have a long-term storage facility.
राइट सर. और जब जैसी जरूरत होती है उसमें से यूज कर लेते हैं.
Yeah. But unfortunately, in rice germplasm, I was telling 110,000 accessions are there, but hardly 3 to 5% have been utilized so far. Never mind, but that is conserved for future, for time to come. Maybe after 100 years, you may need any germplasm that will be useful. So never mind that we are wasting our germplasm, keeping them in the National Gene Bank, wasting our resources. One should not think like that. You never know. As I was mentioning about that ARC collection, Assam rice collection, 50 years after its collection, we got a first high protein rice variety of this to this country.
Okay, sir.
So importance of each and every accession is there. One cannot discard by just that it is not of useful. So we should discard, we should not waste energy, money, everything to that accession.
Right.
That way one should not think like that.
Okay, sir. Okay.
Sir, another one question from Dr. Sheetal Patel. Sir, can please list few germplasms for drilled paddy and how will I get those germplasms?
I got the question. So seed sharing is another mandate of this institute. We are not refusing anyone to supply the seed. You give an indent to our director requesting, sir, I need this type of germplasm, and we have a system of material transfer agreement, and we will supply the seed if it is available in our gene bank.
Any other question? Then If there are no questions, then I may propose a vote of thanks. I, on my behalf and on behalf of organizers, I thank you very much, sir, for delivering such an informative lecture on management of and utilization of rice genetic resources. Sir, you have beautifully explained the journey of rice from center of diversity evolutionary process, wild rice as a source of biotic and biotic stresses, and biofortified rice. Sir has showed diversity in the rice crop with beautiful photographs and given insight over the utilization. Certainly, sir, our participant has benefited from your lecture, and I also urge to the participant, if you are working on rice breeding and you need important germplasms which have a source for high protein content and yield, and as far as and also biotic and abiotic stresses, you can collaborate with the sir, and you can ask for the germplasm, and sir will be very happy for sharing the germplasm. Thank you very much, sir.
Thank you very much. Once again, I thank all the organizers, Madam Jyoti Kumari, Dr. Subhas, Dr. Gautam, and of course, Director and PGR for giving me this opportunity. Thank you very much.
Thank you, sir. Thank you. Namaskar, sir.
Namaste, namaste.
Thank you, sir.
Now I may request Dr. Kuldeep to introduce our next speaker, Dr. Jyoti Kumari, madam.
Yeah, thank you, Dr. Subhas. And I would like to take opportunity to introduce speaker of this lecture, Dr. Jyoti Kumari. Although you all are very familiar with her, but I hope you all are not knowing much about her research, research contributions. Is this in slideshow? Is it visible?
Yes, visible.
As we know, Dr. Jyoti Kumari is the principal scientist, plant breeding at ICR, National Bureau of Plant Genetic Resources, New Delhi.
The slide is not visible.
Okay.
It is not visible.
Let me.
It's not visible.
Yeah, just wait.
Can you try again?
Let me stop sharing, and
Again, share.
some problem with my PC, but I'm trying. No issues.
Yeah. No, it's fine.
Okay. So visible?
Not when you are doing slideshow.
No, sir.
It's not visible.
Again, there is a. Let me again try. Abhi?
Sir, slide is visible, but
Ah,
not in presentation mode.
Do not.
Otherwise, I am briefing. No issues.
Okay,
Whenever
sir.
I am keeping in slideshow, it is not visible. Okay.
Okay.
No problem. Presently, Dr. Jyoti Kumari is the principal scientist in plant breeding working at ICR, National Bureau of Plant Genetic Resources, New Delhi. She obtained her master's and doctoral degree from Indian Agricultural Research Institute, one of the premier institutions of ICAR, in the discipline of genetics. She joined as ARS scientist, plant breeding in Indian Council of Agricultural Research in 2005 and worked at ICR IIPR, Indian Institute of Pulse Research, Kanpur, and also Division of Genetics, ICR IRI, New Delhi. She got appointed as senior scientist, plant breeding at ICR NBPGR, New Delhi in 2010. Her research interests includes varietal development, PGR management of wheat, maize, and pulse crops. Her major scientific contribution includes mega characterization and evaluation of around 22,000 wheat germplasm, development of the core set and mini core set, from which the entire wheat germplasm conserved in Indian National Gene Bank. She has identified several accessions with unique traits, including high popping accessions in permillet, waxy lines in maize, high grain protein, high gene content, heat tolerant, many more. She was involved in the development of lentil and barley core set also. She has also identified tutorials for terminal heat tolerance, salt tolerance, spot blast tolerance, seed size, and seedling root architecture traits using a genome-wide association strategy. She has contributed in augmentation of rice and wheat germplasm from the drought and heat-prone areas of Madhya Pradesh, Rajasthan, and Gujarat. She has also conserved more than 9,000 germplasm of wheat and maize in medium-term storage and regenerated more than 5,000 wheat accessions for long-term storage in National Gene Bank. In addition, she has organized around 10 germplasm fields exclusively on wheat and barley and supplied more than 11,000 wheat germplasm for research purpose. She has to her credit more than 120 publications including 82 research papers, 1 book, 18 book chapters, and 1 catalog. She has also developed 4 varieties in lentil and mung bean and registered almost 30 genetic stocks with PGRC of ICAR. She is a fellow of Indian Society of Genetics and Plant Breeding and recipient of Dr. Harbhajan Singh Memorial Award by Indian Society of Genetics and Plant Breeding. As a chairperson and the part of teaching activity, she has also guided 6 MSc and 1 PhD student of Division of Genetics in school, IRI. Although she is very silent worker, but her contribution, enormous contribution made by her is not silent. So with this, I would like to welcome our speaker of today's lecture, Dr. Jyoti Kumari. Madam, please share your presentation.
Yeah, thank you.
अब वो धूप के इधर जाती जाएगी इस तरह
Thank you,
जाएगी।
Dr. Kuldeep, for the nice.
Dr. Mangala Parikh, please mute yourself.
Whether the slide is visible?
Yes.
Yes, madam.
Yes, ma'am.
Okay, thank you, Dr. Kuldeep and Dr. Subhas for giving me the opportunity. Whether my voice is audible?
Dr. Subhas, you can mute because I'm
Yes,
not.
yes, I am muted.
host.
I am muted.
Okay, thank you. I'm not host of this meeting.
Okay, so very good morning to all the participants. So in continuation to the training programs, training sequences, so I thought that means I should cover some aspect for strategies for efficient utilization of plant genetic resources. So here we have talked several times what is germplasm, what is plant genetic resources. I will not go into the much detail here. So plant genetic resources, when genotypes of particular species collected from different sources and origin, when it has got some potential value after evaluation, it can be used in plant breeding program for develop new cultivars, improved cultivars. So you have to use the plant genetic resources. And in the gene bank, plant genetic resources comprises of different biological status, already talked, but again, I am just so it contains light lines, then hybrid and their parental lines also, varieties, those contain obsolete varieties as well as extant varieties, present varieties, then genetic stocks registered with the plant registration committee PGRC, and then we have land races of all the crops and wild relatives for all the crops conserved in the gene bank. Now coming to the importance of germplasm resources. So this is the plant genetic basic raw material for genetic improvement. It is the reservoir of useful genes which we are using in the plant breeding program after knowing their potential after several years of evaluation. Then this PGR is critical component for food, nutritional, environmental, livelihood, as well as household security. As NBPGR is the partner in one JEC project. So there we are working on the livelihood and household security. So with farmers, this farmers and NGO are the partners, and how the germplasm can be used for enhancing the income of farmers, for enhancing their livelihood security. So like that way, it is driver of the agricultural sustainability and economic growth of the nation. So using germplasm in the better way, great way, it will increase the economic growth of the nation as well. And further, so due to climate change scenario at present, this germplasm, mostly land races and wild relatives, these are the important sources of abiotic stress tolerance genes. So climate resilient genes are also there. And these are the beautiful examples for green revolution genes. That is DGU gene from TN1 Taiwan for rice and Norin 10 dwarfic gene from Daruma, Japanese landrace. So which boosted the production and quantum jump led to the green revolution across the world. So these are the different management activities for PCR: collection, acquisition, regeneration, characterization, evaluation, then documentation of characterization evaluation data, which we share with the partners and stakeholders, and then distribution to the inventors and various researchers for use in the utilization program, breeding program, so that we can develop climate resilient and nutritional improved varieties. Now here, I'm showing the how genetic resource evaluation will be used. So first we are augmenting the germplasm in the gene bank through collection as well as introduction. Then we characterize and evaluate large number of gene bank accessions, and we develop core and mini core set for better and efficient utilization, as less number of germplasm will be easy to evaluate. Then we develop based on their evaluation at multi-location or hotspot locations. We develop trait-specific germplasm, and then some of them we are registering as a genetic stock with the PGRC. And then we do gene identification program. Further, we mine the germplasm through validation and all, and then we put some genomic tools here, mapping, GWAS, and those things. Then it is used for the utilization purpose in the breeding program. And here we also do pre-breeding from the wild relatives as well as from the unadapted land races. So in pre-breeding, we use both the things. And then it is used for the variety development. Now coming to the status of PGR resource utilization. So globally, around 7.4 million accessions are conserved in different gene banks, 1,625 gene banks. But it has been reported that less than 1% germplasm have been utilized in crop improvement program only. So because most of the breeders, they are having some working collection, and they are continuously using that working collection only. So that's why there is a narrowness in the improved cultivar or variety development. So there are several studies for the seeing the genetic base of the cultivars in different crops, and it has been found that so only few working collection has been used in the variety development. So what is the basic reasons for low use of germplasm? As gene bank collection huge number of germplasm, so large size of collection is there. It is very difficult to evaluate large number of collections. So there is low use. So that's why core and mini core came into the picture to enhance the utilization. Then even if you utilize, if you will not put the data in the proper form, in the catalog, in portal or publication form, people will not get aware about their economic importance of the germplasm. So we cannot use also. So it is very important documentation of the germplasm. Then restricted access to the germplasm resources. If we are not making available to the people, then also they will not make use. So germplasm valuation should be there, documentation should be there, and then we have to keep the seed in sufficient quantity that we are doing already, and we are distributing to the inventors and researchers whoever was asked the seeds from us. Then there should be proper linkage between the gene bank as well as the users, stakeholders. So then it will be better utilization of the germplasm. And at present, NUPG are doing this in the good way with all the partners, NARS partners. Then lack of infrastructure, that is the big hurdle for the comprehensive evaluation of PGR. Because for good phenotyping data, we have to have good infrastructure so that we can generate good data, because phenotyping is too much cumbersome. Genotyping is doing mechanically, but phenotyping is mostly manual. So you should have a good infrastructure facility to facilitate high throughput phenotyping and all. Then the big with wild germplasm interrogation, this is the problem: crossability and linkage drag. So these both are the problems. In that, here also many interventions are there, use of embryo rescue techniques and other techniques. By that way, we are introducing the wild germplasm. Now, so how to enhance utilization in the breeding program? So first one is the ability of the seed material in gene bank, I already told. Then documentation of the characterization and evaluation data in the portal or published information or catalog anywhere. Then we should do the comprehensive evaluation, like multi-location evaluation and high throughput phenotyping using phenomics facility. And then now UAV assisted phenotyping also it is being done. So we should use those things to generate the more reliable and precise data. Then we should have the knowledge of gene pool and breeding system, so that in better way we can maintain our germplasm based on their pollination behavior, self-pollinated, cross-pollinated, so that our maintenance and genetic integrity of the germplasm will be maintained, and we can do regeneration in better way. And then development of, so here I will talk for this highlighted one, blue color, development of core, mini core,
trait-specific germplasm, and reference it. So these things we have to do. Our gene bank perspective, gene bank curator, we have to do this. Then only we can efficiently utilize the germplasm. Then focused identification of germplasm strategy should also be. I will come into the detail later. Pre-breeding, I will not discuss into much. Genomic assisted utilization, I will just give the brief account of this, what we have initiated for the wheat at NVPGR, some account only, and then distribution to the partners, and through germplasm field day and other things. And now already talked by Dr. S.D. Upadhyay in the beginning itself. So core collection. So Frankel defined a core collection as a limited set of accession representing maximum diversity with minimum repetitiveness of a crop species and its wild relatives. So here the main purpose is that we have to capture maximum diversity and we have to keep the minimum redundancy. So germplasm should not be redundant and it should be maximum diversity. So that will be the core set, so that we can capture maximum diversity. It should not lose the but what happens during the course of time of evaluation of core collection, we are taking 10% of the solution. I'm going to the next slide. So, for suppose we have entire collection. So in the core collection, we have to take the 10%. It is not exactly 10%. It can vary. It can vary depending upon the crops. Suppose if it is cross-pollinated and often cross-pollinated crops, which is having more variability in diversity, then 10% may go to 15% or 20% also. And if it is self-pollinated crop and relatively the germplasm is having more duplicates and those things, then it can be less than 10% also. So it can be even 5% also, 8% also. So it is flexible. It is not that it should be always 10%, but it is around 10%. Then from core collection, we will again develop mini core. So mini core will be again 1% of the total core entire collection. Core collection ka 10% and entire ka means 1%. So here also our purpose is that we cannot screen everything, and resource is limited, manpower is limited. So we have to efficiently make the core collection to capture the entire diversity and specific traits. Here, one thing is there that core collection, it is based on the neutral theory of alleles. So here, generally 70 to 80% alleles are being captured. So one drawback of core is there, some rare alleles it does not capture. So we may lose rare alleles. So in that case, we have to develop trait-specific reference set also. That in that case we do for evaluation for a specific trait. Suppose some specific insect resistant or pest resistance. So we have to evaluate for that particular, because core collection is developed mainly on, suppose we are using agromorphological data. So it will be a kind of multi-purpose core. So it can be used for further comprehensive evaluation of all the traits. But certainly we may lose for some rare alleles here. So we have to take care of those things, that for which purpose we want to use this core. Then there are different approaches here, like for data sets. Here we can use qualitative data, quantitative data, then passport data we have to use certainly here. And then biochemical data also we can use, biochemical code, then based on marker data also, code is being developed. Then for making code is nothing but is a sampling strategy. We have used different kind of sampling strategy, like random. So we have to random sampling, means we have different classification of the different groups, and from there we have just random. We are not putting any criteria. Proportional strategy means in proportional we have different groups, and there we are taking based on their number. Suppose some group, some area, suppose north western region, it is comprising 60%, northeastern region 20%. So we'll just pick 60% and in the ratio of their number in different groups. Then logarithmic region. So here we are taking the log value from the different partners, different classes and groups. Then heuristic, so that is a quick search, and here we are taking from, we are not ignoring any variables here, characterization variable. Here we are taking, so heuristic search is used in the power core. Then we have several programs for the core set development. So most widely used is the power core and core hunter. MSTAT was also used a long back, but it is not recently used. So power core and core hunter, it is the most widely used right now. And in addition, we can use several classification strategy also to support this. For that, you can use some SAS, SPSS, and R program as well. To use some like classification or principal component analysis. Principal component based strategy also we develop core. So other programs can also be integrated during the core development. Now coming to the types of core. So means that there are 3 types of core. This Odong et al, it is based on the Odong et al. So he has given this classification. Suppose is this first graph, this is a whole collection, and this is the second CCI. Here the concept is maximization of representation of genetic diversity. Here we can see that means from all the groups we are taking equal number of the samples in making the core collection. We are not doing proportional that here it is more, so we'll take more. So we are considering the all diversity we have to capture. Then the second one is the CCX. Here we are capturing the extremes of the whole collection. Here the different we need to develop the collection. We are only interested in the high and low value of the traits, so that's why we are taking this. And in the third options, it is the proportional type. So here in the entire, what is the proportion of all the classes? So like that, we are selecting the proportional sampling from different classes. So this is the third type of thing. Then there are different evaluation criteria for the core collection development. Like here, one is summary status. Summary statistics means mean, minimum, range, CB, and all. So here we have to see that in entire what is the this parameters are there. So that should be almost equal to the parameters in core also when you are doing for different traits comparison. So here, this continuous, it is used for the continuous variables. Then principal component analysis we do. Principal component analysis, we see that how the PC1 is influenced of the different component traits, and we get here by plot also. So we'll see that the core and our entire, so their by plot should be almost similar. Means we can see that, okay, so same component variable is is responsible for the principal component 1 as well as principal component 2 in both the cases. Then categorical traits, these are the 3 criteria: Shannon diversity index, class coverage, as well as the chi-square goodness of fit. It can be done only for the categorical trait, that is a qualitative traits. And we can know here that Shannon diversity, in most of the cases, we have seen that Shannon diversity index increases in the core collection. Then class coverage. Class coverage means we have to see that our all states, for any descriptor, we have all the different states, so all should be present in the our core also. Then chi-square goodness of fit. So that is also very important. In chi-square also, for different states of descriptor, we see the observed and expected value, and then we see that it should be almost equal to the core. So these are the different criteria making the core, whether our code is diverse or not, whether code is optimum or not. So these criteria has been put for their evaluation purpose. In case of power code, so these are 4 parameters. So these are the main evaluation criteria which we receive as output in the power code. That is mean difference percentage, variance difference percentage, coincidence rate of range, and variable rate of coefficient of variation. So mean difference percentage and variance difference, mean difference percentage, it should be less than 20%. Means for 2 traits, suppose it is like plant height. So in plant height, in entire as well as core, it should not be greater than 20%. Only this is the cutoff, that it should be difference between less than 20% only. Then for this other 3 parameters, so the difference should be more than 80%. This value should be more than 80%. Then our core will be the most variable and more diverse. Now, this is just pictorial representation of the our power core program. It is combining both qualitative as well as qualitative traits. So this is some example how it is doing classifying qualitative traits. Then how it is classifying quantitative traits. So this is making the groups automatically based on the range minimum and maximum value of the any trait. So here it is using Esther's rule. It's a formula here. So and the number of classes, it is automatically calculating for the quantitative traits. And like this way, it is giving the, you can see here this normal distribution for harvest length and the spike length. Now, as Power Core is combining both qualitative and quantitative traits, so here this picture is heuristic search. It is looking any variables should not be left out in the core. So it is looking all the variables for qualitative traits also, and for quantitative traits also, how many classes it has developed. So from all the classes, it has to take the variables. So here, when we picture wheat core set, we have used Power Core, as well as in lentil core set also, we have used Power Core program for developing the core set. So wheat core set, it is comprised 2,226 accessions out of 22,416 accessions, and in lentil core set, 170 accessions were developed as a core set from 2,322 accessions. And here, this is the overall the core set developed by NBPGR. So before 2010, some core were developed based on some classification criteria in sesame, green gram, okra, and brinjal. Then up to 2010, we have developed wheat core, wheat mini core, then chickpea core set, and core set of wild lenses. So here, Power Core was used to develop these core sets and mini core set. Then after 2020, so there was like wheat program was the big initiative for the other crops also. So large characterization were taken up in other crops also. And in barley was developed. Here it was based on the Power Core as well as Core Hunter program. Lentil, it was based on the Power Core. In pea, it is based on the Core Hunter. Copy, horse gram, based on Core Hunter. You have compared both the program as well in other crops as well. This is the some like glimpses of the characterization of wheat germplasm. So we have developed the paper in the crop sciences. So we can see here the variability in a spikes and in crop as well. So upper one is the entire set germplasm, and the lower one is here the core set. And here we received the Limca book record for this particular trial. And here we developed a catalog also. So that is being published here. And this is a composite core set. Here all the main cultivated species, breadwheat, durum, as well as dicoccum is present here in this core set. So here the number I have given. Now coming to the lentil core set. So here 2,324 accessions were grown. It has covered all the Indian, mostly wherever lentil is growing, mostly in the northwestern and northeastern part and some central zone of India. And 170 accessions were developed as a core set. You can see the good variability in field as well as in the seeds. And the paper was published in the Frontiers in Crop Science. And this is the example of the barley core set. So here 688 barley accessions from 7,000 accessions were developed. You can see the field view as well as the variability in the spikes. And here also this was published in the Frontiers in Plant Science. So now when we develop core set, so we can see here that core set are not capturing the rare alleles and unique alleles. So that's why you have to better utilize the germplasm in efficient way. You have to use some another approach. That's a fixed approach. It is called as focused identification of germplasm strategy. So here the underlying concept is there that distribution of genetic variation is a function of the relationship between genotype, environment, and conscious or unconscious selection evaluation process. So during the course of evaluation, natural selection play the role in any hotspot conditions. So here, the trait which is more adapted in that particular hotspot condition, so that trait is being selected a long way through the several years. So what do we have to focus here? We have to focus specific adaptive traits, that what traits has been adapted in a particular area, in particular a specific environmental profile and evaluation hotspots. So, and then we have to evaluate those germplasm in the better way so that our chances of increasing better genotypes as a donor and chances of getting genes will be the quick and precise. This concept was jointly developed by ICARDA, the Vavilov Institute of Plant Industry in Russia, and the Grains Research and Development Corporation in Australia. So here we can see that means we should have the different level of information for filtering out the germplasm. So we should have information of collection of the this accessions. So now we have the information of the georeferencing information of all the accession. We have good information in India. We have several software also. So we have good information about soil maps, temperature maps, that rainfall data, everything. So we can georeference our samples and we can get the idea about, based on their collection information, that how this genotype belong to the which particular environment area. So here, so for example, so how can we rationalize the search for useful traits? So first we have to identify the traits, which trait we want to search. Then we have to see in our gene bank, choose accession from environments. For example, suppose we are taking any pest or disease resistance. So we'll search that which area is having more pressure for that disease or insects. So we'll see in our gene bank that which in that particular area we have to focus, and we have to take the accession from that particular area, those accession. We need not to take all 25,000 wheat accessions. We only take only 2,000 wheat accessions. Suppose we want to do for that. Suppose we want to do from north this Uttarakhand region. So there was good pressure of rust disease. So we have found that most of the accessions from there is having rust as well as poddy mildew resistant accessions. So in some study we have done that exercise also. After development of this core collection, we have used this fixed approach for identification of resistant germplasm, and we have validated also. So like that way, so this fix can identify specific traits for breeder more rapidly and precisely, we can say. So here, for example, salinity stress. So first we focus the salinity tolerance that we have to take. So then we define the environmental parameters, that which environment we have to go for selection for the traits. So for that, we'll use the world soil map to see the where the soil is more saline, like that. So like this way, it will focus on the limited number of georeferenced germplasm samples from the evolutionary hotspots. So in this case, we will identify the accessions that were collected in sites that meet the conditions identified. So here in this example, I'm seeing that salt-tolerant rice germplasm collected from Sundarban area as well as the Kutch area of Gujarat. It was explored under NICRA projects. So we went to this particular area and then the germplasm was collected. So we collected also, and in the gene bank, whatever germplasm were available from this particular area, so we took those germplasm also. So from gene bank also, and we explored also, and then jointly we took both the samples and we evaluated for the salinity. So similarly for this drought and heat tolerance, we developed based on temperature and rainfall data, this climatic hotspots region. So this left one is this red area. This is the high temperature area, and here this is the based on the precipitation. So this means light blue area is the drought prone area. So based on that, we have defined the area that from this area we have to make the collection. So we did this gap analysis that from where the gene bank the collections are not available. This Pratapgarh, Dungarpur, and Baswara. Here from here the collections were not available. So we focused from this area, and the collections were made for wheat germplasm from this particular area. Then for rice also, we did this analysis, and in that way we found that from this area it has been collected the germplasm based on the gap analysis, and the collections were made from Jharkhand, Tamil Nadu, Rajasthan, Maharashtra, as well as the Madhya Pradesh, and it was used for the evaluation purpose. So now we have core, then we have fixed collection, but both are independently not efficient that way. So we have to combine both the approaches together so that we can have the better result for the utilization. So the paper was published by Hort and Smith in 2019 that combining focused identification of germplasm as well as core collection strategy will be better for the precise knowing of the gene as well as the donor germplasm. Then another weakness of fixed approach that it only utilizes information on evolutionary factor, natural selection, but actually some other process also play the role here, that is non-adaptive evolutionary process, because in nature we have gene flow and genetic drift also. So we have to consider this point also to make our collection, our finding of accession from any particular collection more better way. So this paper was published by the Stenberg and Oris in 2021. You can go into the detail to know about these features. So very nicely means they have described that we have to modify the fixed approach also a little bit so that we can get the better. So now core collection than fixed. Pre-breeding is another very strong approach, which is already discussed by several speakers. Today also, sir has talked, Dr. Patra sir, then Dr. Anas Bains. Before that also Dr. Sir Yadav has taken. Then we have on Pulse. So I'll not talk into much detail about the pre-breeding aspect here. So just this is this classical example on the tomato, and this is the Oryza rupipogon, and here we can see that domestic bottleneck after adapting from genes from the wild, from when we integrate from wild into modern variety. So there is the problem with domestic bottleneck, and there is a less variability in the modern varieties. We have to consider these things. Then at NBPGR, we have several programs, but the pre-breeding was initiated with the work on wild sensor and lens collection. So there is some view of that one. It was followed by several others like lens, P, Bigna, then recently in Brassica and Sesame also it has been used. So here, this is the view of the screening of wild. The paper was published in Journal of Genetics and Plant Breeding. And then this were further screened for several biotic stress and different sets were developed. And based on this integration program, some varieties has also been developed recently. Then coming to the utilization of plant genetic resource in the variety development. So through this graph, I want to show that 57% germplasm have been used from the indigenous background. From exotic germplasm, only 8% have been used, and 20% use is from the hybridization between the indigenous and indigenous, and only 6% is used by the indigenous and exotic crosses. Here some example of germplasm utilization for the variety development in some crops. I will not go into the detail here. And here just I have taken some like rice germplasm that the sources, and some gene has also been identified here, like Pokali salt gene has been identified from Nona Bukra SKC1. And then in wheat germplasm also, the Karachia landus that is widely used, and several varieties has been released as a salt plant variety. Similarly, this Cook Australian cultivar, it has been used in the development of HT2402 and HS240. And the Bhiri, it is very popular cultivar. It is having one art location. So it has been found in the several cultivars at present. Now, so pre-breeding, I will not go into the detail, just I touched upon. So now coming to the genomic assisted utilization. So here, just I will brush up that this genomic tools, it is being used in the for several purposes already dealt with: DNA fingerprinting, molecular diversity analysis. So this is used for the genotype identification. After this purpose, we come to know about the unique genotypes and diverse genotypes, so that we can make the crosses and we can integrate the diverse parents. You can broaden the genetic base of the population. Then we have to identify the gene. For identification of gene, there are several approaches. We need mapping population here. So one is the biparental mapping population. Then we have magic population also. I will not go into detail. Dr. MC Yadav in next lecture, he will be talking about. Then at gene bank, we are mostly using genome-wide association panel because it is based on the diverse collection of the natural population, which is unrelated, using some SNP marker data or GBS data. So we are using this particular approach for identification of genes at NBPGR. Then for genomics, if you have to identify some rare alleles, in that case, we have to see for the fixed set. We should not go for the core and mini core for rare alleles. So we should target fixed set, and that can be used for the GBAS. For example, for Russian wheat aphid resistance, so this fixed set has been used for the GBAS study. Then haplotype mapping is the very useful tool for mapping haplotype, and haplotype can be introduced in the allied cultivars for the cultivar improvement. And then allele mining, it is very important for the trait discovery as well as the gene discovery. Here use various approaches. It will be dealt in the next lecture. Then utilization, when we got the gene, when you get the QTLs after GBAS or by parental mapping, then you use those gene or QTLs for by marker assisted backcross breeding, genomic selection based. Then here come the role of genomic selection based prediction also. I will not, this is the new approach, and genome editing also. Genomic selection, it is widely used in the US and Canada, Europe. In India also, we have started even at IRI, in chickpea program and in wheat program, genomic selection based prediction have been recently initiated. Now, just a study, just I will sum up our this wheat program. I already showed that how at NVPGR we took up 22,000 wheat germplasm for the phenotyping purpose. So simultaneously, it was done for the abiotic stress, terminal heat tolerance. Then it was for characterization. That was for core set development. Then it was for biotic stress tolerance. So it led to the multiple disease resistance line. That is a reference set that was used for genome-wide association mapping later on. From terminal heat stress also, we sequentially evaluated for 5 years. So we narrowed from 22,000 to 3,000, then 1,000, then 300, and then it was used for the identification of reference set as well as development of the genomic wide association mapping population. So similarly for this multiple disease resistant line, 56 lines were developed, and these lines have been distributed to the indenters or researchers for use in the breeding program. This is some glimpses of and flow chart of this terminal heat stress evaluation. This is glimpses of the dot tolerance evaluation. So for dot tolerance, we have not done for the all 22,000. For here, we took up the core collection because for comprehensive evaluation, we need to have a smaller number. So this core collection were taken multi-location evaluation at Delhi as well as Hisar. Here also we developed diverse panel of GBAS, and the difference set also we identified, and it has also used for the GBAS study. Then this example for wheat mini core study. As in phenomics, even core set is not optimum because core set is the larger number. So we need to further reduce the number so that our evaluation will be in better way if you used to do in the phenomics study. So this mini core was screened in the phenomics condition. And now just I am seeing here that how core, mini core and difference set flow here, and we studied for several traits like heat and dot tolerance. We studied for root architecture also, salinity tolerance, then seed morphometric analysis and disease resistance. I'm giving some glimpses of our GBAS study. So for heat tolerance, 205 diverse wheat panel were studied. So here we studied agronomic traits under late zone condition, and a total of 69 QTLs for different traits, potential killers, then grain yield, grain filling rate, and harvest index, grain number for these things has been identified. Here we could identify 3 hotspot regions on chromosome 1B, chromosome 2B, and chromosome 2DS. So these QTLs are being validated in the by parental mapping population for use in the marker assisted selection. And now coming to this root architecture study. So here we used another approach, that is multi-locus GBAS model. In earlier we have used MLM model. Here different approach was used, which is more precise. And here 136 diverse wheat panel were taken up and 50 seedling root architecture were taken up for this study. So here 7 QTNs were consistently detected by different all 6 models. So here some of the glimpses of Manhattan plot. Here the favorable alleles graph is shown. Here the paper was published in ISMS. And this is the study about the salinity tolerance. So here 153 diverse wheat panel were taken up for this study. So here also we could identify 12 major QTNs in this study, and it was published in the genomics. In addition, another program, here several multiple disease resistant lines we have obtained. So here some of the glimpses of these lines here, like 29 accessions were resistant and tolerant to rust mildew, and out of this three are important lines for which has multiple resistant to all the diseases. Many of them are being used in the mapping population development and GWAS study. We have also developed several genetic stocks, for example, for spot blotch-based resistance, for stem rust, leaf rust, and stripe rust. Then stem rust again. Then we developed for drought. Then we have developed very good zinc, high zinc line containing 53 ppm and 51.3 ppm based on the multi-location trial which was undertaken at 10 different locations under AICRP program. Then these are the glimpses of some spot blotch resistant lines. So we are using these lines into the crossing program for developing a mapping population as well as integration purpose. So these are the some example in extension used in the crossing program for different traits. Now afterwards, now we are focusing land races after development of core set and evaluation of that.
Please try to conclude in.
Okay, it is a second last slide only. So these are some glimpses of different Durum braid wheat and dicoccum land races. This is Asperococcum wheat germplasm. It is the Indian dwarf wheat, which was long back very popular. So we are evaluated for all the traits here. These are glimpses of some field days, and we had supplied to partners core minico referenced Tetus germplasm around 11,000 samples to around 80 indenters. So thank you all the participants. So if you have some questions, you can ask.
Please be very quick in questions also.
I'm
one in chat box. What is the ideal plot size for characterization wheat germplasm?
Yeah, for characterization, we not to have a large number, but 1 meter square is the optimum. We should have 1 meter square.
Rows like.
Like if it is 4 rows, then you can have 1 meter row length and 4 rows. So it will be a kind of a square plot.
Okay. Any other questions from participants? If no, then okay. So thank you very much, madam, for giving insights about the major issues of germplasm limited use and how the strategies for overcome these issues. So you have explained nicely all aspects of core, mini core, and fix everything. With this, thank you very much for your thoughts and giving insights about these issues. Thank you. Now I'd like to request Dr. Subhas
yes,
for next.
Okay. Thank you, Dr. Kuldeep. Now we have with Dr. Mahesh Yadav from Division of Genomic Resources. Welcome, sir.
Good afternoon.
Good afternoon, sir.
Good afternoon, sir.
Good afternoon, sir.
Yeah.
Welcome, sir.
Thank you.
Friends, we have a next lecture on allele mining and trait discovery in crop genetic resources. First of all, I will introduce our speaker. I'm privileged to introduce our speaker, Dr. Mahesh Chandra Yadav. Dr. Yadav is a principal scientist in genetic. He's from genetics, and he's the head of Division of Genomic Resources in ICR and UPGR New Delhi. He completed his graduation from Chaudhary Chandrasekhar Azad University of Agriculture and Technology, Kanpur in 1989, and MSc and PhD from IRI New Delhi in 1992 and 1998 respectively. He started his professional career as a ARS scientist at Directorate of Mushroom Research, Solan in 1996, and in 2005 he became a senior scientist at same institute. Later in 2019 he became a principal scientist as ICR and UPGR New Delhi. Sir has a specialization in molecular genetics and breeding, DNA barcoding, molecular analysis in rice, wheat, and maize for biotic abatic stress tolerance, especially drought and heat. Among the major achievements, his group of NICRA project has developed a mini core and trait-specific core for terminal heat stress tolerance in wheat, and this core has been molecularly validated and also collected 542 accession of rice and wheat under this project. He also developed a biparental and a magic population in bred wheat, and several QTLs were identified for salinity, stress tolerance, and root rates. He had developed 4 varieties in lentil and mung bean and also registered 30 genetic stocks. He has published more than 90 research articles in peer-reviewed journals and written 15 book chapters and edited 2 books. Dr. Yadav has presented several oral and lead papers at various national and international fora. He has guided 6 PhD and 3 MSc students of PGR and biotechnology. He also had overseas experience as a foreign training at Horticultural Research International, Wellesbourne, UK. In biotechnology, ITS rDNA sequencing and genetic transformation in agaricus mushroom. He has conferred with several awards and honors. Among these, few I am mentioning here: silver medal in during his graduation, and Yadvindra Young Scientist Award he received from Mushroom Society of India, and he is conferred with the Fellow of Bio Research Society Allahabad, and Krishi Jivan Jyoti Award for Research in Genetics and Training of Youth in Agricultural Biotechnology by ISA. He has received a Distinguished Scientist Award in 2016 by SD Merit, and fellow. He is received a fellow of Indian Society of Plant Genetic Resources in 2019. Now I request sir to share his slides for his presentation. Now up to you, sir.
Okay, thank you, Dr. Suvas, for such a elaborate introduction that does not require. Okay, so I am sharing my slide.
सर स्लाइड दिख नहीं रही है.
Hello.
I'm
I'm not able to share the slides. Just trying to share the slides.
Okay.
क्या डॉक्टर साहब कुछ प्रॉब्लम आ रही है क्या?
वो उसको शेयर नहीं हो पा रही है।
वो बताएंगे सुभाष या
हाँ
कुलदीप।
सर, अगर देख सकते हो तो देख लीजिए आप कैसे अगल में। सर का ऊपर ही होगा आपके रूम।
मैं जाके हूँ सर, मैं
हाँ,
आता हूँ।
ऊपर ही।
सर स्लाइड तो खुली हुई है ना?
हाँ स्लाइड तो खुली है।
पीपीटी को
उसके
मिनिमाइज
ये
कर
सर
लो ना।
तो
नीचे
वहां
थे।
पे कर्सर ले जाकर डबल क्लिक करना है सर, जहाँ पे स्लाइड पे
सुबह सर देख लीजिए, सुबह सर देख लीजिए। मैडम अब बताइए आप।
डॉक्टर साहब पूरा पीपीटी खोल के मिनिमाइज कर लो और फिर शेयर करोगे ना, वो दिखेंगे। उस फाइल को सिलेक्ट कर लो, वो दिख जाएगी। सिंपल सा है।
हाँ, उसको फिर डबल क्लिक करेंगे जहाँ पे स्लाइड दिखेगी ना वहाँ।
चलिए, एनीवे सुबह इज गोइंग तो हम देखते हैं।
ठीक है
सर तब तक इंट्रोडक्शन दे दिया कुछ थोड़ा टॉपिक का, जब तक वो जा रहे हैं। इज गोइंग।
हाँ ये एक्चुअली इसमें शेयर करने की कोशिश कर रहा हूँ तो दिख ही नहीं रहा है। ठीक है, ये आंसर सही है। शेयर पॉइंट। उसमें कर लीजिएगा सर। So good afternoon. Can you see my slides?
Yes sir, please put on slide show.
स्लाइड शो कर लीजिए इसमें नीचे।
फर्स्ट वाली से।
ये वाला
स्लाइड शो किया हुआ है। Now
नहीं
it's
आया।
visible.
ना,
नहीं,
स्लाइड शो
नहीं।
नहीं किया हुआ।
वो चलेगा क्या?
Now it's visible.
नहीं सर, your all slides are coming.
Okay.
वो बीच वाला आ रहा स्लाइड शो।
हाँ, अब ठीक है, अब ठीक है।
हाँ, अब इसमें स्लाइड शो कर दीजिए।
Now it's okay.
नहीं, अभी स्लाइड शो नहीं हुआ। सिंगल तो आ गया नीचे। ये दबा दो नीचे कब सा।
नहीं वही दबा रहा हूँ मैं। Now it's coming. No.
ना, या
Now
फिर
it's
आप
coming.
पहले स्लाइड मोड में ही ले लो, उसको मिनिमाइज कर लो, फिर वो सीधा ही ले लेगा। कुलदीप
Now it's visible.
नहीं, वो तो स्लाइड छोटा बड़ा वैसे हो रहा, लेकिन स्लाइड शो नहीं है ना।
सर पुट इट इन स्लाइड मोड सर।
मैं यहाँ से ही कर रहा हूँ लेकिन
हो नहीं रहा वो। मेरे ख्याल से सुभाष शायद नीचे से ही करना पड़ेगा। पहले ही खोलो उसको स्लाइड मोड में ही खोलो मिनिमाइज करके।
अब आ रहा है नहीं आ रहा है?
सर वैसे अब अगर नहीं आ रहा है तो इट्स विजिबल, आप कर सकते हैं। यू कैन स्टार्ट बिकॉज वी आर रनिंग शॉर्ट ऑफ टाइम।
अभी आ रहा है?
सर एक बार F5 प्रेस कर दीजिए, फर्स्ट स्लाइड पे ले
बड़ा
जाओ।
बड़ा हो गया डॉक्टर साहब। ठीक है, चलो
ठीक है सर, इट्स ओके। फर्स्ट स्लाइड ले लीजिए सर, फर्स्ट स्लाइड।
सर फर्स्ट पे क्लिक करिए एक बार, 1 पे।
नहीं उसमें स्लाइड शो पे करता हूँ लेकिन आ नहीं रहा हूँ।
कोई नहीं, फर्स्ट स्लाइड पे कर लीजिए डॉक्टर साहब। फर्स्ट स्लाइड इजी में हो जाएगा।
आ रहा है अभी ना?
सर सेकंड विजिबल है, प्लीज एक बार फर्स्ट पे क्लिक करिए।
और ठीक है।
नहीं, फर्स्ट स्लाइड फर्स्ट। ये सेकंड स्लाइड आपकी।
आई मे कम सर।
हाँ,
सर F5 प्रेस कर दीजिए कीबोर्ड में, F5।
ओके ओके। नाउ इट्स ओके। हेलो।
नहीं,
F5 किया हुआ है।
मैं आती हूँ सर।
I don't know what's happening.
देख लीजिए एक बार।
तो ओके।
अदरवाइज यू कैन स्टार्ट सर।
बात होगी एड्रेस लिख दिया everybody and this my topic is lean mining of discovery and crop genetic resources. So how this lean mining can be done for jump-large resources of different crops. नहीं आ रहा है, इसी तरह कर रहा हूँ। आ रहा है, दिख रहा है? Now is it is visible? हेलो।
यस सर। यस सर।
आ रहा है ना?
विजिबल।
ठीक है।
बस स्लाइड शो में नहीं है।
वो कोई बात नहीं होता तो इसमें कर दीजिए, हो जाएगा। इसमें प्रेजेंट हो जाएगा।
चलेगा सर?
चलेगा, बड़ा हो रहा ना ये, आपने बड़ा स्लाइड कर दिया।
एक मिनट देखने दीजिए।
सर ये स्लाइड मोड में आ रहा है क्या सर?
ना स्लाइड मोड में नहीं आ रहा, मुझे लग रहा है कि वो वहीं से
अभी आ रहा है सर?
मिनिमाइज कर
नहीं आ रहा है, बट इट इज विजिबल। आई थिंक
कर
कंटिन्यू
सकते हैं हम, चल सकता है।
सर विजिबल सर, कंटिन्यू सर।
अदरवाइज टाइम फिर कम है। फर्स्ट स्लाइड से
ये फोटोग्राफ है ना, थोड़ी हेवी हो जाती है ना, तो आगे से क्या कर सकते हैं ना?
वॉइस आ रही है, आवाज आ रही है?
अरे इट्स ओके, कैन यू स्टार्ट?
Okay, so this allele mining is very important biological tool to identify different alleles of a gene, and that will aid in the trait discovery and germplasm, and that will be useful for the crop improvement programs. So as introduced by our Dr. Subhas, I am Dr. Mahesh Yadav, the Division of Genomic Resources. So let us see what is the mining before really understanding the allele mining. So mining is the extraction of valuable minerals or other geological materials from the earth. So you can see the coal mining, how it's been done. So this mining in a wider sense comprises extraction of any non-renewable resources like petroleum, natural gas, or even water from the earth crust. So in the context of allele mining, this in biology, the allele mining is an approach that is used to dissect naturally occurring variations for suitable alleles of a candidate genes controlling the key agronomic traits which are having the potential in crop improvement programs. So this allele mining is helpful in the identification of haplotypes and development of allele specific markers for the different programs of crop improvement, especially the marker sister selection. So before really understanding the allele mining and crop plants, we need to know what is the allele. So this allele is a Greek term coming from the allelos, meaning each other. So an allele is the one of the series of different forms of a genetic locus. The word is a short form of allelomorph or other form which was used in the early days of genetics to describe variant forms of a gene detected as different phenotypes. For example, a very good example is the gene locus for ABO blood system in case of human being. So here the 6 possible genotypes are there, and there are 4 possible phenotypes of A, B, AB, and O blood groups. So now in the era of genomics, the alleles are understood as alternate DNA sequencing at the same physical position, which may or may not result in the different phenotypic traits. So these very good example of how different alleles have been identified in case of Drosophila, fruit fly, a very important organism for genetic studies. Similarly, in case of flowering plants like Cantirana majors, the different petal color is coded by different alleles. So these are like in Drosophila from white to the intense red eye color has been identified, and different allelomorph for alleles have been identified. So the next question is how to identify these alleles and how to mine these alleles in the germplasm. So this procedure is to identify the accessions or target germplasm and then selection of genes or target genes associated with these phenotypes. Then development of primers for the target gene and the amplification using these primers, the gene of interest. Then finding of different allelic variations of the target genes. So there are 2 different approaches are there. One is eco-tilling based allele mining, another is sequence based allele mining. So particular eco-tilling based allele mining uses the germplasm, whole collection of germplasm or the smaller set of germplasm lines, and the sequence based they are either the certain regions or genomic region sequence based mining or the whole genome sequence based allele mining. So there are different markers for identification of new alleles. So here I have given 2 most important markers. One early was the microcytid markers where number of alleles of a gene have been identified. So say for this example of 3 accessions, diffraction having 3 different microsatellite alleles. So one allele is like one is showing the 12 repeats of GA, and other is having 16 repeats, and still third allele is having 8 repeats of this GA repeats. Di allele means di repeats microsatellite motif. So similarly, this particularly SNP marker or single nucleotide polymorphism, they are sequence-based markers. So these have been really pinpointing where the particular base is changing. So there are 2 types of this particular substitution, base substitution, like transition type SNP and transversion type SNP, based on the how the appearance in the primate they are interchanged at a particular locus. So this is there are 6 alleles are there. So this particular GA mutation, transition mutation is there. Another CT transition mutation is there. So third one, this is AC, means the transversion mutation is there. So how to identify new alleles in crop germplasm? So this is again example of 3 SNP. These 2 SNP, G2A is transition form of SNP. Another is also TC is a transition, and third this is CA transversion type of SNP, or we can say a base substitution. So particular say for this is our breeding lines, this but having this kind of haplotypes. So there are 7 possible haplotypes using these 2 or 3 SNP can be found. So the different allele forms can be identified in the jumper lines having different either phenotypic expression, or they may be somewhat maybe having the similar phenotype. So identify new alleles from germplasm. Say this is the under heat stress, yield under heat under stress, especially the biotic stress, because biotic stresses they are coded by major genes. So this is kind of like a gene, particular unidentified gene that is giving this much expression, and there are different alleles from allele 1 to 7, and they are having different expression. Also, allele 3 is the best, which can be identified through association mapping. So identification of new allele, we need to have a different strategy. So for having the strategy, first what we have to do, we have to take out the entire collection of germplasm of a particular species or particular crop plant, and morphology characterize over the years for multiple locations, and molecular characterization with different robust molecular markers, then to estimate how much diversity is there. Then based on these morphological characterization, molecular characterization, we can develop a usable set of germplasm that called core collection, that is around 10% of entire collection, for efficient mining of collection of genotypes with the least redundancy in diversity estimates. Then if still it's a larger set, then we can go for mini core, which is around 1% of entire collection or 10% of the core collection. So we can develop the allele mining set for the best identification of or efficient identification of different novel alleles. So this the development of different sequence databases, the particular reverse genetic approach has become a important tools. So here high throughput tools have been developed, and they are being used for the development of this particularly NGS sequencing technology and genotyping, and high throughput phenotyping. These are 2 important for identification of new tools, new alleles. So there are 2, as I said, that there are 2 allele mining approaches. One is tilling, the target induced local lesions in genomes by using different mutagens, so particularly the chemical mutagenesis that can be induced easily, and with the least deleterious effects. And another approach of this tilling is like modified tilling, that is eco tilling. So that is very popular nowadays, having natural mutations in the germplasm lines that occur over the years for hundreds of years. and second is the sequence-based allele mining. So there are two important nowadays used these approaches: eco-tilling and sequence-based allele mining. So the tilling procedure of allele mining is how we need to follow different steps. First is the development of tilling populations by using different chemical mutagenesis. This is the case of like rice, where EMS mutagenesis have been used to develop the tilling populations. So after this M1, M0, this M1 plants are there. Then M2 plants, they have been phenotype M3 and M4 seeds have been developed. So this during M2 population, we can collect the samples for the DNA extraction, and they can be sample. Some of the samples can be pooled for further detection of the mutations. So there are these approaches, especially the heating or cooling of that particular, then it will form the homoduplexes or heteroduplexes. So homoduplex DNA shows the normal binding of wild type DNA sequences. So heteroduplex DNA shows unpaired regions which are site of possible mutations. So how the mutations using this approach can be identified, and this heteroduplexes, they will be electrophoresis and fragment separation using different the electrophoresis technique like PAGE, Lycor gel, capillary electrophoresis, DHPLC, and to identify these new alleles by using tilling approaches. So another is sequence-based allele mining procedures. So here we can have the entire collection or global collection for any crop for a specific trait. So we need to isolate DNA, then we need to design the primer and the PCR amplification, and this sequencing and identification of variation. So here the two approaches are there: direct gene cloning and sequencing, or direct PCR amplification of particular gene of interest and sequencing. Then the sequence has to be aligned to mark the variant, allelic variant, and that can be analyzed to see the how many alleles are there and how many haplotypes are there using this sequence-based allele mining procedure. So the trait discovery, how it can be aided by using that allele mining approaches. So for that, the selection of germplasm carrying allelic richness is very important. So the should be like global collection or mini core or core of the global collection for a particular trait. Then development of core mini core sets from the entire germplasm accessions. Then precise phenotype using high throughput phenotyping tools and techniques. The use of artificial intelligence and machine learning technology for detecting phenotypic variations in the germplasm of interest. Then molecular characterization, identification of novel alleles. So how this let's see what are the purpose and application of allele mining. So allele mining has a lot of potential for crop improvement, especially after the generation of a lot of sequence databases which are available in public domain nowadays. After not only the major crops, but the entire set of the germplasm, and sometimes mini core has been that whole genome sequence or resequenced. So a lot of information, not only the crop species, this cultivator species, but also their crop relatives is available in the public domain. So it is important to use this genomic information for identification and isolation of novel and superior alleles of agronomically important genes from the crop gene pool, and deploy these novel alleles for the development of improved cultivars. So this allele mining is a very important the promising approach to detect natural variation occurring at a candidate gene locus, controlling key important agronomic traits, and it helps in tracing the evolution of the alleles and how the alleles have been from there were pre-domestication and post-domestication, how these alleles have been changed, and identification of new promising haplotypes. Then development of allele specific marker for use in markers selection, So realizing the potential of these allele mining techniques, a lot of efforts have been now underway in international as well as national institutes for allele mining in different crops. So the application of allele mining are diverse, especially allele mining has tremendous application in agriculture, more specifically in crop improvement programs. Then that includes the identification of haplotypes and molecular markers for genetic diversity, characterization, and syntenic relationship, and use of these in marker selection. So we can say that allele mining can be used for the identification of novel alleles, promoter mining, allele specific variation characterization, allele synteny development, haplotype identification, analyzing haplotype diversity, evaluation study of the evolutionary relationship studies, similarity analysis, and allele marker development for the different purposes it can be applied. So there are the number of cases where these allele mining has been used successfully for crop improvement. So this is the example of identification, utilization of new alleles in tomato. Here the QTL for flash color has been identified in crop relative of tomato. That is species Solanum hirstum that not turning red on ripening. But when it was crossed with another process in tomato cultivar E6023, and lot of nils near isolate lines have been developed that carrying the allele from the Solanum hirstum for red pigmentation, red pigment of this fruit. Another example surprising was the QTL identification fruit size from the crop variety of tomato. So here the small fruit of Pimpernifolium, Solanum pimpernifolium has been, it was crossed with the processing cultivar E6023, and the F1 then the smear isolate lines have been identified that carries allele from the Pimpernifolium with 10% increase in fruit size. Another successful example has been the allele mining for blast resistant genes in case of rice. So allele specific PCR based SNP and DIL markers were used to identify the novel alleles for rice blast resistance genes. This study was published way back in 2006 in TAC, and they identified 9 alleles of this particular gene, blast resistant genes. So they have been physically mapped and validated using SNPs. They are different structure of these alleles. So similarly for the powdery mildew resistance and wheat, this paper, very good paper, was published in PNS in 2009. And here the core collection of 1320 wheat landraces screened for powdery mildew reaction, and 211 landraces was complete or partial resistance. Then 111 landraces had PM3 gene specific markers. So sequence analysis and functional validation by gene silencing were done to identify new alleles. So here this is the resistant landrace with new allele for powdery mildew resistance, and this resistance landrace with the new allele silence shows powdery mildew symptoms. So it was silenced by using the this transgenic technology. So by using this paper, they have identified 7 new functionally active alleles of PM3 gene. Another very good example of allele mining is mapping of red pericarp in rice gene. So this RC gene, a VHLP protein, that's a change from red grain to white grain in rice has been cloned and characterized by Sweeney et al. 2006, and that was published in Plant Cell. So these are the how the phylogenetic relationship of different indica and japonica cultivars. Then red, this indica, red indica 1 and red indica, they have been red pericarp and red os also. So this particularly gene has been identified, and they gave clue for single origin domestical event of indica and japonica rice cultivars. So allele mining using next generation sequence platforms has been now very much in vogue, and earlier as the Rocky 454 pyro sequencer, then it came Illumina Solexa, Genome Analyzer, ABI Solid, and latestly the nanopore, Oxford nanopore technology for allele mining purposes. So these are the different crops and they are the different references. They are the like in Arabidopsis 8,23,325 alleles were mined using Solexa NGS platform. And similarly in chickpeas more than 500 the alleles were mined using again Solexa. Then eucalyptus, wheat more than 100, around 100 by using this 4-5 their parasequencer. Whole genome sequencing contrast RAS genotype. Here 160,000 SNP that alleles were mined by resequencing microarrays. So that was published by McNally at 2009. So here by using this NGS platform, now it's very easy to identify novel alleles in different crop germplasm. So again, this example in maize, natural allelic variation in barren and fluorescent 2 that VAF2 gene that regulates the maize architecture and fluorescent architecture. And here this study was published by Professor et al. In Plant Journal. They mined the novel alleles for VAF2 gene, a maize ortholog of the Arabidopsis pinicoid protein kinase PID using 277 diverse maize and lines. They used the LD mapping and identified two major SNP haplotypes in B73 and MO17 at the promoter region of VAF2 gene, and then associated with the wide range of plant morphological phenotypes. So allele specific expression analysis were done and they identified these two alleles, which association of MO17 allele with a reduced distal branch zone and showed lower expression than the V3 allele by modulating oxygen transport during vegetative and opposite development stage. So this is a latest example published in BMC Genomics 2021. So here they study by Liu et al. 2021. They identify different alleles and QTLs for 1000 grain weight, grain length, and grain width length ratio in case of rice. So by using this sequencing, they identify 7 haplotypes in the rice for 1000 grain weight, and they were located on chromosome 3. And so they are the like a whole set, and the germplasm and As germplasm, basmati, and then inter group was there. So different groups, they analyze how these different haplotypes they are having the diversity. So they identified 7 haplotypes, or you can call alleles, in for 1000 grain weight in case of rice. So allele mining has a lot of potential in different crops, and a lot of examples are being the reference are there. They have been utilized for this is the some of the old slide there. They have been used for different purpose. Say for Agilops, for allele locus, that was used by Gills and Brown 2006, agropyron, wheat, and then barley and different crops. In case of rice, this quality gene, fragrance gene, VAT2 has been used, and our lab also developed different alleles. We found 2 alleles particularly for this VAT2 gene that having 8 base pair deletion or not having 8 base pair deletion at 3 SNP in the VAT2 gene. Similarly, in rice, other genes like PETA, OSC1, they have been used for allele mining. So different crops they have been used, and particularly nowadays it is a lot of using sequencing. The lot of alleles have been mined for different agronomically important traits. So there are different bioinformatic tools of behavior sources for the which can be used for allele mining. Like this place plants is acting regulatory DNA elements. So this particular software database of transcription affected binding motifs developed by Higo et al., very old one. And then plant care, that plants is acting regulatory elements database developed by Lasquat 2002. Then transfact database of transcription factor and transcription factor binding motifs developed by Mighty et al. And Jasper W. Alangis meme this for motif discovery. Then the plant from DB, plant promoter database by workers in Samurandam and Matt Timser. Nowadays, number of others tools have been involved for identification of alleles, novel alleles, especially which have been well characterized. So there are a lot of challenges out there before this allele mining. The important challenge is the selection of jump plan, target jump plan. How to select the target jump plan to identify the novel alleles is very important. Then precision phenotyping of the selected jump plan for the trait discovery. So both like we can use allele mining for trait discovery, or first discover the trait, then mine the different alleles for a particular trait. So that is a big challenge of selection of diverse jump lines and their precision phenotyping. Then handling of large genomic resources for appropriate use by multidisciplinary expertise. So here, single man cannot do this kind of job. We need to involve multidisciplinary based expertise for the allele mining and their subsequent use in crop improvement. Then identification of promoter and its demarcation from the non-coding and coding regions for marking of the true alleles. So sometimes the true allele means here that their effect can be realized, or that their effect can be phenotypically verified. So identification of this promoter region and their demarcation from the coding and non-coding region is very important. Then characterization of regulatory element, a lot of regulatory elements, especially the microRNA and non-coding RNAs, they are playing a role in the expression of gene. So important is to characterize these regulatory elements or regions, and determination of sequences, changes in loci interest using precise or highly efficient genotyping methods for large scale identification of alleles. So they are some of the challenges for allele mining, and so this is all about the different allele mining approaches and how these approaches can be used for the identification of different novel alleles, and that further how to use these novel alleles for the crop improvement for a particular trait.
Thank you, sir. Friends, now we can you can ask any query or question from sir. Sir, there is one question from Dr. Vinod Kumar Jain KB.
Okay.
For allele mining, can we take a seed, or which plant part is much suitable?
Okay, can you repeat that for allele mining?
Sir, Dr. Vinod.
Dr. Vinod is asking, for allele mining, can we take seed, or which plant part is most suitable? I think for DNA extraction he is asking.
Yeah. Actually, if you want, first you have to target the trait. Which trait you want to target, and for that trait you need to mine the alleles. So say for some trait are expressing, in case of say for in wheat, they are expressing in the spike. Say for 1000 grain bed, other things are there. So you can go for that particular like flag leaf or the grain itself, or the part for the DNA isolation and their allele mining. And say for some of the say for target rate, they are expressing in say for root. So in that case, you need to isolate the RNA from that particular portion of or tissue.
Any other question? You can also put on in this chat box.
Yeah.
Any question, please? Sir, I think there are no question.
Yeah,
You have
actually, it's
explained very
lunchtime, I
beautifully.
think. So they are, I think, from morning, they are busy in listening the lot of questions. So actually, this particular, the allele mining is very important, especially for if the particular allele is contributing more to the phenotype, then it's very good, especially for the major genes.
If there are no question, then I may propose a vote of thanks to our speaker. Sir, I, on my behalf and on behalf of organizers, I thank you very much for delivering such an comprehensive lecture on allele mining and trait discovery in crop genetic resources. Sir, you have given insight in allele mining and its application in PGR. And you have explained briefly about different procedure and strategies of allele mining using molecular markers and next generation sequencing. Certainly, sir, our participant has benefited from your presentation. Thank you very much, sir.
So thank you very much for giving me this opportunity to interact with the audience. I
Sir,
think
another one question, sir, received, sir. Is of agronomic trait or can be used for biotic and abiotic traits.
Can you repeat that?
Allele mining is for agronomic traits or can be used for biotic and abiotic traits.
Yeah, it can be used for both, like trait that are quantitative traits, like some of the quality traits, or the quantitative traits, like some of the biotic and abiotic stress, especially the abiotic stress tolerance trait. So they are can be used for both purpose. But say for like quantitative traits, they are governed by small genes with the cumulative effect, and they are located on different chromosomes of the genomes, called QTLs. So the easy way to identify different alleles is for the qualitative traits governed by major genes. But it's also because in case of quantitates like abiotic stress tolerance, lot of genes and their alleles are interplaying to give the final resistance phenotype in that case. So it's very easy to identify novel alleles in case of the qualitative traits, especially governed by major genes. But it can also done by for the abiotic stress tolerance, like salinity tolerance are there, like heat stress, drought stress, all for all you can identify different alleles with a means say for 10 to 12% phenotype variation.
Okay, thank you very much, sir.
Okay, thank you.
Okay, then. Friends, then
okay,
thank you, sir. Thank you very much.
Thank you. Good afternoon.
Good afternoon, sir. Friends, we will meet at 2:20 and for another important lecture, that is on application of nanotechnology in PGR management.
Okay. Now, hello.
Yes, sir.
Okay, I'm trying. It is shared, sir.
Yes. Now it's in a presentation mode.
Okay, good. Okay. Now I want to change my video audio. How to change my audio?
Your audio is very much clear, sir.
No, sir. I am just using my headphone, but the headphone is
Okay.
the headphone. It is the voice is coming from the computer. Can you talk? Hello?
Yes, sir.
Oh, okay. So my
I think
1 second, sir. Sounds. 1 second.
you have to do settings.
Yeah, I'm
Mic.
Okay, now, hello?
Now you go to, sir, select a microphone.
Yeah,
Which as same as a system.
Yeah, I selected. Now also it is not, I'm getting audio only from my computer.
Your computer.
I'm just getting only audio from my computer, not on the headphone.
Okay. You want it from.
I think that's not a big deal.
Sir, working.
No, he is asking for headphone. I think sir, you are using your Bluetooth speaker.
Pardon?
You are using Bluetooth headphone.
Yeah.
Sir, please ensure that your Bluetooth is connected with your laptop.
Yeah, it is connected. Now I have another option.
Okay.
Okay, you have to add a one number of wear gloves first. Add a one number and add a 2 drops of and then you mix it. Hello? Hello?
Yes, sir.
Yeah.
Now it's working.
Yeah, now my headphone is working.
Okay.
Okay, clean. Perfect. So the audio is okay and the video is okay.
Yeah. Video is okay, sir.
Okay. Fine. Good.
Audio and video are both are clear.
Okay, perfect, sir. Thanks. Madam.
How are you, sir?
Yeah, I'm good, sir. I'm good. I'm totally good.
Okay.
Thanks for the opportunity.
Sir, yes, sir. Please unshare your slide first, no? Then.
Okay, let it be here so that audience will join. They see the title only, and then because I don't know whether it is easy for me to.
Sir, before that, I have to share one slide, sir. So that's why I'm asking you to unshare your slide.
Okay. Now is it okay?
We'll wait for 10 minutes, sir. Then we'll start.
Okay.
Because we have given time at 2:20.
Okay, sir. Okay, understood. Okay.
Because 2:30 is time is given for presentation.
Sorry. He made a hole. Where, oh, you got one. I was making a hole.
क्या डॉक्टर सुभाष कुछ प्रॉब्लम आ रही है?
हेलो
हेलो, डॉक्टर सुभाष।
सर, प्रॉब्लम नहीं आ रही है. वी आर वेटिंग फॉर पार्टिसिपेंट टू ज्वाइन सर.
ओके ओके ओके, ठीक है।
The time is 2:30.
या टाइम इज 2:30 सर.
इसीलिए
अच्छा अच्छा अच्छा, इसका हम लोग को डिले नहीं है, अपना टाइम से
no
चल।
no no
Yes, 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 sir.
testing was going on, sir.
ओके।
Dr. Jangi Raman sir, can you hear me sir? Dr. Jangi Raman, can you hear me?
Sir, good afternoon, sir.
मेरे ख्याल
Dr.
में
Jani.
उनको साउंड नहीं जा रहा ना?
I think.
No, no, he's putting.
Sir, can you hear me?
No. But earlier he was hearing, no? We
It
were able
was,
to talk to.
I think you connect your, yeah.
Then
Now can you hear me, sir?
You are talking to me?
Yes, yes, sir.
Yes, sir. Oh yeah, I can hear you. Yes.
Okay, okay, sir. Sir, let me introduce that our training participant first, sir. In the
Okay.
meantime, our participant will join.
Okay.
Sir, we have 84 training participants in this virtual training program, and they mostly are from SAUs. And we have training participants from KVK, Central Universities, then ICR institutes, Central Silk Board, and also participant from international organization like ICARDA.
Okay.
Okay, I think we have now 45 participant is there. Let me introduce our today's speaker, Dr. Madhurimuthu Janvi Raman. Sir, I formally welcome you, sir, in this training program. And I'm sharing the slide.
so we kept this very important talk today to Dr. Jani on nanotechnology. So he has a vast experience on nanotechnology. Before that, he has worked on avitic stresses, long years of experience at Kansas State Agriculture University. So yes, Subhas, you can continue.
Sir, can you see my slide, sir?
Yeah,
Madam.
I can.
Yeah, okay.
It's visible.
I will take this opportunity to introduce our today's speaker, Dr. Madhurimuthu Jangi Raman. He is delivering a lecture on application of nanotechnology in PGR management. Dr. Jangi Raman is an associate professor in a department of crop physiology, Tamil Nadu Agriculture University, Coimbatore. He completed his PhD in plant physiology from Tamil Nadu University, and also did his postdoctoral research at Kansas State University, United States. Dr. Jangi Raman has established research collaboration with more than 10 international scientists working in different state universities of from Finland, Australia, USA, and different CGIAR institutes. His research area of interest and specialization in abiotic stress physiology, especially in sorghum crop. He has a wide research experience and had several achievements, and few of them I am mentioning here. First time, he has quantified the effects of a short episode high temperature stress on pollen and ovary function in sorghum and wheat. He also decoded mechanism of reproductive failure in sorghum and soybean under high temperature stress during pre and post anthesis stage. He identified for the first time the lipid signal molecule responsible for maintaining the pollen T polarity and its function under high temperature stress. He is also pioneered in the development of high throughput screening tool for identifying crow varieties more tolerant to abiotic stresses using in vitro probes. Dr. Janagiraman dissected the mechanism of chromium tolerance or susceptibility at the physiological and molecular level, which has made the way to bioremediation. Based on his findings, a sorghum variety was identified which is less susceptible to chromium damage, which is now grown in large areas which have higher soil chromium content. He has published more than 60 peer-reviewed journals, articles, and 8 book chapters, and he is presented his research work in more than 24 national and international conferences. Thank you very much, sir, for joining with us. Now, I would like to and request you, sir, to share your slides and give your lecture, sir.
Okay, so we have kept this very special lecture to the participants. So hope they all will enjoy about this nanotechnology and their application in agriculture, specifically PGR. Okay, start, sir, please. Okay,
Yes,
sir. Thank you.
Thank you. Thanks for the opportunity. And first of all, I'd like to thank the introducer for introducing me nicely. And also I thank profusely to Dr. Jyoti Kumari Madam, for giving me opportunity, for connecting me and getting an opportunity to present my experience in nanoscience and technology. Is my slides are visible? Can I start?
Yes, sir.
Okay, perfect. Thank you. Participants, you can stop me at any point of time if my concepts are not clear. Okay, I'm happy to explain in detail. Okay, let's first define what is a nano. Okay, nano means a particle which is in the range of 10⁻⁹ meter. So anything in between that range. So any one dimension of the nanoparticle from 1 nanometer to 100 nanometer makes it a different physical, biological, and chemical properties. For example, look at this. So this is a water molecule, the size of water molecule, size of glucose, antibody, virus, bacteria, cancer cell, and tennis ball. And mostly nanoparticle which will be size will be similar to the size of a single glucose molecule and in a virus. So these are the example I'm showing other nanomaterials, lysosome, dendrimers, gold nanoshell, quantum dots, fluorines. So these nanoparticles, based upon its functionality and the composition and the dimension, we can classify. That will be too fundamental to explain. So I'm just giving a brief intro and I want to move on. So here you can just see a slide. And this slide, you can, the point what I'm making here is nanotechnology is not a new science. This is in a very old ancient science, but we forgot that science for more than decades or even the years today. The first cup A is a lighter the sky, and when light was not passed, it looks green. When a light was passed, it becomes a red color. So indicating that the inner side of that glass is coated with the nano silver. So the royal kings are having their wines in that, so they lived for a longer period of time. And the second one, you can see the plate, where these plates are made up of nano silver. So this is the reason why our ancient fathers, our forefathers are telling to us, wear gold in a body in the right hand and have your food, or you have the food on the silver plate, so that some amount of silver will be taken into the body that will act as a antimicrobial activity. The third diagram C is, and this will be usually you guys will be seeing that in the churches in the European countries, and this is are made up of gold and silver nanoparticles. So they make a dichromatic glasses. So different colors will be formed when light is falls on that. And we are just thinking that it is a painting, but the purpose is to clean the air in the church. That's the purpose what my senior, our forefather used them. And F, E is a sword of Tipu Sultan, where you can see the Tipu Sultan sword, well, we know that it was very strong and it was never broken. And after it's a closer observation indicates that it has carbon nanotube. So then it was very light and giving a stronger strength. Then people now using this technology and they are making their building by instead of iron, single iron TMT iron bar, they are adding a CNT carbon nanotube to become more stronger. The same concept was used in the tennis racket. You want very lighter and a stronger bar. This is in a slide which explains to us the nanoscience is not a new emerging science, it is a very old science. And these are all the potential use of nanotechnology in agriculture. The very first application or use is the crop improvement. In this, we can develop an efficient gene transformation vector by using nanoparticle. Our nanoparticle can be used to deliver your gene of interest by using a CRISPR or microRNAs. That's the first thing we can use nanoparticle.
Sir, your voice is breaking, sir.
Through robotics. So that means how best we can cultivate the crop without human interface. So that is a precision agriculture. For that, nanotechnology is used. The third area in which nanotechnology or nanoscience is used in agriculture is soil and water treatments. Nanofibers, nanomembranes are used for water purification. Apart from water purification, so the nanomembranes are used for degradation of bacterial contaminant, heavy metals, desalinization, and nanoclays in water. So this is another domain in which cleaning up of environment is done by nanoparticles. And agriculture engineering, we use nanomaterial for coating the materials, to increase the shelf life, biosensors to quick detection of pest diseases in the seed or in the plant, and also precision weed control by machine vision or robotic weeding. And the second one is storage and transportation. And in crop production, we use nanotechnology for effective delivery of inputs. And now we are using nano fertilizers, nano urea, for example, nano DAP, and nano pesticides, nano herbicides, all these. These are the examples in which in the spheres where the nanoscience and technology is used in agriculture. And here, these are the list of potential use of nanotechnology in agriculture. We just look at here, we can apply nanomaterial through soil and as foliar. If you apply any of this method, you can able to see starting from improved seed germination to improve the sustainable agriculture. So everything, the whole plant physiologies get modified. So this is what people have observed for the last 15 to 20 years. So this made an interest to all the people working across the globe. Hey, can we have able to synthesize nano materials, and we can able to mitigate the abiotic stress, because we able to show to the people when a carbon nanotube was treated with the seed viability, seed germination is improved because CNT is acting as a water channel. And then we applied fertilizers, nano fertilizers using zeolite, the nitrogen use efficiency was improved. And we formulated nano herbicides and nano insecticide efficiency was better. And so in all sphere, we proved that nanomaterials or nanoscience is helping agriculture. And finally, in this global climate change era, people are asking us, hey, can you develop any nanoparticle which can able to mitigate the abiotic stress? Yes, look at this picture. You have a different option. Cerium, zinc, iron, silicons, iron, Mn, Mg, zinc nano carbon nanotubes, magnesium. So many nanoparticles are there, which can be applied both in the soil and foliar to mitigate salt, drought, heat, cold, pest, pathogen, bacteria, and also weeds. So many avenues are there. So this is what my interest is. So we started research on this area for the last 10 years, and we have developed or engineered 4-5 nanomaterials, and we are evaluating them under different ecosystems. And also we are trying to prove them whether it has any beneficial effect to the plant species. Okay, the first particle I want to discuss is selenium. So our objective is to improve the high temperature tolerance in sorghum by foliar application of selenium. So this is what our interest is. So this graph or the picture explains what happens to the future. So we all know that the future climate is going to get increased with the air temperature of 1.5 to anywhere to 5.5 degrees Celsius. So the globe will be more hotter. And under high temperature stress, the most common abiotic stress or the mechanism which affects the plant is oxidative damage. And we all know that selenium is a strong antioxidants which are used by the human people to delay the aging process or to improve their resistance or immunity. So we found that, and also the report says that the people who are affected by acquired immunodeficiency syndromes are given with the selenium particles. Then we discussion with the people, so they said that selenium is a strong antioxidant that can able to reduce the infection rate. And our previous result indicated that high temperature causes oxidative damage. Then we connected these two dots and asked our question, hey, can we synthesize nanomaterial named the selenium and we can counteract the oxidative damage, which is a very common under high temperature stress. So our hypothesis is through the antioxidative property, selenium nanoparticle can elevate the oxidative damage in sorghum under high temperature. So the objectives were to investigate the effects on oxidative damage, lipids, on pollen grain function, grain yield of sorghum. So how material was synthesized? We have used a very simple method, a simple chemical method synthesis. We used ascorbic acid as a reductant and used the precursor as a sodium selenate. So sodium selenate was dissolved in the conical flask 100 ml, and ascorbic acid was added drop by drop from the burette, and then the solution was turned from colorless to colored red or orange. Then the product was centrifuged and it was washed with water and ethanol 5 to 6 times to devoid of any sodium, and it was dried down at 50 degrees Celsius. And then the product was looked very deep red in color. So this is a synthesized selenium nanoparticle. So the material was synthesized and it was characterized by using scanning probe microscope, scanning electron microscope, transmission electron microscope, high resolution transmission electron microscope, X-ray diffraction, thermogravimetric analysis. And then this nanoparticle, once we synthesized, we want to test up for its toxicity. So we used mouse fibroblast L9 to 7 cell line culture and the viability by using MTTTSA and a DAPI staining. And the uptake and translocation of selenium was done by using ICP-OES by using sun culture method. So what we have done is we have grown DK28 sorghum. And from sowing to booting, the plant was maintained at 30 to 20 degrees Celsius, day and night time respectively. And the experimental design was split plot, and we had 10 applications. In main plot, we had a temperature regime, So one group was maintained at 32-22, and another group was maintained at 38-28. And the foliar sprays were water spray and a nanoselenium 10 milligram per liter. So duration of the stress was 10 days. And we gave 4 hours of high temperature stress only. So these are all the different trials, physiological trials we recorded, and these are all the references for that. These are all very standard method, you guys knew it. So what we have done, we measured the gas exchange, we measured oxidant, various types, we measured antioxidant, and we go for lipid analysis. And then the reproductive trial, we measured pollen germ, seed set, seed yield, and data analysis. Madam, is that I'm audible or
Yes,
anybody's-
Yes,
audible. It's audible.
yeah, because I'm talking, no one is interacting. So I'm just
Okay, okay. Achha, generally they are interacting
At the
means
end.
after the lecture.
Oh,
So
okay. After
if
the lecture.
okay, so
okay.
if
Okay,
you
okay.
use means they can interact.
Okay, fine.
Sir,
Thanks.
sir, one more question. How we are
Yes.
maintaining the temperature regime?
So the temperature regime was maintained. The plant was grown in a convirant growth chamber. So the growth chamber was given with 38-28. So the growth chamber has an heating filament and it has an water sprinkling system. So the water sprinkling system maintains the relative humidity, and the coil heats, the infrared heater maintains the high temperature stress. The air was heated up. The heated air was blown inside this chamber. This is how we maintain the high temperature stress.
Okay, sir. Okay. Thank you.
Okay, good. And these are all the results. Look at here. So these are all the spikes. Do you see my mouse? Do you guys see my mouse? No?
Yes. Okay.
Mouse, okay. Okay, there you see so many spikes, and I have shown it in the arrow mark. This is 3-dimensional information. So X, Y, and the Z. And the left-hand side is a 2D information, and the nanoparticles are small dots, and you can see the nanoparticle range is maximum is 20 nanometer size. So the nanoparticle size of selenium, what we synthesize, is in a 20 nanometer in size. And this is the image which we collected from the left-hand side image is a scanning electron microscope image. And the right-hand side image is a transmission electron microscope image. You can see, and the insert is an EDAX, X-ray diffraction. You can see only pure selenium presence, no impurities. So index indicates only pure selenium, and the material is less than 40 nanometers. And this is a high resolution electron microscope image. You can see selected area electron diffraction, SCAED. So what we have done is we have focused on a single atom, and we got the electronic configuration, and we found that it follows in a clean hexagonal phase. So this is a clean hexagonal phase, and that we got it. So one, the FFT, for fast Fourier transformation image, expands this is hexagonal. Then we found that our material is pure crystalline material. So we got this perfect lens. And apart from that, we want to verify the H atom results. So we also did for X-ray diffraction and the thermogravimetric analysis. And you can see a typical formation of peak at 101. This is nothing but growth of the nanoparticle in 3 dimensions. The first one represents growth in X plane, and the second 0 indicates the growth in Y plane, and the last one indicates the growth in Z plane. So that means it indicates that most of the particle was growing in as a rod, because we don't got any Y phase, so we got X and Z. So these are the rod. This is what the inference we got it from the XRD information. And this is in a thermogravimetric analysis graph, and you can see all the particles are completely volatilized at a 400 degree to 450, which indicates sublimation of elemental selenium, which is happening at this temperature. So this characterization indicates that the particle what we synthesize is very pure, and it is crystal, and the material synthesized is a nanoselenium only. So we confirm that. Now we started our routine experiment. Namely, first, we want to test the toxicity, whether synthesized nanoparticle is toxic to the cell line culture. So I already told that we used the murine cell line culture, murine L5909 cell line, and we found that we treated the nanoparticle with the cell line culture and we incubated for 48 hours. And then we recorded the OD value. And then we considered the OD value of the control as 100%, remaining as the percent inhibition. And you can see that at more than 50% viability was observed at 250. So LD50, EC50. So that is what the mean is, TC50, toxic concentration. So from this, we found that it was close to 275 milligram per liter, or otherwise 250 ppm. So that is the result from MTT. And this is the DAPI assay. You can see the blue color. So once the blue color is visible, that means the nuclear membrane is intact. And if that blue color is not clear and it is not enclosed by a perfect membrane, the shape will not be maintained. Once you could not able to see a perfect blue, that means along with the surrounding, that means its membranes are damaged. That is the conclusion you need to make from the DAPI. And you can see at 0 and 100 and 250, there is not much difference or damage to the margin of the cells, and no abnormal or condensed chromosomes were observed up to 250. Whereas under 500, you can see presence of apoplastic cells, and you can see the damage of the genetic material. So that means at 500, the nuclear material, namely the genetic material, was getting damaged by presence of nanoselenium, indicating that L929 cell line was adversely affected by nanoselenium at a concentration of 500 milligram per liter. So that is 550. And then we have identified the concentration. So we could able to see that anywhere close to 250, it was not toxic. But however, we don't want to try more than 100 because the human dietary requirement of selenium is only 40. So we want to try up to 100, double the concentration. And we found that up to 50, the growth and the concentration of selenium in the root and shoot was proportional. Whereas when you apply huge quantity, more amount of selenium was accumulated in the root and a lesser amount was accumulated in the shoot. That means in this experiment, the plant was grown at sand culture. In the sand, we applied selenium, nanoselenium. That means there was a less movement from the soil to from the root to the shoot. That's the conclusion, but it is moving. That's the point, because we observed a significant selenium concentration in the shoot at 100 and 50. And this is 0.001 microgram per kg. Okay, now these two experiments indicates that the material was non-toxic up to 50, and then this material is able to move from one cell to another cell through the simplest pathway. This is the two conclusions we got from the toxicity and also translocation experiment. And then once this was proved, then we started our sorghum experiment. In sorghum, I earlier said up to booting stage, the plant was maintained at 32 to 22 degrees Celsius. And after that, it was moved to a growth chamber, which was maintained at 38 to 28 degrees Celsius, day and night temperatures. You can see during the stress period, we have documented the impact of chlorophyll content on the thylakoid membrane damage. You can see these two bars, which is in green, and this is an orange. These two bars are always indicates optimum temperature. Red bar and the purple bar always indicates the high temperature effect. The first bar is control, second bar is nanoselenium. So you can see under high temperature, there is a higher thylakoid membrane damage, which was documented as F0 bar FM ratio, which is on a chlorophyll A fluorescence index. And when we apply nanoselenium under high temperature stress, the damage was slightly decreased. And you can see the photosynthetic story. There is no variation in nanoselenium spray under optimum temperature, whereas high temperature there is a decrease. However, foliar spray of nanoselenium there was a slight increase in photosynthetic rate. Similarly, there is an increase in the stomatical conductance. Then we all know that high temperature will increase the oxidative damage. That's what our hypothesis is. As per our hypothesis, we have observed a higher concentration of superoxide radical and hydrogen peroxide. This is what's shown as an error mark under high temperature stress control water spray plant. Whereas nanoselenium, you can see there is a significant difference, and there is no variation under optimal temperature, indicating that selenium nanoparticle could be able to decrease O2 minus and H2O2 under high temperature stress. And the proxy for the membrane damage or membrane integrity, that is MDA, which was higher under high temperature stress water sprayed plants, indicating that the membranes are highly sensitive to high temperature or it was damaged, whereas spraying nanoselenium has lesser damage, indicating that the integrity is maintained. Same story we observed for cell membrane damage also. Then the second portion of our question is, apart from the decrease in the oxidant level, whether nanoselenium is increasing the antioxidants, that's what the question asks. Then we analyzed all the antioxidant enzymes, namely superoxide dismutase, catalases, peroxidases, and ascorbate peroxidases, and we found that in here this is an SOD, this is CAT. In both high temperatures decrease antioxidant enzymes. Nanoselenium sprayed at a higher activity of this enzyme. So the product is lesser produced, and also the enzyme responsible for cleaning is also higher, resulting in better protection at high temperature stress. And similar to SOD and CAT, CAT, we have same trend was followed in peroxidase and glutathione peroxidase. The purpose of estimating glutathione peroxidase is selenium is a cofactor in glutathione peroxidases. And so we want to make sure that whether the increase in the antioxidant enzyme is due to the expression of glutathione peroxidase or SOD. This is the question, indirect question we asked to ourselves. So we measured this GPX and we found that there is a higher increase in the GPX. So the selenium, foliar spray selenium, has been incorporated in the glutathione peroxidases that might have increased the enhanced activity of glutathione peroxidase. Then this story till now, it is very clear that the leaf, the vegetative biomass, is experiencing oxidative damage under high temperature stress, and the foliar application of selenium has improved the photosynthetic rate by decreasing the oxidant level and increasing the antioxidant level. That means more amount of photosynthesis can be produced under high temperature stress when it was sprayed with the nanoselenium. Then we have another question. Hey, yes, whether the carbohydrates which are produced under high temperature stress by selenium additional amount, whether that is responsible for increased graining or it is really helping the reproductive tissue to withstand. So then we documented what is the germination percentage. And we found that high temperature stress-free plant had a pollen germ of 50%, whereas nanoselenium sprayed had a germination percentage of 62. So 12% increase in the pollen germination has resulted in a higher seed set percentage. So increased seed set means more number of seeds per panicle. So we got a higher seed yield, and because there is no variation in the seed size, because the stress was given during budding stage only for 10 days. After that, the plant was maintained back to normal temperature, namely 32 by 22 degrees Celsius. And then we want to know, hey, really, what is the mechanism which is operating in the plant under high temperature stress and a nanoselenium sprayed plant. And here you can see this is an optimum temperature, high temperature stress. High temperature is red color, optimum is green. You can see the unsaturated fatty acids, 36:6. and 34:3 or 34:2 is decreased significantly under high temperature stress. So what that means? Under high temperature stress, the saturation level of the lipids in the membranes or thylakoid membranes or cellular membranes or cellular membranes becomes chances for them to get transformed into a solid phase. So this prone for attack by the RYS resulting in the membrane leaking. So our MDA data, cell membrane damage data is validated in our phosphatidylcholine data and MDDG. I have represented only MDDG and PA value because 80% of the thylakoid or the chloroplast lipids are made up of MDDG, and PA is a precursor for synthesis of all the phospholipids, namely phosphatidylcholine, phosphatidylethanolamine, phosphatidic acids. And you can also look at the phosphatidylglycerol and the phosphatidylcholine, similar trend, namely nanoselenium are in a higher levels or higher level of unsaturated fatty acid. That means it tried to maintain the fluidity of the membrane. So with this, we are concluding the first experiment. Nanoselenium synthesized by chemical method is pure, having 40 nanometer, and it is not toxic to mouse cell line culture up to 250 ppm. The material can move from the root to shoot. High temperature stress increased ROS and membrane damage, and high temperature decreased antioxidant enzyme activity. And also the lipid unsaturation level decreases. Foliar spray of nanoselenium under high temperatures decreases ROS, namely O2 minus and H2 and membrane damage. The antioxidant enzymes, namely SOD, CAT, FOX, GPX, are increased by nanoselenium. Nanoselenium foliar spray increased membrane unsaturation level, and foliar spray of nanoselenium at high temperature increased 6%, resulting in increased grain yield. With this, we finished the selenium portion. If there is any question, I can take it up. And then after this, we have a cerium. If you want, I can continue the cerium.
Yeah, participant, please ask if you have some
Worries, any
some
discussion,
short
or
queries.
there is a chat. What is the question they are asking? Okay, that's a meeting. Fine. If there is no other question, I can start with the cerium. Okay, fine.
Okay, sir. You
Continue.
can proceed.
Continue.
Uh-huh.
Yes, ma'am. I'm continuing. So after this selenium, we just come across another nanoparticle, cerium. Cerium is a rare earth metal. So what is that? Rare earth metal. So which is very heavy metal. Its molecular weight close to 175. So, but it has a strong antioxidant property, and it was used as a chemotherapy for the cancer patient. And it is also used in the battery of the cell phone or the laptops or any of the maintaining the charge. That means it can able to maintain the charge potential. That's what we got from our literature review. And we also interested to know, hey, whether similar to nanoselenium, nanocerium is also having the same effect or not, because both are having the same electronic configuration, having the same valence state, and having the same property. So to validate this, we have conducted experiment under drought. So our hypothesis is cerium nanoparticles have possessed this antioxidant properties. So it will alleviate the oxidative damage under drought stress. So we synthesize and characterize and to quantify the nanoparticle different species and elevation potentials. So similar to selenium, we have 3 objectives: to study and characterize, to quantify the effects on pollen physiology and antioxidant defense system. So how we synthesize? We used cerium nitrate as a precursor, and we used NaOH as in a reductant. And then we slowly titrated and we got yellow color powder. And this yellow color powder was washed and then deionized with water and ethanol for 5 to 6 times and then dried them here for 100 degrees Celsius. And you should know that cerium is one of the important rare matter element which is used in all type of batteries. Now we are using a thin batteries, liquid batteries, where cerium is used because of its property, specific property. It has to withstand a high temperature. So we dried at 100 degrees to eliminate the water. And then it was calcinated at 450. Calcination means nothing but under heat them under high temperature in the absence of oxygen. So it becomes perfect. Yes.
नहीं तो उसको बोलो कि पहले पहुंचा दे.
Please mute yourself, please.
Hello.
अरे ए पैदल क्यों जा रहे हैं?
अरे please mute yourself.
Mute yourself.
Oh, it is not related to us or somebody else. Okay. So we use atomic force microscope, transmission electron microscope, high resolution transmission electron microscope, XRD, and a TGA. So we used all these technologies, which is similar to cerium, and we want to study the topography of the nanoparticle, microstructure, ultrastructure, crystallinity, and also its thermal property. Now, look at this image. So earlier we have seen selenium nanoparticle. Selenium nanoparticle, you have seen it is in a single nanoparticle. And the material is pure crystalline. And whereas in the cerium, you can able to see it is in a rod shape. You can just see just like our human hair. And the thickness is close to 20 nanometers. So we have synthesized the cerium rods and the nanoparticle size is close to 20 nanometers. And you remember the electronic configuration or the EDAX pattern of SID pattern of selenium. You saw an hexagonal shape, 6 dots, whereas here it becomes well crystalline, a single crystal. And it's a pure single crystal where you can see without any impurity, we got pure cerium. And X-ray EDAX pattern also proved that it is a well crystalline nanoparticle. And here in XRD, we got in the growth in the triple plane, 1, 1, 1. So all the X, Y, Z has grown in cerium. In contrast, the selenium we got 1, 0, 1. In TG analysis, and we have heated up to 400. So there is no weight change, which indicates that the particle is pure and all the impurities were removed at here. And the calcination temperature was close to 400 degrees Celsius. And again, we tried and tested its toxicity of nanoceria using the same cell line culture. And the first one is a DAP assay. You can see at 250 nanoparticle, which is similar to even cerium. So that means similar to cerium. So similar to selenium, cerium is non-toxic up to 250, but it damages 100% in 500 milligram. You can see there is no single cell is visible like this under 250 or 100. All these cells are getting damaged. So the whole cell in culture was damaged, abnormal nucleus, the margins are condensed, the chromosomes are stained, indicating that the cell membrane genetic materials are getting damaged under higher concentration of selenium, or that means genotoxicity is happening at 500 milligram per liter of nanocerium. And then we want to test whether the nanoceria is toxic to aquatic ecosystem. So we have tested them for whether nanoselenia is toxic to azolla. So we put nanoselenia in the medium. Azolla was grown in a airy medium. So you can see 5, 10, 25, up to 1000 ppm we used. And we found that there is no growth inhibition and no phytotoxicity for azolla. And then we extracted the anabinum from the azolla. And then see whether the azolla there is no change in the growth of azolla, but what happens to the anabinum? And we found that up to 100, the structure of anabina was maintained. After that, there is a disintegration starting at 400. So you can see all these are getting disintegrated. This is a control. So so we conclude that up to 50, this is fine. Even from 100, the damage is slightly happening in Azola. And then we looked into the phone. If the nanoparticle is not affecting the Azola, whether it is affecting the phone, yes, we have seen a dark accumulation of anthocyanin, which is an indicator of the stress, which is happening at 50 ppm. That means the Azola is protected due to the presence of huge quantity. And then we have also tested the growth inhibition effect of nanocerium on microalgae. So we have grown the microalgae in the different concentration of nanoceria, and we found that up to 200 microgram, there is an increase in the growth. After 200 ppm, the microalgae got precipitated naturally, resulting in the overgrowth. And then we want to test whether the nanoceria nanoparticle is toxic to plants. So we collected the pollen grain of the sorghum and we tried to germinate in a different medium containing different concentration of nanoceria, and we found that the germination is happening up to 200 ppm. You can see the pollen tube growth here, up to 20%, whereas at the 500 and 1000, no pollen is germinating, 100% inhibition, whereas 50% inhibition is happening at up to a level of 25 ppm. And then we want to check whether the nanoparticle of ceria is moving inside the plant or not. So we used the sand culture and we found that yes, the nanoparticle from the root, because the nanoparticle was applied only to the soil and we detected the ceria in the shoot. That means it was taken up by the root and through transpiration stream, it is moving to the root system. Again, so our next question, whether foliar spray of nanoceria could able to elevate the drought stress in sorghum plant. So we used a 430, Coimbatore 30 plant, and the plant was grown from sowing to booting under 100% pot capacity in a split plot design. So the plants are grown from sowing to booting under 100% pot capacity, and the design was split plot design, and we had 10 replication, and main plots where irrigation regime subplots were foliar sprayed, and one group was maintained at 100% pot capacity, and the second group was maintained under drought for 21 days, and the foliar spray was in foliar spray, the one was water spray, and another one was 10 ppm of cerium nanoparticle. And the duration of the stress we given is 21 days. And similar, the same right what we documented in selenium. The same methodology, same traits, gas exchange, oxidant membrane damage, antioxidant enzyme activity, and the reproductive structures or reproductive traits were recorded here also. And here the green color and the purple color indicates the irrigated condition. Red and the gray color bar indicates the effect of nanoparticles under drought situation. Similar to nanoselenium. Here you can see the first slide. I'm following the same pattern so that you can remember the sequence. Foliar application of nanoselenium improved the Fe per FM ratio. That means which is the inverse of thallic membrane damage. So damage is less, so the ratio is higher. Quantum yield is higher. And then the photosynthetic rate was decreased under drought stress, whereas foliar spray has recovered the photosynthetic rate under drought stress. And we have another question, whether the decrease in the photosynthetic rate is due to the closure of stomata or increase in the epicuticular bag disintegration, integration of epicuticular bags. We can see these dots, yellow color one coming out, and whereas under nanoselenium spray, you can see the waxes are smooth, the leaf surface, and you can see the stomates are open, so that and even under drought nanoselenium spray plant, can able to do the photosynthesis. This is the document we observed, which explains that nanoselenium improved the photosynthetic rate by opening partial closure of stomata, and the wax was maintained. And we want to look into the xylem anatomy, phloem, and mesophyll anatomy. So naturally, everybody says that drought, the xylem area or the xylem diameter will get decreased, resulting in better absorption of water. However, we found that there is no variation in the leaf or vascular bundle anatomy under drought under irrigated condition, because the plant stress was imposed only at the booting stage. At the time, the leaf, all the vegetative growth and development was finished. Again, we have another question. Nanoparticle, namely nanoceria, is really reducing the oxidant level or increasing the antioxidant enzyme activity. So we documented the oxidants, namely superoxide radical, hydrogen peroxide, membrane damage, and the thiobarbiturate acid or MDA content, and we found that under drought stress, the superoxide radical concentration is higher than nanosprayed plant, and hydrogen peroxide content is also higher than nanoceria sprayed plant. The MDA is higher than the nanoceria plant under drought stress, indicating that nanoceria could able to clean up the system, resulting in maintenance of the membrane integrity. The antioxidant enzymes, namely SOD and the catalase, was monitored, and the results indicated that drought stress decreased the antioxidants, namely SOD and the catalase enzyme activity, and the foliar application of nanoceria has improved the enzymes, namely SOD, CAT, and peroxidase. This is an image which is showing the effect of foliar spray of nanoceria and pollen germination. Here you can see this is in a controlled water spray, and this is control, that is irrigator plant, nanosprayed plant. In both this plant, you can see all these thread-like structures. So no inhibition in pollen germination of sorghum when the plant was exposed to nanoceria. Under drought condition water sprayed plant, there is a poor pollen germination. Most of the pollens are not germinating, whereas when it was sprayed with nanoceria, you can see there are tubes indicating that there is a restoration or the membrane, or the pollen membranes or pollen function is maintained by nanoceria spray, which resulted in better seed set. You can see there is an increase in the seed set. So, and all similar to Serenia, we observed no change in individual seed size. So we got a higher amount of grain. So this is our conclusions. So drought stress produces increased ROS content, ROS production. Drought stress, the pollen function is lost by increased ROS production, and the foliar spray of nanoceria reduced the concentration of O2 minus or H2O2, or I can say it dismuted the ROS into normal one, and the pollen function was restored, resulting in a better grain set and the grain. With this, I finish this, and I would like to acknowledge my mentor, Dr. Prasad, and Ruth Welty, and Bosman, the professor of chemistry, who is the man who really motivated me all behind my nanoscience and technology. And this is the person who always helped me in lipidomics, and he is my mentor. I'd like to thank the Kansas State University for the opportunity to work at Department of Agronomy and Chemistry for 6 years. And I'd like to thank the TNAU for the opportunity for sending me to for a postdoctoral training. Happy to take up any questions if you have.
Okay, dear participants, you can ask some questions if you have.
Sir, one question is there.
Yes, please.
Is there any adverse effect on the forage quality as far as sorghum is concerned when we applied these nanoparticles?
Forage quality. Okay, sir. We applied only 10 ppm, 10 milligram per liter. So I just gave the samples to my veterinary scientist. The doctor is here, and he said that up to 60 ppm we can, up to 60 milligram per liter of nanoceria or nanoselenium per kg, I'm saying, per kg can be fed to the cattle. So there is no toxicity effect on the animals. And for your information, my field experiment, sorghum plant sprayed with nanoselenium and ceria was taken by the university and it was fed to the cattle. Nothing was happened.
Excuse me, sir.
Yes, please, ma'am.
So this is Dr. Suma from US Dharwad, Principal
Yes.
Scientist, Plant Breeding. Have you worked on increasing the efficacy of bioagents through the use of these nanoparticles, sir?
That is on a different domain, ma'am. So for that, what we can do is, what the best option for me is to, what I will suggest you is, we can form a different formulations. For example, we are claiming that only 20% of the foliar spray chemical was taken up by the plant and the remaining 80% goes as a waste. This is our claim. And we say that for a systemic insecticide, fungicide or herbicide, only 3 drops per plant is sufficient to cause the desired effect. However, we are applying more than 200 droplets. So the purpose is low penetration, low mobility. For that, we have nanoparticles and we have nanoformulations. So that will increase the efficiency of absorption by the plant system. That there is a technology right now available with Syngenta. It is called as nano drop. If they're having a chemical, if you mix 1 drop of nano drop to our foliar spray, they are saying that the size of the particle is reduced up to 200 times. So there will be direct infusion. I'm saying infusion. Remember, infusion. It's directly penetrating the plasma membrane, even up to there, and enters into the tissue. That's what their claim is.
Yeah, so anything on this, it would be very beneficial for farmers because they're spending so much on fertilizers. And if there's a breakthrough in this, then it will be helpful, sir. At a low cost, they can meet out the nutritional requirement of the plants.
Yes, ma'am.
Without
perfect. So what we are doing is only we synthesized. After that, once we go for the field spray, I'm formulating oil in water formulation, and we use an egg yolk and a milk casein protein for dispersion of nanoparticle. We are also using chitin from the orange peel, and we are also extracting the chitosan, and we are making them into negatively charged, and then we are dispersing our nanoceria and also nanoselenium, and then we are making a stable emulsion, and then we are spraying. That increases our bioefficacy. This I have presented only 2 metals. We have been working on a zinc selenide, and also we are working on composite zinc selenide ceria, and we are also working on iron oxide, zinc oxide, and a copper oxide. So these are all and the silicon too. So we are working on this to improve the abiotic stress tolerance and also productivity. We and also disease. We are using silica to control insect and also to pathogen. And for improving the seed germination, we are using hydrogels and methoxycellulose. I have not touched the seed portion alone because I thought this will be more interesting to you. Methyl cellulose and carbon nanotube experiment and zinc oxide, these 3 nanoparticles are very prominent and what I can say striking effect on improving the seed germination in close to 20 crop we tested. 20 crops. In all the 20 crops, zinc oxide 100 ppm, 0 valent ion 50 ppm, and methoxycellulose. In combination of these 3, we can sustain the germination or improve the germination by 15%, or we can maintain the feed germination at 90%. So that is on a great presentation. That's another presentation that will go for another 1 hour. I didn't touch that.
So, sir, can I ask you another question?
Yeah, sure, ma'am, go.
Yes, sir. Sir, how do you exactly prepare these nanoparticles, sir?
Ma'am, we have a chemical vapor deposition, CVD. We have an instrument. So what this is another method I'm saying. Just like our cooker. In cooker, we keep our water and then we cover with a lid. Once the water gets boiled, the vapor will go and hit the lid and then it condenses and then it falls back. Okay, this is a normal process which everybody is very aware of that. Similarly, what we do, we keep in the bottom cerium nitrate. And once we heat up to 450, 650 degrees Celsius, this cerium nitrate will be converted into plasma. And then it tries to rise up. And then it sticks on the surface, on the top, where we're having a sort of tapes where this material will be atom by atom, layer by layer, this go and deposit. After the complete combustion of the precursor, we switch off and then cool to room temperature, and then we scrape up all this nanoparticles, and then we ground them, and we could able to get pure nanoparticles. This method is CVD method. Another method, a simple chemical method, where the reductant and the precursor were allowed to react. In this our experiment, we used cerium nitrate dissolved in water, and then to that solution we added NaOH drop by drop, till the change in the color from colorless to yellow. And during this process, we need to keep this solution in a 60 degrees Celsius, and we need to keep it in a rotary shaker or you can a magnetic stirrer, and then we need to maintain the RPM at close to 100 ppm, so that the material will be synthesized and deposited back. And if you want a bigger particle, we can leave it alone like this, or you want a smaller, you can add a capping agent, namely PVP or CTAB or TVN20, so many things. It's a very simple, ma'am, according to me. Earlier I thought it is difficult. Now I think it is very simple.
Sir, actually, can we make use of the sunlight also by keeping the sun?
Yes, people's right now, our partner in engineering department, he's using sunlight to synthesize copper oxide nanoparticle. So sunlight is driving using the sunlight for synthesis of zero energy. Is synthesizing copper oxide nanoparticle.
So, and
we have several other questions in chat box. Suma, do you have some more questions?
Yeah, ma'am.
Please be quick.
Yeah, ma'am. I forgot what. Yeah. Somebody has asked in the chat box only what I had to ask. Actually,
Okay.
we use heavy metals or chemicals. The disposure of these this thing aftermath, whatever you use, that becomes difficult, sir. So what's your take on green nanoparticles, sir? From plants only we can use.
Yes. So in a green synthesis, what we are trying to do is we will be using the same nanoparticle, namely the same cerium nitrate precursor. But instead of reductant, we will be using the extract from the parthenium, and we will be extracting from the tridents, precumbens, and then extract from all the medicinal plants. They are rich in phenols and carbohydrate reducing agent, ascorbic acid. So that will convert zinc oxide. And our previous experiment in Gingerly, we synthesized green zinc oxide by green synthesis approach. So what we have just dissolved zinc oxide, zinc nitrate in the water or zinc chloride in water, and then we added the trident procumbens solution, which is these trident procumbens are kept in water, soaked in water for 24 hours, and then we filtered, and then we added to that, and then we got that white precipitate. We filtered, washed, cleaned. This is green synthesis. So here, what is our question is, green synthesis means don't think that it is non-toxic to the plant or it is compatible with the plants, not like that. Here, green means energy. The energy for the synthesis is totally avoided. That is called a green synthesis.
Okay. Thank you.
People will be having two notions. Green means it is safe, so we can spray. No.
Yeah.
Green means eco-friendly.
So one last question. What's your take on nano-urea, sir?
What is that? What is the question, nano-urea?
What's your take on nano-urea, sir? There are some disadvantages also, I feel. Nano urea.
Yeah, actually nano urea, there is no disadvantage in nano urea, but the farmers has to be clearly educated on use of nano urea. Nano urea has to be applied only as a foliar. It should not substitute the soil applied nitrogenous fertilizer. Clear?
Okay.
So for if you're using a nano urea, the soil has to be applied with the urea. On top of that, you can spray nano urea. So the nitrogen requirement can be met. So please don't advise the farmers, no need to apply soil fertilizer. So this is what we are facing with rice farmer here. Are you audible to me or I am audible?
Yes, you are,
Hello?
sir. You are, sir. You are.
Okay. Here, what is the problem we are facing with Tamil Nadu farmer here? The farmer, since the government is promoting nano urea, and the farmers are asking what it has, we say it is a urea. So they are very reluctant to apply fertilizer, urea as a basal. We need to apply 50% of the urea as a basal, and the remaining, as no need for crop dress, instead go for urea spray. That is what the government recommendation is. And the disadvantage, according to me, this is my personal opinion, I'm saying, the nano urea was commercialized without testing for 3 seasons in a different ecosystem. This is what I have seen from that IFCO. So it was evaluated only for 1 season at one institute, not a different ecosystem. That's what my opinion. Personally, this is my personal opinion.
Yes, Dr. Jani.
Yes, madam.
I think another question has been answered. Dr. Sunil from Kerala. Does nanoparticles have environmental hazards?
Yes, of course, it will have environmental hazards. We need to, before using in any experiment, you need to see for its environmental safety. That's why I have shown the images or the data on 3 different ecosystem or trophic level. Soil, I have not shown the effect of nanoparticles on rhizobium growth, bacillus growth, phosphobacteria growth. So that explains the soil level. And in aquatic system, we need to show them in azolla, microalgae, fish. I have not shown the zebrafish result. So these 3 explains the effect on aquatic system and terrestrial, photosynthetic rate, pollen germination, biomass. So which explains effect on soil, effect on water, effect on the aerial plant. So we need to make sure there is no impact of nanoparticle on any of this ecosystem. If it is affecting anything, then it is environmental hazard.
Okay. Another question,
Wheat
Kumar
crop,
from...
yes. Vinod Kumar, yes. This wheat crop you can use nanoparticles, but make sure what is the purpose. Nanoparticle having many number of purpose. So that's why the first 2 slides application, I explained 5 minutes on that, because please don't carry away that nanoparticle is very good to the agriculture. We can improve. No, that's why I showed in everywhere. Yes, we can use in wheat crop for pest control, disease control, improving the iron and zinc content. Then what about the residual effect on the straw and the soil? Yes, sir, we are working on that. And the soil study is called as generation effect. So once you spray any nanoparticle and it falls on the soil, again, we need to sow the another crop without nanoparticle, and then we have to make sure, see how many generation it takes to clean that soil with nanoparticles. That means I sprayed 2 milligram. In the plant, I got 0.5, remaining 1.5% in the soil. Then you need to grow the next crop and then see how much is present in the soil. Then you need to grow and make sure that at what generation the soil is clean from the nanoparticle. This is called a generation study. We are working on that. But across the globe, people are working on that. Now we started with sorghum.
Yeah. So
yes, madam. I have answered all the questions, ma'am.
Yeah, all the questions are answered very nicely, very beautifully. You have taken the question answer session. So it was a good interaction with the participants. So I think all the participants have enjoyed this upcoming technology, which will be widely used in the agriculture. It has been started being used, like for fertilizer application. So slowly it will be taken up in a big way. So so thank you very much, Dr. Jani.
Thank you, madam.
Yeah, for delivering the excellent talk to the trainees and enlightening with the new views about means what it can be applied in the agriculture and in PGR of different crops also. So thank you very much for accepting and delivering your talk to the trainees.
Yeah, you're most welcome, and thanks for the opportunity. Have a good day. Thank you.
Okay, sir. Thank you. Sir.
Thank you. Bye.
Bye. So now we will have our next lecture. Just me check whether our speaker has joined or not. Okay, sir has joined. So our next speaker for today's session is Dr. K. Suda. So before he will start his lecture, let me introduce sir. So whether the slide is visible?
Yes.
Okay. So Dr. K. Suda, he is presently Principal Scientist Plant Pathology. He is posted in Division of Germplasm Evaluation at NBPGR New Delhi. So regarding work experience of sir, so sir has served in NBPGR since 2019 to till date. Before that, he was posted at ICAR Indian Institute of Maize Research for 10 years, 2009 to 2019. As a senior scientist at Almora for 6 years, 2002 to 2008. And as a senior scientist come head KBK at Udaipur for 2 years. And before that, he was a scientist at KBK Ujba. Regarding research experience and research areas of sir, so he has a vast experience in plant biosecurity, post-plant resistance, PGR, pest surveillance, integrated disease management, on-farm validation of IPM technology, disease forewarning models, AI-based models. Then AI-based, he has also been involved in development of AI-based mobile app, then development of appropriate technology in maize pathology under AICRP program. After that, sir is fellow of Indian Phytopathological Society since 2014, then another Indian Society of Mycology and Plant Pathology, then Indian Society of Plant Pathologists since 2009. To his credit, he has published more than 75 research papers, then 3 new records, 11 review papers, and he has delivered 8 invited talks. So, sir, we welcome you to present your lecture on the biosecurity aspect to the participants. So, thank you. And I am stopping my slides.
Thank you, madam, for giving nice introduction and giving me the opportunity for delivering lecture on biosecurity. Thank you. My this one PPT is presentation is visible.
Yes, sir. Yes, sir.
Visible.
It's in the slide mode.
Yes, sir.
Thank you. So as Dr. Jyoti has told that I will share my views on plant biosecurity perspectives in Indian conditions. Can we remove the upper top? There is some hiding the title in the top.
Sir, it can be moved, sir, means it can be lowered. Just cursor ke saath, it can be lowered. Just try.
Okay. Oh, this right.
Move with cursor. Okay, sir.
Now it's all right.
Yes, sir.
So national perspectives on biosecurity. Actually, I attended in postdoctoral fellowship in Australia, and I studied in detail the Australian plant biosecurity system. Based upon that, there is need to revise the biosecurity laws or policy in compliance to FAO. That is still pending. I'll discuss in my succeeding slides. It's not moving forward now.
No. Slide पे click करिए एक बार, then you can.
Okay. Oh, thank you, madam. So, as Dr. S.C. Dubey has told you in his lecture that biosecurity nothing, it is the safeguarding of resources from biological threats. And what are those biological threats? Any organism that has the potential to harm the people's health and life, food and agriculture, environment and the economy. And those biological threats may be insects, mites, snails, nematodes, pathogens, and weeds, which may harm our plants, plant products, including honeybee. So these are the biological threats. And Mumford in 2008 had said that these 4 T's, that means trade, travel, transport, tourism, they are responsible for the movement of the pest along with the people, along with the luggage, consignments, people. So they are responsible for it. And with the globalization, you see the people said that we have become a village and we can exchange our products from one country to another. But with that, there is danger that along with that, the harmful organism may also move. It was estimated that around 70,000 species they are there, which can move with the movement of the people, movement of the luggage. So around US dollar 1.4 trillion worth loss is going on with this invasive pests. And this loss is much more in developing countries where still the biosecurity system is not so strong. I'll give some few examples which have moved throughout the world. This is the yellow rust, and this one stem rust, which has moved from this one African continent towards this Asian part and including our Indian. And you know how much the rust damaged the wheat crop. You see citrus wanglong being, it was reported from China, and you see from Central American countries, then Southern American continent through Africa, it has infested this one Indian subcontinent, including Southeast Asian countries. Banana bunchy top virus, you see from the American, this one continent, it has moved Africa and our Asian parts. Then recent example of Magnifica triticum. Earlier, this was a disease of rice, and ignoring the advice of the scientists of Bangladesh, some minister has allowed the import of wheat, and along with that, this disease has this one entered in Bangladesh, and along the border it was reported now. Somehow it was managed by the Directorate of Plant Protection quarantine. Then recently in 2018, fall armyworm of maize were reported in Karnataka, this part, and it has moved from the American continents, Africa, and this one, including China, India, and all that is infected. Even Australia is having a strong biosecurity. There is also fall armyworm has been reported. So these are the few examples. Even varroa mite in honeybee was reported in different parts of the world. So the plant pests, they do not respect the boundaries, geographical boundaries, which normally we say that we can make our this biosecurity strong. But every country has to make its biosecurity this one strong so that they cannot move. Another aspect of is the terrorism, that may be of 3 kinds. What is bioterrorism? Biological agents target the humans, the animals, plants. Agroterrorism is the biological threats, they are targeting the crops. Then food supply, livestock, industry, all are affected with this one. And third type of terrorism is other conventional like nuclear bomb and all. Directly they are killing the human being. You must be surprised that around 30,000 kilos of Puccinia gramis stratus spores from 51 to 69, targeting the Russian wheat by US, and 1 ton of Pyrochlora oryzae causing the rice blast was targeting the Asian rice. And you see the in brief how the people in the First and Second World War, they were targeting the crops so that there is a problem of food security. So it is very important that we should maintain the profitability of the crop production. We should maintain the security of the food production. And third one, to protect our crop supply and our natural ecosystem against the invasive pests and diseases. So protecting our plants before the infestation is much more cost effective than managing full-blown plant health emergencies. Now, if we see, there are 3 types of biosecurity system. What is human biosecurity, then animal biosecurity, then plant biosecurity, and 4th one is the environmental biosecurity. Human, that means that addresses the zoonotic and emerging disease diagnosis and administration. COVID is the recent example. That means zoonotic, the germs which can hop between the animals and people, like COVID. They are called zoonotic, and they can affect the human as well as animals. And in animal biosecurity, disease prevention, control in livestock production facilities, farm, fish farms, storage facilities, that addresses the disease or the invasive pest in animal. In plant biosecurity, we safeguard the plant industries and crop production. And 4th one is environmental biosecurity, because once the invasive pests enter, exotic pests enter in some new environment, though the indigenous species or the living organisms, they may be affected by the entry of such harmful invasive pests. So before that, I will touch few agreements. Dr. Dubey has already told that, but my purpose is that you should know what is the international framework, what is the regional framework, what is the national framework, before I go to the perspectives of plant biosecurity. So there is WTO, which makes rules for trades, but there he has agreed that there should be SPS agreement and people should obey it. And if those who are not this one complying with the SPS agreement, they have to pay for the, there is provision for penalties also. Then this is the SPS agreement, difference, they are scientifically based, least trade is restrictive. Basically, SPS, the government can apply these measures for the food safety, for the animal health, for the plant health. Then there are 3 sister organizations. Dr. Dubey has already told, this OIE, World Organization for Animal, IPCC, Plant Pest Disease Weeds, and this one, 4th one is the Codex Alimentarius, toxins, microbial contaminants, pesticides, heavy metal, all those they are addressed by this Codex Alimentarius. So IPCC, 184 countries, there is a convention, and its basic purpose to align the convention with the WTO SPS agreement so that we can save our crop. These are the different themes, activities, and ultimately the purpose of IPCC is to protect the farmers from economically devastating pest and disease outbreaks, protect the environment, protect the ecosystem from the loss of viability and function as a result of pest invasions, and to protect the industries and consumers from the cost of pest control or eradication. It also frames the international standards for phytosanitary measures, ISPM. Till date, it has made 47 ISPMs, but it is not mandatory that we should follow it. We can make our NPP also, National Phytosanitary Measures, NPPO, National Plant Protection Organization, like Directorate of Plant Protection Quarantine, Faridabad is our NPPO that represents India. And till date, we have made 24 NSPMs. If they are not suitable to us, we can modify and we can make our standards so that we can manage, we can stop the entry of those invasive pests from other countries. Then it was an international, then there are also regional biosecurity framework where the 10 regional plant protection organizations combined together for different regions, and they can make plan, and IPCC convene a meeting for technical consultation of those regions. These are the 10 regions, 1 to 10, Asia, AAPPCC, CHFSA, COSAVE, CAN, EPPO, all those, this one. But Australia is the only country which falls, this is the Australia, which falls in both PPO as well as the APCC, this one, toward this side also, toward this side also. So they make regional planning. And if you talk of the national quarantine system, tomorrow Dr. Celia will tell in detail. There is DIP, the legal framework, the state to insect and pest, under which all this one, our biosecurity system is addressed. Detail tomorrow we will listen, but I just, my purpose was that these are the legal framework of India. This is the DIP Act, and under that there are so much modifications of the PQ order as per the requirement. So these are the different schedules. There are 12 schedules in DIP, which tells about the schedule 1 tells about the what are the points of entry. In schedule 4 deals with the very high risk this one pass. So they are completely prohibited. In schedule 5 they are high risk. They are also prohibited, but some additional declaration we can allow them. Very strict. There is moderation with the schedule 6 pass, and least is this one 519. Then weeds risk is this one scheduled in the schedules 8. So these are the different points, and what type of the consignment they can be this one allowed through which entry. The domestic quarantine is also very important. These are the different pests which are covered under the domestic quarantine. You cannot move like apple scab in Himachal Pradesh. So you cannot move the infected material from Himachal Pradesh to other apple growing areas. Similarly, tomato, there is a golden cyst nematode problem is there. So different parts. You cannot move the material, infested material from towards the healthy localities. There are two systems. One is the bulk consignment, one is the germplasm. Germplasm dealt by NBBGR, and bulk consignment DAC and AFRID, Department of Agriculture Cooperation, Family Welfare. So that dealt with, there are 7 regional plant quarantine stations and 66 PQS. So detail tomorrow we'll listen, but I want to bring in your awareness that this is the Indian system. So already covered. Then even in this system is existing, quarantine system is existing, but in the last few years, there was a publication in the Down to Earth. You can go through this one. I think you should go through it and you will know many things. Why in spite of our quarantine system, the pests are coming to India, and recently the fall armyworm in 18. There are many flaws. The custom official, they do not allow to check the cargo or the luggage. So this is the statement of C.D. Mai, former president, Indian Federal Society. He said that at a time when bioterrorism is a global reality, it is imperative that we get our quarantine system in order. So these are the invasive species 173, which can enter in India and can devastate our plants, animal. So caution is that as per the FAO guidelines, why we should not strengthen our system instead of quarantine, which is nothing but a border biosecurity. At the border we are checking. We have no system or defense system like other countries that in the pre-border, in the country of import, we have some system that we can check the infestation of the pests there only from where we are importing. So there is need to revise our regulations. There is need to strengthen the diagnostic system. There is need for capacity building. In Australia, there is a biosecurity continuum approach. Continuum approach means that addresses at the pre-border and before reaching our border, and at the border we should check. And then the post-border, if the pest has entered, what techniques or activity we should undertake so that we can manage at the 3 stages. So under the biosecurity continuum, there are three these activities under the pre-border. We can assess the risk in the country from where we are importing. We can verify at the offshore inspection audits we can do. We can collaborate with those countries. And there should be some regional capacity building activities, then intelligence gathering. All pre-border activities, they should come under the Indian government domain. At the border, we can screen the material, inspect the international vessels, passenger, cargo, mail, animal, plants and plant products, so that we can manage the high biosecurity risks to our ecosystem. It should also be taken care of by the Indian government at the border. But at the post-border, my view is that there are 3-4 organizations that should combine together so that we can manage the entry of pests. Like Indian government, we can make a company like Plant Health India, the state government and UT government, they should be there, and industry, which is plant industry. So these are the three biosecurity continuum approaches: pre-border, border, post-border. We can do. We can manage the biological threats. So, like America is doing, if it is importing mango from India, so you can see how much care he is taking in Maharashtra. There are facilities they can check. If there is stone, they will reject the material. They will take only when there is zero risk with the import of mango or any other fruits to America. So there are standards, imposition of import standards, then pre-border checks and the treatment of goods. If there is some risk, they can be treated in the country itself, so that it should not reach American border. Sharing and international network with research and development biosecurity agencies. So at the pre-border, that activities we can do so that the risks even do not touch the border of the country. In the border biosecurity, we should combine together, like Ministry of Science and Technologies, Ministry of Health and Family Welfare, Ministry of Environment and Forest and Climate Change, Ministry of Agriculture, Farmer Welfare, Ministry of Finance, States and Union Territories. This is not happening presently. So my view is that different ministries, they should be involved in the biosecurity system. In the border biosecurity, we can screen, inspect and clear millions of the people, mail, parcel, baggage at the other good countries like Australia, New Zealand, China, America and European countries. They are doing it, and they are checking the entry of the invasive pest in their territories. In the post-border biosecurity, in spite of all the things, I have shown in the previous maps that FA was not reported from Australia, even that has entered in one of the territory. So risk cannot be made zero in spite of best efforts. So if there is some incursions, if there is an entry of any pest, then it must be eradicated once it is entered in the boundaries of the country. So there must be some plan to manage it. So if there is some pest is identified, then it should be easy, promptly detected and diagnostic technique using the recent techniques, and there should be response mechanism in place. So further, there should be some intergovernment agreement on biosecurity with different states and territories and central government. There should be some legal agreement that can reduce the likelihood of exotic pest and disease from entering, establishing and spreading. Then prepare and allow for effective response to and management of exotic emerging pests and disease. Take minute, can you hold? There is some phone from the director. जी मैडम क्या चाहिए? हाँ जी रंगा को सुरेंद्र जी थैंक यू sorry. So that agreement should prepare and allow for effective responses to and management of exotic emerging pest disease or ensured that a significant percentage is already in India, they should be contained, suppressed, or otherwise managed. So these three categories of pests, they should be managed through intergovernment agreement also. Then different agencies which can help in strengthening the biosecurity system, that in Indian government that can coordinate the national plant biosecurity policy. General public also should be aware. Once there should be some hotline where the growers can report on the hotline immediately that I have seen in the backyard, in the field, that there is some pest which is damaging the crop. Growers also should be aware. They can take photographs and send to this hotline. Then there's some non-profit making company like Plant Health India or any name they can give. There's a non-profiting company which can coordinate between the private industry and the government of India. Then plant industry, plant-based industry representative body should be there, and states and union territories, they should come together. These 6 agencies, they should come together to make the biosecurity system of the country strong, as good as other countries like America, Australia, New Zealand, and European countries. The plant energy company that should take care of the biosecurity across Indian plant industries, capacity to respond to plant pest emergencies, and custodian of the emergency plant pest response need. So, main purpose of this non-profit making company is plant biosecurity, how to strengthen, how to execute the plan. So it should make a deed, emergency plant pest response deed should be there between the industry and the government, so that we can take shared decision, we can share the cost, we can provide insurance to the growers, and if there is infestation of the pest, we can go for preemptive action like breeding for resistance. So any emergency plant pest, that there are 3-4 categories. It may be a new pest to India, it may be a different variation of strain of the established pest, or it may be previously unknown pest, or already existing but it does not spread to different parts of the country. These 4 these pest categories can be there, which is called emergency plant pest. So at the national, there should be 2 important committees. One should be the consultative committee on emergency plant pest, and second is the national management. This is the decision making, this is consultative and giving advice to the government that if there is emergency plant pest, how to manage it. So my suggestion is that there is the Plant Protection Advisory Government of India that should chair the consultative committee, Chief Plant Protection of the each state, representative from affected plant industry parties, this non-profit making company, Plant Health India, and in government representative should be there, so that they can discuss and they can advise the government that such and such pest sanitary in such and such territories, and they submit the recommendation to National Management Group. They are the Indian government person, secretary, DAC, maybe the chairman, and chief executive officer of these different states, and affected industry presidents, chairs, and Plant Health India, which is not running, but it will be a part in it. But it will not this one take decision. And there may be some specialist also. Scientists, plant scientists should be there, so that they can take a decision for the management. Reporting is very important. Once you see, the reporting should be very prompt through hotline, through telephone. It should be reported immediately. National Response Management, there are 2 committees, as I had discussed, Consultative Committee on Emergency Plant Pest and NMG. So this should be the procedure. Once we find that some unusual pest is seen in any part of the country, then there should be notification, initial response action should be taken, consultative committee should be there, and recommendation should be submitted to NMG, and ultimately management plan, and this one how to manage, how to restrict it, how to eradicate it. Then response plan, the status report on the emergency plant pest, technically feasibility of the eradication, all those points in the response are mentioned, and it can be revised depending on the situation. If the occurrence of the new outbreak of the in different locations, the point when agreed funding, there is financial crisis, funding is less, or other indicators of the effectiveness of responsive plan active to date, that is revisable. Then eradication strategy is made, but it should be confidential, because anything leaked to the press, that may affect the trade of the country. So everything is eradication strategy confidential, only a few talking points, what is happened, how it happened, that can be given to the press through briefing to authorized person only. No other person should speak to press, because it's a matter of trade and direct financial loss to the country. And biosecurity is not a cheaper, this one. There is a cost always involved. Then who will bear the cost? So there are 4 categories of the pest. 4 categories means category 1 pest is very high public impact. So for category 1 pest, Government of India 100% cost will bear. For category 2, high public impact, there will be 80-20. 80% cost will be borne by the Government of India, 20% by the plant industry, which is affected with the pest. And category moderate public impact, 50-50. And category 4 pest, mostly if not wholly private impact, then 20% government will bear, 80% plant industry. Such arrangement should be there, that cost should be shared between the affected industry and the Government of India. You can see we have federal structure, quasi-federal structure. The government share, that should be 50-50%. 50% states and territories, affected states and territories, and 50% from the Government of India. Industry 1 and 2, they can be shared as many as industry which are affected in that area. So such should be the arrangement of cost sharing. Always when we manage or eradicate any pests, there is destruction of the property also, like your orchard, building, all that. So if we not compensate the cost of the property, the people will not report. So to increase the reporting, always there should be some payment on the agreed valuation approach, how much loss we have caused to the farmer, that should be compensated. Then finally, there should be arrangement of monitoring of the response plan. Take from the technical point of view, financial point of view, communication updates, and proof of freedom. Once they free the pests, the territory is free of this one, that cost sharing, incident debrief, come under the monitoring of the response plan. There are some tools of biosecurity. First is the international standard fertilizer. Till date, 47 international standards are there for addressing the pest invasion. Then most important, which India is still lagging behind, on that day, Dr. Dubey has also told that we need to revise our biosecurity laws. These countries are leading countries: Argentina, Australia, Belize, Bhutan, China, Kenya, New Zealand, Norway, USA. They already revises their biosecurity laws, and they have made this stringent. You cannot take a single seed or plant part to that country. Third thing is the pest surveillance, which is still. Can you hold on, please? There is some important call. हाँ सुरेंद्र, अब मैं बताता हूँ. अभी मैं बोला है गौतम साहब को ना, मैं बता हाँ कल की है, बता. हाँ रहेगी वो. हाँ तो मैं बताता हूँ डॉक्टर साहब, मैं लेक्चर ले रहा हूँ अभी ट्रेनिंग में, मैं बताता हूँ. So pest surveillance should be strengthened, which is still not very stringent, so that we can get what are the high priority pests, what are the low priority pests. There should be some general surveillance, specific surveillance, detection survey, monitoring survey, So different survey categories are there, which can tell us that such and such territory is infested with the biological threat, and we can accordingly make plan of eradication. Then I can give some few examples of Australia, where Queensland, that is the area infested with Queensland fly or Mediterranean fly. You cannot bring apple from this one. You cannot bring. Already they will be discarded at the border. There are special arrangement of biosecurity. Nobody can bring single apple from that place. Varroa mite, there is a very serious mite of honeybees. Australia is free of that mite because of the strong biosecurity system. That fourth tool is pest diagnostics. Diagnostic system, National Plant Diagnostic System should be strengthened, which is still not so strong. Once we survey, once we find anything unusual in any part of the country, there should be some diagnostic immediately. That should be diagnosed that such and such insect is there, such and such pathogen is there, nematode is there, or something varroa mite is there. And then fifth tool is pest risk economic. There should be analysis should be there, and economically that how to manage it, how much cost will be involved. That arrangement should be there. And then finally, eradication and pest management arrangement should be there. Finally, we should communicate to the public that such and such pest was reported and we eradicated it from the country, and it could not make any significant loss to the country economy. There is a reference during 20, Mr. Athawale, just a minute. The Way Forward by Colonel Ram Athawale in 20. You can go through it, it's available on this one. He has also emphasized that there should be some effective strategy to mitigate the biosecurity risk. And he has also said that a national biosecurity expert group should ideally be created under PMO. He has suggested involving different ministries. So biosecurity is a national security matter and needs to be viewed with the appropriate seriousness and importance. It needs to be central, not a state subject. In other countries like US, UK, Japan, biosecurity is under the PMO, Homeland Security and the Ministry of Defense. Biosecurity should come under the National Chemical, Biological, CBR and security paradigm. And the central government directives and they should be there, which can be executed forcibly. They should not be guidelines. Now, ultimately, from the foregoing discussion, there is need to develop some national agricultural biosecurity system as per the guidelines of FAO, which is saying for the last many years, still we are lagging behind. And if need be, there are voluntary persons from New Zealand, from Australia, they can guide us how to make what arrangement of biosecurity system should be in India. They're ready to advise also, free of cost. So there should be some agriculture biosecurity bill that should be brought in the parliament, which can reduce the redundant and old restrictive insect pest 14 and Livestock and Importation Act 1898. Ultimately, to have a better regime of quarantining and control the pest and even exotic species, so that we can cover the 4 sectors of biosecurity: human, animal, and plant life, and associated risk for the environment. It's not that we have not made efforts. In 2013, Honorable Minister of Agriculture, Sharad Pawarji, has brought one biosecurity bill. Under that, there was a provision of Agriculture Biosecurity Authority of India, but it was half-hearted and it did not include the epizootic or zoonosis, the pests which are moving between the animal and the human, like COVID. That was not included in that legislation. Further, it does not clarify how states should act to contain the spread of disease in the country. That means domestic quarantine was also not properly taken care of. So there is a need to consider and establish proper agreements between the central and state governments to ensure commitment in implementation of quarantine measures that are compliant with the sanitary and phytosanitary agreement of WTO. These are the views of Professor Shashi Sharma, who is alumni of IERI and who moved to Australia. He was retired as Director Biosecurity of Western Australia. I have worked under him as advisor. So there are several issues in the national plant quarantine systems. There is need to strengthen the quarantine legislation. There is need to revise the BDA. There is need to review the state of domestic quarantine, upgrading the plant biocontrol stations in terms of manpower, infrastructures, expertise, establishing intrastate quarantine checkposts, risk analysis system need to be strengthened, strengthening of survey and surveillance systems. Now, ultimately, we can say that what could be done so that our biosecurity system should be as strong as of other countries. There should be effective strategies which can be implemented to safeguard the food value chains worldwide on priority. FAO says that priority areas for interdisciplinary action at promoting, developing, reinforcing the biosecurity policy and regulatory frameworks for food, agriculture, fishery, and forestry. Many countries have revised their biosecurity systems. Global perspective in our biosecurity policy and approaches so that we can maximize the benefit of international trade and globalization. Then biosecurity and food security must essentially be the priority program of the individual countries, and locally owned research and formation strategy and management systems must be in place in each country, as is rightly said. In order to be food secured, for food security, we must be biosecured also. Otherwise, the invasive pest will come in regularly in the country and our crops will be destroyed like anything. We can face a food security problem with the burgeoning population. So, Professor Pish, this one Shashi has made 3 P's, 3 P mantras. P1, produce the food sustainably, protect the food from loss in the value chain, provide the biosecure, safe and nutritious food. These 3 P are important for the country. There should be national plant biodiagnostic network like other countries. We can know which pest, which foreign pest, exotic pest had entered in the country. There should be regular capacity building program also. There should be HRD. We can learn, we can update our knowledge on biosecurity. So staff of DPQs posted different import entry points that should be trained regularly regarding the policy, regarding the diagnostic. With the recent example of this zoonosis, COVID, Professor Sharma has given his with what from COVID we should learn. There will be increase in the emergence of new biological threats. This is sure, and their inconsequent impact to human population environment is inevitable. In future also, there is no guarantee that no COVID will come. They are likely to come. So therefore, we should not think in the boundaries, geographical boundaries. We should think globally so that such epidemics could be avoided. Then we can think when concurrent epidemics of biosecurity threats affecting the humans, affecting the livestock and food plant, they come together. COVID is only affecting the human and animals, but think of that, they start affecting the food. So what will happen? Our survival will be at stake. We cannot survive without the animals, without the plants and the ecosystem. So COVID has also highlighted that lack of effective global governance apparatus. Global governance arrangement is aimed to safeguard the health and well-being of our planet. In the next 100 years, we will be around 12 billion, double. We are around 6.5 billion. Now will be double population. To feed that population, I think biosecurity system of not only of India, but the global biosecurity system should be strengthened. Then you can think of money because this is a costly affair. You see how much weapons cost is there in the world. Around US dollar 1.9 trillion in 2019 was spent on weapons. Even one fighter jet is costing around 94 to 122 million. And for flight, for use once that jet, that costs around $44,000 US dollar. So we can, the money which is spent, we are spending on the weapon that can be diverted to this biosecurity system, so that we can save our crops, we can save our human life, we can save our animal life, fishery, and ecosystem, and natural resources and biological resources. Instead of World Health, WHO, why we are not thinking of the Planetary Health Organization, which can deal with the plant health, animal health, human health, air health, soil health, water health organization. Everything should be covered with the Planetary Health Organization. So such is the views of Professor Sharma, which today or tomorrow or in future, we have to at least think that we have, for the safety of the human, animal, plants, we should strengthen our biosecurity system. In Australia, you can see the successful example of pests in which citrus canker was this one free of, Australia is free of citrus canker. They have eradicated. Then bunchy, this one, banana freckle disease was managed, eradicated from the country. Thanks with the slogan that biosecure movement of the people and goods. Thank you for patient hearing. You can discuss regarding the biosecurity. Thank you. Thank you, madam, for giving the opportunity. Hello. Hello.
सर आ रही है आपकी आवाज.
Okay, okay. I'm not listening, Dr. Jyoti.
नहीं, सब सुन रहे लेकिन अभी आगे कोई बोल नहीं रहा.
She is speaking something, but I don't know. I was thinking that there's some problem.
Okay.
Now it's okay.
So all are requested to ask question if they have any doubt or anything.
Any question or this one?
Actually, today they are overloaded with different sessions.
Anytime you can discuss. My mail ID is there, ks.huda@icr.gov.in. And tomorrow, Dr. Celia lecture is there. She will elaborate what is quarantine system of the country. So I have given based upon my experience of the policy, biosecurity policy of Australia. And I think on those lines we need, and we have tried in 2000, but that was half-hearted. Again, we have to take up, and Dr. Dubey was telling on that day that we are discussing, probably we will be having some new legislation, new legal system with the new robust biosecurity system of the country, so that we cannot face the COVID, we cannot face the far-like situation or other pests which have entered in the country. Or if they have entered, we can have some eradication plan. Thank you very much.
Okay, thank you very much, sir, for your nice talk on biosecurity issues related to national perspective. So, so participant may post the questions on your mail further if they have any thought related to it. Okay, so thank you very much, sir.
Welcome, madam. Thank you. Thank you very much. So this is the last lecture today?
No, actually, Dr. Shivkumar Agrawal from ICARDA, he will deliver the last lecture, and we will be starting, I think, in 5 minutes. Let him join.
Yeah.
This will be going also one of the important lecture, and interesting because he will be presenting some international perspective. So we'll get back as soon as he will join. Till then, if any question regarding anything, regarding the organization of the training and anything, if you want any clarification.
हाँ, आपका फोन अटक सा मेरा लेक्चर चल रहा था, बताइए please.
हुडा सर एक बार म्यूट कर लीजिए अपने आप को। मैडम
can you come to my room?
Huda sir, please mute yourself.
I'm forwarding this one on the WhatsApp.
आप होस्ट भी नहीं
Number as well as the address. ठीक, भेजता हूँ अभी। Thank you very much डॉक्टर साहब। 8:30 पे आप ब्रह्म प्रकाश जाएंगे ड्राइवर। Morning 8:30. Okay.
वो म्यूट करना।
Can I speak, ma'am? Hello.
Sure. Who is there?
I'm Dr. Suma speaking from Dharwad, US Dharwad. Actually, the lectures have been very good, but we tend to forget whatever we have heard so far. We are trying to make some notes as far as possible, but it would be helpful if you share the presentations, at least whoever are willing to share.
We will certainly share whoever are willing to share, but I don't think most of the person are not willing to share. Okay? This is for not just a speaker, this is for us also. If I have to share my presentation, I will not prefer to share, to be very honest. That's why we will share you with the compendium. And if somebody has shared, only one person I think shared, isn't it, Jyoti madam?
Only 1% till now.
Share. We will share you, who all will share, if PDF and anything. Otherwise, beside this, we'll be also sharing the compendium with you also. That's we are working.
At least the references, if you have in the compendium, we can revisit and make use of them.
Yes,
Sure.
compendium, all the informations will be there, so you can make use.
Okay. Thank you so much.
Okay.
एक बार मैं सर को रिमाइंड करा देता हूँ।
In the meantime, I would like to announce, although we will be posting in the group, so tomorrow we have an interactive session with Dr. R.S. Paroda. So everyone is requested to join by 12 noon. So there should be 100% attendance in that class. So it will be a good interaction with Padma Bhushan, Dr. R.S. Paroda. So please join. There will be morning session 9:30 to 11:30, and then 11:30 to 12 there will be a break for half an hour, and then all are requested to join for that session. So that will be a good interaction.
Okay, ma'am.
Good afternoon, sir.
Very good afternoon.
We welcome our speaker of today's lecture, Dr. Shiv
Good afternoon, sir.
From
Good afternoon.
Sir, just now I really feel privileged to have an opportunity.
Welcome, sir.
Okay. First, because sir, you are very famous, that's why everybody is willing to welcome you personally. So no way. So I really feel privileged to have an opportunity to introduce our today's speaker, Dr. Shiv Kumar. Just I am sharing the introductory slide. Ma'am, is it visible?
Yes, it's visible. Just
yes.
Fine. So ladies and gentlemen, Dr. Shiv Kumar holds a PhD in plant breeding from the Govind Ballabh Pant University of Agriculture and Technology, Pantnagar. His area of expertise includes crop improvement through resistant breeding, widening the genetic base through pre-breeding, and development of genetic and genomic resources in food legumes. His enormous contribution in identifying donors for new plant type in rice, extra early genotypes in chickpea, lentil, and mung bean. Basic information on genetics of important traits and pre-breeding populations in Vigna, Lens, and Lathyrus species are noteworthy. During the last 33 years of professional career, he, in collaboration with national partners, has contributed to the development of 51 lentil, 5 mung bean, 2 urad bean, 1 rice, and 3 grass pea varieties. He has published 290 peer-reviewed journal articles, 87 book chapters, 11 books, 7 technical bulletins, and 2 training manuals. He is also recipient of Fellow of professional societies and prestigious academies, including Crop Science Society of America, International Legume Society, Indian Society of Genetics and Plant Breeding, and Indian Society of Pulse Research and Development. Dr. Kumar has received many academic distinctions, fellowships, including Rockefeller, and awards from national and international organizations, societies in recognition of his contribution in food legumes. Presently, he is leading ICARDA's South Asia and China regional program from New Delhi, India, which aims to deliver improved germplasm of lentil, Kabuli chickpea, faba bean, and grass pea to national partners in South Asia and China. As a breeder, he works on developing short duration climate smart varieties of lentil and grass pea with high iron and zinc content of sustainable intensification of cereal-based cropping system. In a nutshell, I must admit that he is a great supporter of legume research in India. His selections at ICARDA led to the development of modern ruling varieties of Indian pulse breeding program. So with this, I would like to request Dr. Shiv Kumar to share your thoughts and views. Thank you, sir.
Thank you very much, Kuldeep. Very elaborate, introduction. At the same time, you have put my, photo of very young, man. So I have grown, in shape and size. But anyway, thank you very much, because I know many of them. I know Jyoti. We have worked with Jyoti, when we were together in IIPR Kanpur. And of course, many of them, Dr. Pyasi, many of them I know. So I will not go to, in detail. So thank you very much for inviting me to, present our, efforts towards accelerating genetic gain impulses. And it's so, let me share my presentation. Can you see my presentation?
Yes, it is coming. Sir. Yeah. Yeah, okay, sir, fine.
It is there?
Yeah, fine, sir, fine.
Okay. So today we are going to talk about, accelerated genetic gain impulses. As a breeder, when I joined, DRR, at that time, now the name has changed. So we were making, we are making our efforts toward genetic gain, but we have never realized in terms of really measuring it. We never used to measure, what we generally used to do. We make a cross, then we look for a particular traits, and when the traits is there, then we find the fixed line, and these lines were evaluated in AICRP trials, and then if it is performing at least 10% higher yield, we used to release. But now, in the modern time, this kind of approach has been changed. Nowadays, if you want to get support from any of the donors, the first thing they want is what type of genetic gains you are making in your breeding programs. So if you do not have answer to that, if you just say that 10% yield increase or 5%, they are not very much convinced. Though it doesn't mean that we have not made the progress. We have made the progress. But today we have to think in terms of the real applying science, and in terms of giving them proof, yes, that there is a genetic gain, not only in terms of the yield, but also in terms of the traits for which we are breeding our a variety. That's why if you just compare between the private seed sector and private seed company and this public sector planting programs, you will see that the kind of gain this private sector has made is much more higher than what as a public sector breeders we have done. It was mainly because we were working on many things. We have never had a kind of variety replacement strategy. We never had a kind of approach having this market information, which kind of varieties are required, what are the deficient traits in a variety which is most popular, and how we can replace that. So we were not taking those things. We were never involving socioeconomists in our breeding programs. We never had a market intelligence people. The similar is never we had this gender specialist. But now things have changed over the years. But in the last 10 years, I can say that these are the new kind of approach which is coming. So today we will be talking about how at ICARDA we have worked to accelerate the genetic gain in pulses. And then one can ask why we want to go for accelerating the genetic gain. As you know that if we have to feed the growing population, which we expect that it will be 9.2 billion by 2050, and which is almost 37 more mouths than what we have today, then we must increase our food production by at least more 60%. And that too, when there is a challenge from the climate change, when if I take into account extreme weather, sea level rise, and agriculture productivity losses because of these two, then one can see in this global map the South Asia and Sub-Saharan Africa. These are the two regions which are going to be affected more. And by one of the estimates from the FAO, it is said that by 2050, the crop yield will decrease by 25% if we do not address the climate change now. So under this scenario, we must think in terms of how we can accelerate the genetic gain if we have to feed this growing population. As you know that, as far as pulse is concerned, around a dozen pulse crops are being grown globally, and they are being grown in 92 or 93 million hectare area, and giving us protein-rich grains almost 88 million ton. If you just go through the data available on the FAO state, then you will find that the area under two crops, pea and fava, it is decreasing. But then at the same time, the productivity in these two crops has gone up very high. If I take into account the beans, chickpea, lentil, cowpea, and pigeon pea, the area is increasing and productivity is also increasing. And as a result, what happens that if you see the overall production of legumes, I think it is increasing since it's data available at FAO. Then, as you know that we have released almost 3,500 varieties of major pulse crops globally. And this is the old data. If I just add of the new data, then it is coming to 3,700 varieties. And out of that, more than 1,200 are released in India. And these varieties are not only high yielding, but also having nutritionally rich. Many of them are biofortified with iron, zinc. And at the same time, some of them very high adaptability, particularly like, IPM 2-3, which was, adopted across the seasons as well as across the region. If I take data of only, ECARDA, how we have released varieties, you can see in this map, in many countries, we have released around 85 varieties in 10 years period. Now this data has to be updated. And then these varieties, if you see, they are not only having high yield, but also having some of the traits, which are very, very important. Like in the case of chickpea and fava, they are machine harvestable. In the case of lentil, they are extra early, maturing. At the same time, they are also rich in micronutrients. At the same time, now we are also releasing some of the varieties soon, which will be having a kind of high biomass, because in North Africa, lentil is not only for food, but also as a kind of dry extra for the animals. So now we are working on that. So, but then if I take those varieties, then you will find that the genetic gain in these crops was very limited, even after having so many varieties. And when we have worked out the genetic gain, it is only 0.6 to 0.8%. And if we have to meet the future demand of pulses, it must go up. It must go up to 1.5% at least, so that we are able to meet the future demand of pulses globally. We have done some of the studies in country specific. For example, in Ethiopia, we have released around a dozen varieties of lentil, and then we have conducted this very experiments at 2 or 3 locations to estimate the genetic gain. I should say this is more of yield gains instead of genetic gain. We found that we have made only again 6.6 to 0.8% per, and that yield gains was more associated with higher biomass and harvest index. The similar is the case with your Morocco, and so it's country specific historical data has helped us to see that still we are lacking in our genetic gain. As a breeder, what we generally do, we always look for increasing the trade value. Each cycle of selection must give us a higher trade value than what was in the previous cycle of selection. And that's how it, when we are dealing with the quantitative traits, then it becomes much more improved. We can't have a perfect varieties, and no breeder can breed a perfect variety. We can only add value to the varieties, from the existing, as compared to the existing variety. That's how we are able to make continuous, gain, incremental gain in yield or any traits, particularly those which are inherited quantitatively. So then question comes, how to accelerate the genetic gain? So as you know that this is the classical, breeder's equation, we call it, and that is about the genetic gain, and that is about the heritability, that is about the time taken for the, in the generation, and then also about the selection accuracy and intensity. And of course, all these things works when we have enough, phenotypic variability in the population. So if we see that if we have to improve our genetic gain, we must act in one of the four these, the part of the equations. For example, if we have to go for higher selection accuracy, we must improve the heritability in our population for the trait. It means we have to reduce the environmental, and interaction part and error part. When we talk about the, selection intensity, that when we say selection intensity, we are saying about the proportion of the population which we select from the available population to have to carry forward them to the next generation. So if we have to increase the genetic gain, we must think in terms of optimizing our breeding scheme. It means we must think in terms of the choice of the parents, also in terms of the type of crosses which we want to make, and also then how we can increase the selection accuracy, intensity, and how we can shorten the breeding cycle. So we will concentrate our discussion today on these points. If I say that if we have to accelerate, then we have to modernize our breeding population. So as I said in previous slide that these are the four components which are very important. So if I talk about the generation interval, then we are talking about either in the past we used to do subtle breeding, and then recurrent selection. Nowadays we have another options like genomic selection, which can also shorten the this generation cycle. Then speed breeding, it is another very good example of taking 3 to 4 or 5 generations per year. Then when it comes to genetic variance, earlier as a good breeder and in the private sector, they always go for best into best to get the best. And by that one can say that what we are doing, we are just reshuffling the same kind of alleles. Means we are not bringing the new kind of alleles. Then breeding programs have diversity, but sometimes that diversity may not be very useful if we are not going to bring in new alleles into that parent crossing block. Then we need, for that what we need to bring the new exotic diversity. That is where the crop biorelatives comes into the pictures, as well as the land races. They also becoming very, very important nowadays. Then of course, if you go for genotyping, parents and post collections, that will also help us to see what kind of genetic diversity we want to bring into our breeding programs. And then selection intensity accuracy. And these are some of the components, like we must think of having a very high throughput, precise phenotyping. We must think in terms of the analytical pipelines. We must have a very efficient, experimental designs. Earlier, we used to have this, RBD and RBCD designs. Nowadays, you have number of designs there you can, because every plot cost, and then we have to make our breeding cycles more efficient. Then we must reduce the cost of the, our evaluation. And for that, nowadays, partially, replicated designs are available. That is very much being used, in our breeding programs. Then we must understand the target population environment. Earlier, if you are not able to characterize the environment for which you are breeding varieties, then you cannot get varieties which really match the requirement of that particular environments. Therefore, we must have a very good understanding of the target population environment. Then we also have a large population and few processes, or we should have more processes or less population, because our land resources are very limited. Generally, earlier, I remember I used to make large number of processes and having a small population per process. I think that doesn't work when we go, when you really reach to a certain stage and you really want to make incremental increase. Then one has to think, because when you have large population or when you make a cross, what you are looking for? You are looking for a rare of the rare, recombinants, and which is one in thousands. So for that you must have a large population so that your selection intensity can be improved. And of course, now genomic selections available and data digitalization is very important. Earlier, our breeding program was less governed by the data. Nowadays, the whole breeding program is driven by the data. If the quality of data is good, then your selection is better. If you don't have the quality data, then it is more of a kind of your perception about the good or bad. Then other is operational efficiency. That's also very, very important. Like number of the process, how many locations you want to test your your breeding program, breeding lines, and then genomic selection again that can help into operational efficiency, barcoding, mechanization, and data digitalization. I tell you, all these things brings in efficiency. For that, what you have to think in terms of standard operational procedures, SOPs. That is very, very important. And why we want to do all these things? Only the key drivers are your time, cost, and precision. And once you act on any one of them, then you will find that somewhere one of the drivers has been implemented into the whole this equation. So first thing is about efficient mining and use of genetic resources. Most of the gene banks are having very large population. Let us say in the case of Ekada, we have more than 70,000 accessions of legumes. But then, can we evaluate them in one go? Though NVPGR has done a good job by evaluating a large number of accessions at many locations in the case of wheat and many other crops. But in international centers, I think having that kind of programs, it's very, very costly affair. Therefore, we always think of in terms of having a smaller set of germplasm which can be evaluated, schemed more precisely and more more effectively. So we do that, and then for that we comes out different kind of fixed set. I will let you know what are the fixed sets. Then we also think in terms of genetic diversity panels, magic populations, and of course then we also think in terms of the crop biolatives and the integration lines. And of course, then we also have the special intention nurseries. Only thing is, when we are talking about this germplasm, our major target is to precisely phenotype them. Then also bring those or integrate those traits into the breeding program. So one approach, you might have heard about the core set, mini core set, coming from EquiSet. As far as Ekada is concerned, we talk about fixed approach. That's called focused identification of germplasm strategy. In this, it's a very simple approach that if you can find a particular traits, only here there is a selection pressure for that particular one. So we go back to the gene bank and we see that, let us say we are looking for winter hardiness. Then we look for the germplasm coming from Central Asia, Russia, and Turkey. And then you will find that the number of accessions having winter hardiness is much more in that smaller set of germplasm as compared to taking any random set or taking the mini core or core sets. So that is what we do in our case. And we have now fixed sets available for most of the crops, and they are trait specific, and the frequency of identifying the germplasm having that particular trait is much higher than than as compared to the random set which we have done. So we have done that kind of study, and we could find that this fixed sets really works for some of the traits, but at the same time, it doesn't work for some of the traits. For example, for any laboratory stress, this is one of the best approach. Even laboratory stress, it works. But for some of the laboratory stress, it doesn't work. For example, escoped light in chickpea, we wanted to have this approach and it didn't work. So it has a very trait specific and also very crop specific in some of the cases. Second is, if you want to go for higher genetic gain, we have to think in terms of high throughput phenotyping solutions to increase selection accuracy. So at Ekada, we have established a very elaborate precision platform for phenotyping not only the above ground traits but also the below ground traits. And good part of this is that it is fully automated control systems. And then this facility can also be split into different kind of sectors where we can apply different kind of with different level of stress for water, heat, or anything, any other stress. And of course, then we can run not only one experiments, we can have multiple experiments at the same time. So that gives us a very good information to compare between the crops, and between the traits and between the populations. So that is very, very important nowadays, that because now we have a lot of genomic data available across the crop. Even though in past legumes were lagging behind, but nowadays even legumes are far ahead of many of the cereals. But then again, when it comes to phenotyping, I think legumes are still far behind as compared to cereals. And there is a reason for that. We have done number of studies and we found that if you just have a multi-location yield trials of legumes, let us say chickpea or lentil, and if you just take out if you divide the total variation available from here it is coming, you can see almost 70% is coming from your environment, and maybe 15 to 20% is coming from the G to E interaction, and it is hardly 5% or below that is coming from the genetic part. As a result, as a legume breeder, when you go for selection, visually, you are selecting not the variety, not the genotype, but you are selecting the environmental effect, and you are selecting this G to E effect. As a result, when you put up those selections in next season, you will find that the performance is different than what it was and for what you have selected that particular. So that's why we have to think in terms of having this high throughput phenotyping solution to increase selection accuracy in legumes. And we also have a very, a kind of, high throughput, phenotyping solutions. This is a movable, kind of equipments. We call it PhenoBuggy. It really allows us to dissect why one cultivar is performing better than others in a particular, climatic conditions. And then you can have as many data as you want, automatically recorded, and then all the data is, when it records, it comes to you. And that really helps us to have someone who is very good in analyzing the data. Now creating data is becoming very easy with all these automated solution available in our hand. But now we really need someone from the data manager who can manage the data, come out with a kind of inference from those data, analyze the data. So these kind of facilities are very much important for nowadays to have better characterization of germplasm as well as breeding populations. Then earlier as a breeder, what we used to do, we wanted to do everything. We used to have a core breeding, we used to do pre-breeding, and we also wanted to explore, your crop biologicals. Nowadays, many breeding programs are being, shifted into different compartments. Core breeding is led by a breeder, while this pre-breeding efforts are taken up by the germplasm managers or someone who's involved in the gene bank. And of course, then when it comes to the game changing traits, again it remains with the gene bank people who really contribute to that. So when I say core breeding, actually we are talking about making crosses between elite into elite. One can say, if we are going to have the same kind of process involving the elite into elite process, it means we are not infusing the new blood into our breeding programs. But then most of the private sector, they have been successful in giving you better variety only because of they were concentrating on into elite process. The question here, the crux of the thing is that how you select those two elite parents. That is what we are going to do. I will let you know in the next slide. So that is why once you have this, poor breeding, then you are just adding a particular traits. You are adding a traits to replace the existing variety from the farmer's field. And that is where every year, every cycle of selection gives, higher trait value, and that's what. And when it comes to the pre-breeding, it is confined to elite into land races as well as elite into introgression lines. Here directly we don't go for the CWR, but then it takes a little more time than what it used to in the core breedings. In core breedings, you can have a fixed line in 3 to 5 years, but in the case of pre-breeding, it may take much longer times, maybe 3-4 years more. But then that is where we target for the as a breeder, we must perceive what kind of varieties are required after 5 to 10 years. And that is where the pre-breeding's role becomes very, very important. That is where you are working to add value-added traits for future. But then many times we require the game-changing traits. Many things are not known to us. But when you involve our CWR, crop-wide relatives, then you can find that there is a, you can recover some of the alleles which are not there in your breeding program, which is not there in breeding populations. And that is why CWR, particularly under the climate change, has become much more important. But of course, then it requires much longer time, 8+ years. But then, as you know, that when we are working on CWR, we must have a long-term investment into that activity. And that is where many of the breeding program fails. If you divide your resources between these 3 compartments, then you will not come out with any a kind of a great success. Therefore, for this 2 compartment, that is your game-changing traits and future value-added traits, we must keeps on burning the we must keep them alive. We must put up more money on these 2 activities so that your breeding core breeding programs always have a new blood into the breeding cycle. So then I was saying that how you can find those elite parents, and that is possible if you want to find this hidden cross. For that you have to have a genomic assisted crossing plan. As you know that when we make a elite to elites, what we want to have the best combiners. If these 2 parents are the best combiners, they have to be complementary. They should not have the same kind of alleles. And then they must represent the genetic diversity for the traits you are making the cross. So that once you select based on the genomic informations, then you will find that the chance of finding the superior lines from a given cross, that is elite into elite, then it will depend on average performance of the progeny as well as the potential for transfer subsequence. So that is what we are looking for. Potential for combining different positive alleles from both parents. That is what, then that's how you get the transmission. So only from this graph, you can see how these 2 parents having average progeny, and when you go for this genomic assisted crossing program, you can increase the the performance of the progeny coming out from these 2 parents. Another one, now we are working on this marker assisted selection, particularly chickpea. Lentil we are yet to start, but in the case of chickpea, what we are doing, we go for selection at the very early stage. In F2 itself, we start going for mass application, and that is where what happens that we just take from each F2 SSD plant, we take whatever seed we harvest, we take a part of that, and that is where we though it is planted. After plantation, you find that only those lines which are having that particular markers, only they are taken to the full cycle. That is how you can reduce the cost of your operation, and that also increases saving money. That also faster. At the same time, it allows you to focus on those plants which are having that particular traits, and that's how you increase the selection accuracy. As you know that in general, we know that variety development is long process. It takes almost 12 to 15 years. If I take from trait evaluation to gene discovery to its deployment to varieties, it's a long-term process, almost 12 to 15 years. The question comes then what? The question is that breeding program must at a minimum systematically and incrementally release new cultivars which are superior for key traits. At the same time, they are also having the traits so that they have the potential to replace the existing varieties. So genetic gain or response to selection or genetic trends, all these are the common, means one can, all are the same actually. How genetic or breeding value changing over time in a population, that is what is about the genetic gain. So nowadays, what we do when we have this core breeding programs, elite into elite, though we make a cross and once you have this genotypic and phenotypic data available, we have to think in terms of taking elite germ plants into your crossing program as fast as possible. So now in F5 itself, we take them up at cross programs. Once we identify that they are having that particular traits, that's how you keeps on increasing the breeding value of parents. That's how it has become much more profitable, as well as much more efficient and much more I should say better genetic gain. So we have to think in terms of earlier what we used to do. We used to fix, then we evaluate at multi-location when it is released as a variety or not released as a variety. But then after 10 or 12 years, we used to bring that particular line or genotype into crossover. Nowadays, it has to be as fast as possible. Then earlier, that we used to have subtle breeding. So I give you one example of flower bean. If we want to go for 2 seasons per year, we used to at our main location in Lebanon or Syria, and then we used to send it for different locations for evaluation or screening against important traits, and then again bring back that population to the main station, and that cycle keeps on happening. So then we used to reduce our breeding cycle from 12 years to 7-8 years. But nowadays we have this speed breeding solutions, and that is where you go for rapid generation advancement. And currently at Ecarda, we have established a capacity of 100,000 or 1 lakh entries per cycle. We can do it, and this year we are expecting to increase it by 50,000 more. So we will have total capacity of 150,000 entries per cycle. And that is how now for cereal as well as legumes. It is not only for legume, but also it is for cereals. So that's how it is going on in Morocco as well as in Egypt also we have established. And in Lebanon also we are having a smaller facility for that. And good part of this speed breeding is that we are also screening our population from F2 onwards to F5. Every year we are also screening for diseases. In this system we also screen for foliar disease as well as root disease, and that has really helped us to enhance our genetic gain much faster than without having this screening. So now we have standard operating procedures for speed breeding, and we have published, and it is available for all the crops. One can and then one can see the how this disease assisted speed breeding is really working very well. So if I take into account only lentil, now under this rapid generation advancement, from seed germination to harvest, it takes only 50 days. And this 50 days means we can go for up to 6 generations. But if I also take into account of the turnover time, then we are saying 4 to 5 generations you can take. It means from F2 to F6, you can go in the same year. So that's how we are able to reduce our breeding cycle time. But then when it comes to evaluation, you have to continue with what we are doing at present. Of course, now we are using these genomic tools to characterize our characterize our germ plasm and as well as go for genomic selection. We have now training populations and they are very well characterized and they are very well characterized phenotypically as well as genotypically. And based on that, equations, we are able to go for selection using only the genome-wide markers. At least in chickpea, and now we are going to implement for lentil, fowlweed, and grass pea. In grass pea also, earlier we did not have this, draft genome sequence. We have got it, recently, and now we have also got the sequence 384, this grass pea diversity panel. So as a result, now we have a lot of genomic resources available, lot of genetic resources available. That's how we are able to get those genomic selection in place. That's how in the case of lentil also we have this genome sequenced, but the problem is that we are not able to have published. I think soon it will be published. And once it is published, then it will be, we will go for this genomic selection. And another one is the genome editing. That's also one of the, this is one technology I really believe is going to change the whole breeding, system. How we are breeding today, it will change dramatically. Because there is one tool which is very precisely, you can discover and deploy particular traits. In the case of grass pea, we have been very fortunate to have a collaboration with the Queensland University of Technology, Australia, and Jonin Center. And as you know that in grass pea, this ODEP content, it is a kind of plant toxin which is not good for health, and this is controlled by 3 genes. So we are targeting those three genes called ODEP synthase, LSAE3 and LS-Cas. Once these three, we have already have this guide RNA and construct designs, and based on that we are able to come out with our T2 stage plants which are having a zero ODEP, but then it needs to be tested many locations. It has to be tested in many labs also before we really announce this particular things. So this will be one tool which is really going to help us to go for accelerated genetic gains across the crop very soon. Then it comes into bringing the new genetic diversity into breeding programs. That can come through breeding efforts. And we at ICARDA, we are very fortunate that we are our main station is in the Fertile Crescent region, and that is where most of the wild relatives have evolved, domesticated, and now we have in the cultivated forms some of them. So at ICARDA, we have been extensively using these wild species in our breeding programs, particularly in the case of lentil, it was Lens orientalis, Lens cervoides also we have used it in our programs. And in the case of grass pea, we were not successful in making crosses with wild species or crop wild relatives in the past, but last 3 years with big support from the crop trust, which is a global crop diversity trust, we call it, and we have been getting support. And recently they have again given us a project for 10 years, in the name of BOLD, and that is where we are going to take these pre-breeding lines into the farmer's field. So in this, I just want to show you an example how we have been very successful in transferring this high iron zinc content into our cultivated lines using this Lens orientalis. One can see there is a very good improvement not only in terms of iron zinc but also in terms of the yield advantage. So in many crops they have a kind of impression that if you improve iron zinc, then there is a penalty on yield. But my studies and my our experience says that there's no kind of negative kind of correlation or a kind of so I have not seen. In fact, I have seen many of them going up. So other things, which is very, very important in a breeding program is to have a digitalization. And we have to have a data-driven breeding populations. first thing what we have done now, most of our bidding programs are in the bidding management systems, and it is fully integrated with BMS. We must have a seed inventory, and we must have quality control, and all these things are in place. So now we have a very well characterized seed inventory, and it is also online. So if let us say we are working on 4 locations, so if the seed is available in any one of the locations, then we need not to multiply the seed at other place. Instead of that, we can just ask other one. Sometimes earlier we used to have seeds, but we don't know it is there. So we simply write an email, kindly send this seed. So nowadays it is changed. And as a result, now that cost has gone down because of the carrying cost is very high. But since we have a very optimized kind of seed inventory, the cost has gone down, very low. Then most of the observations are now driven by barcoding, so that and the moment our assistant goes into the field, once he record data, that is made available immediately to the terminal. That's how it is. And then we also have an analysis pipeline, so then you can analyze. So this is how you have to work nowadays. And you cannot do everything yourself. Always have a connections. Having like, for example, most of the data which I have, I generally put them into Germinate. That is UK based locations. So once you put your data in Germinate, then that is made available to everyone. Of course, you can also put a restriction till you want that it should not be in public. But I always find that sharing is always more beneficial to bidder than not sharing your data, because there are some specialists who can analyze your data is much faster way, much better way. They can give you much better interpretation of your data. So it's always better to have a kind of partnership and collaboration with other advanced institutions. And then one must have a standard operating procedure. For example, in the case of lentil, you have a SOPs, then how you should make the cross, what should be the plot size. Every information is available. That is also available online. One can see in the case of, and here you can see you have to mechanize your field operations. One can see here how precisely the plants are put into the plot, and then you have to have a very good kind of machines, farm machinery. That is very, very important for So after all these things, we could see that we were able to accelerate the building in the genetic gain, though it was still not up to 1.5% as we desired. But of course, in the case of chickpea, we have gone to up to 1% plus, and that is very good. And this is what we have to report to the donors. Like in our case, we used to get funding from the Gate Foundation, and then we report them, and that's how our support to breeding programs continues from these donors. So as you know that legumes are very, very important, and legume market is increasing. I tell you that nowadays how many companies are really working. They are not only working for this food, feed, or seed. They are also working on the ingredients of pulses. They are extracting protein, starch, and fibers. And they are saying this particular market is in this, last year it was 20.7 billion US dollar. And it is still growing at the rate of 4.5%. It is expected in the next 5 years, it will be around $26 billion. So one can assume that in future, this lentil breeding programs is going to have a much more impact on that, because if you really go for accelerated genetic gain. So in conclusion, I can say that conventional plant breeding are nowadays fully integrated with technology innovations, and their changes are happening very rapidly. So as a breeder, you must make sure that you must integrate your breeding programs with the technological innovations which are now made available. I'm sure that many of you may be knowing that accelerated breeding initiative of 1CGIR. That is, here every month we have 1 or 2, kind of, lectures where they give you information on how to accelerate the genetic gain. That is where you should get connected with this accelerated breeding programs. And they also provide to our national partners different kind of trainings. You know, for example, as a breeder earlier, we never used to go for selection index. You know, so you can say how you can develop the selection index, to have a kind of integrated approach towards selection. Earlier, we used to go and see one traits at a time. But by using the selection index, you have a kind of a holistic approach towards making selection. And of course, for most of the legumes now, genomic maps and are available, and that is where you have this genome-wide markers available, step markers, and that is where you can think of going for genomic selections. These are a few things which are very much required for before we go for genomic selections. In some crops, genomic selection is, I should say, is good. It is giving much higher kind of return, but in some crop it is still not that much I should say worth applying. And again, it depends on the traits also, which traits we are working for. That also depends on whether we have a better approach, multi-modal approach or not, when we are establishing this training population, whether this training population is evaluated phenotypically at many locations, or it is only one locations. So these are all matters. The size of the number of the markers which you want to employ, all these things matters when we go for genomic selections. So it is still evolving. In some crop it is very beneficial. In other crops it is still not. It's still some breeders are not very much convinced that it is going to give them return. Of course, the rate of genetic gain will be faster if we take up all those things which I have just talked about. Then product development life cycle will be shortened. Generally, you might have seen our Indian breeding programs that variety release, let us say 90s, still it is there in the seed chain. But now, the last 10 years, this scenario has changed. But in still many countries, Ethiopia and many countries, when I go, I see that the varieties which I have seen in 2009, still it is the leading variety. But once you have all these things, once you have a variety replacement strategy, then the product development life cycle will be shorter. So it means you will be having, you have a much better variety, in a shorter, means variety replacement rate will be much higher, much faster. The scaling of breeding programs will come from the integrated use of germplasm knowledge. That's very, very important. Sometimes we ignore this part as a breeder. High throughput genotyping capabilities, we need not to do ourselves. There are now, you just, send your samples to any of the company and they will do for you. So I need not to just think that you will do these things. Just take the advantage of the companies which are carrying out this services. Then we have to improve phenotyping. I think that is where also again there are some companies which can phenotype your germplasm, but here I always believe that as a breeder you should be involved in that. You must see that in the field. That's very, very important if you really want to have a feel of the breeding programs. Development, deployment, modeling, and prediction methods. Now these are also another part where, as a myself being conventional plant breeder, I could not get hold of, but I have seen that this is very, very important. This simulation model help you to predict the genetic gains. They also help you to predict what type of selection intensity you should employ. So this very, very good area where we should get ourselves acquainted. Then breeding by design, as I was saying, that by this genome editing, that is what will happen. Breeding by design, that will change the whole process of developing new and high performing varieties. So with this, I come to the conclusion of my presentation. So I'm thankful to all of you. And now we can have at least 10 minutes for discussion. That's why I made my presentation of around 50 minutes. So 10 minutes. Let us discuss, so that we can know more about accelerating the genetic gain in pulses. Thank you very much.
Thank you very much, sir, for giving a very detailed account of ICARDA and legume research and new technologies like high throughput phenotyping platform, modern tools, digitalization of the data, and many big data platform. You have really enlightened us, and I must say that it is a treat for all of us, and always it is a big source of motivation to listen people like you. So with this, I'll be taking a few questions, if any, and please feel free to interact. Sir, shall I take questions from chat box? Okay. There is one question from Dr. Chandrakant. Any practical measures or suggestions to get genetic gain or yield gain while breeding for earliness?
See, in the case of earliness, then you have two different approaches. Earliness, that there you have to go for the genetic gain in terms of the earliness, not in terms of the yield. If you mix up these two traits, because in many crops, you can reduce the crop duration to certain extent without having compromise, without having yield penalty without any penalty. But let us say, if you, in the case of lentil, if you reduce your duration from 160 days to 120 days, there's no penalty. And between 120 or between 90, there's a slight kind of penalty, but still it is manageable, because there you take in terms of the system productivity, gain in the systems, because there you are fitting into the system. You, let us say, when we want to go for extra short duration lentil, we are fitting them between two rice. So what happens? The system gain is there. It is a kind of bonus to the farmers between these two main crops. So you are making very good genetic gain in terms of yield in these two rice crop. But in the case of lentil, you are getting a kind of bonus. It is a kind of game-changing traits that you are bringing one crop which cannot fit into that, but you are fitting it. So whatever is there, it is a genetic gain. It's a complete, a kind of gain which you are finding. So of course, there are two ways. One is genetic gain in terms of only yield, and other one is about the traits for which you have bred. That's why there are 4 different methods of genetic gains. One is historical, a kind of genetic gain where you have the, you have a set of varieties raised in different era. We call it era genetic gain. Okay, so that is based on the overall performance of the varieties. But then there are other traits where you take a long-term check into involving, based on that you can work out the genetic gain. And other is, which is real genetic gain, is your breeding cycle. Means when you make the parents, what was the breeding value of the parents? When you take out parents from that cycle, and when you infuse into your breeding program, then how much breeding value is there? That is actually the real genetic gain in terms of breeding programs.
Another question from Dr. Suma. Poor seed filling and chaffiness is a major constant in lentil breeding. Kindly throw some light on resolving this as a breeder. And she was also requesting for sharing of some elite genotypes from ICARDA.
What one word she used?
Poor seed filling and chaffiness is a major constant in lentil breeding. Kindly throw some light on resolving this.
Actually, maybe because in our Indian conditions, because of this grain filling period, in the there is a like forced maturity. So when there is a forced maturity, then you can have a this grain filling problem. But as far as our program is concerned, I have not come across much of this problem while breeding varieties for lentil in micro environments. That we have never come across this problem. But of course, because in the case of lentil, the grain is so small, and then the pod, so in which environment you are working, Suma?
Sir? Sir?
Yes.
Excuse, sir, I didn't get you, sir.
I'm asking you in which environment you are working on lentil, you know?
Yes, sir. Yeah, yes. Sir. As you rightly pointed out, this problem we are seeing of late, sir, due to changes in, difference in maximum and minimum day temperature, sir. This is fluctuating a lot, and untimely rain is another problem, sir, that may be affecting the pollination and fertilization. Since 2-3 years, since 2019, we are seeing this, sir. Otherwise, we didn't have a problem of seed filling, sir, earlier. I think this is due to climatic factors only, sir.
Yeah, you are right. See, if there is a heat stress, you will find the number of grains are reduced. You will feel, you know, maybe 30 to 40% or even 60% pots are unfilled, and only few pots are filled. But then the grain size increases, you know. And in the case of drought, it is overall the seed size, reduces, and then maybe number of seeds may not be reduced, but of course the size increases. In the case of heat, when you have heat, you always have a kind of heat and drought coming together. You know, if you have a kind of separate experiment under control condition, you will find that heat reduce the number of grain, and in the case of drought, it is the size of the grain which is reduced. But in the field, you will, since both comes together, it is, you will find that there is some unfilled grains.
Actually, sir, the same genotype is behaving differently over the years. So to release a variety, it's taking too long because we're not able to zero down on consistency of the performance of a particular genotype, sir. So I think physiologists have to address this problem. But at least in collaboration with physiologists, we breeders have to resolve this problem, sir. Because if we release a variety and if it starts behaving like this in the farmer's field, then we'll be pelted. That's the problem, sir.
Yeah, you're right. If you remember, I was saying in one of the slide that we have done some meta-analysis, and we found that if you just take up all those, yield trials, total composition, then the total variance almost 70% plus is coming from the environment. It means if you are in a heat and drought one, then how much it is affecting, So performance from year to year will change, isn't it?
Yes,
because
yes.
it is only 5% which is reflected into the variance, in your trial, which is coming from genotype.
So breeder's job is at a greater stake because of climate change, sir.
Yeah, that's true. That's true. So, but I think, with climate change, in many crops, our varieties are also changing very silently.
Yes. So is it possible to spare some sessions, sir, from ICARDA?
Yes.
I think we have to go through that MOU process and all that.
Yes, sure.
Should we get that from your website, sir?
No, you just send me an email and then
Yes.
we will see what we can do together.
Yes. Thank you so much, sir. Thank you.
Thank you. Is there any other questions? Anybody would like to ask any question? Okay. If no, then sir, I would like to thank on behalf of my own and organizing committee for sparing time. I know since last couple of days you are traveling through, from Delhi, Bhopal, and you have given a time. And really we enjoyed. I hope all participants have been enjoyed and they have benefited with your words. So with this, sir, thank you once again, and thank you very much for spending time and giving.
Thank you very much. Thank you very much, Kuldeep. Thank you to NVPGR, your director, and Jyoti, that you invited me, and I am now sitting in Bhubaneswar.
In chat,
Okay.
chat several messages, sir.
Otherwise, I would have been happy to be in person to
to
do this this training program. Thank you very much.
Thank you.
Thank you very much, sir.
Thank you. Bye bye.
Thank you, sir.
Thank you. Bye bye.
Any message for tomorrow? You can make now.
I told already that time during the break. So further we'll update in the chat for tomorrow's schedule. So everybody has to be online by 12 noon. So we have a very important session tomorrow, interactive session with Dr. R.S. Parada. So means the interested persons would interact with him.
And yeah, all questions should be very, straight. It's not like that you are asking this kind of questions, scientific questions. It should be related with the PGR education and training, some policy kind of thing. We will allow some person. Okay. With this, thank you. We'll see you tomorrow.